I don't know whether this works. Yeah. OK, thank you. Welcome to MLCB, day 2. So the opening keynote today will be given by Nilah Ioannidis from UC Berkeley. She's going to talk about personal genome interpretation.
OK, yeah, thanks for the introduction. Can everyone hear me well? OK, great. Yeah, it's great to be here. Thanks for getting up early for the first keynote of the morning. I'm going to talk a bit about how you can use the sequence-to-function models that we've been hearing about yesterday and very prominent in this meeting, whether we can use them for personal genome interpretation and some of the evaluations we've done in that area, and some ongoing challenges that I think highlight some important kind of creative work that can still be done in the field. And I think there's a lot of kind of different training strategies that people might want to try out to kind of improve performance on this kind of problem. So, OK. Since we've talked so much about sequence-to-function models yesterday, and including in the first keynote talk from John, I'm not going to go into too much detail. I just have a quick overview slide from a recent review that he was actually co-author on, just to remind everyone that you have kind of an input DNA sequence. And the point of these models is they're processing just sequence input alone. In theory, that could be any arbitrary sequence. But they're trained on endogenous sequences from the genome to predict kind of functional annotations that we have on transcription factor binding, histone modifications, and also gene expression levels from CAGE data or RNA-seq data. So we have these models that should be able to go from sequence to predict these kind of molecular functional properties of the genome. And these models have had good success in doing that. And the kind of standard evaluations that are done looking at kind of held out chromosomes and held out regions of the genome. And they've been very useful, as you've seen yesterday, in looking at what are kind of some sequence determinants of these molecular properties of the genome. And model interpretability techniques have really shown us kind of helped highlight certain motifs and regulatory grammar that's involved in these processes. So these models have been really useful for studying endogenous genomes. So the question is, can we also use them to understand kind of variation between people? And that's what kind of my lab's perspective is on these kind of models and where we're coming from. So I'm coming from this background in kind of clinical variant interpretation, worked originally with rare disease patients, and then in my postdoc did some work on methods for predicting the pathogenicity of individual variants. So in general, in clinical genomics, we have kind of a patient genome that gets sequenced, and we want to know, of the many mutations in that person's genome, are any of them causal for a clinical phenotype or disease? And so in the past, a lot of machine learning tools were developed with kind of manually curated features about each of these variants. You can say, is the variant in a conserved region or overlapping certain annotations? If it's in a protein, what kind of domains does it affect in the protein? And you can build a predictor of whether the variant is pathogenic. So this is kind of our ultimate goal, is to predict some kind of clinical phenotype from this personal genome. OK, but since we have these nice sequence-to-function models, we should be able to plug people's sequences into these models and predict all these molecular phenotypes. The question is, can we just take someone's personal genome, run it through something like Informer or Expecto or any other type of model, and get predictions of these various molecular properties that the models were trained on? And these can be kind of a proxy for whether there's any potential clinical phenotype that might arise. If someone's gene expression levels are disrupted, then we might be concerned about those variants potentially being causal. So this is especially important for non-coding variants. Of course, there's been a lot of work on protein coding variants that we heard about yesterday. But for non-coding variants, they often, if they are going to affect clinical phenotype, it would be through an effect on gene expression. So we kind of focused in on, we initially thought, OK, we can take some existing models, apply them to some personal genome data, and check for how well they predict gene expression levels in different people. So the way this set up is a little bit different from how the models are trained. If you think about it, so the way current models are trained and evaluated is you have kind of a single reference genome that you're training the model on, and you have different gene expression measurements or other molecular phenotypes, but here we're focused on gene expression. Different measurements kind of across different genes in the genome. And these different genes in the genome, like some are highly expressed, some are lowly expressed, they all have pretty different promoter sequences from each other. And the model can kind of pick up on what the sequence features are of these different types of genes with different expression levels. But this is, kind of the training strategy. You're training across genes, and a lot of evaluations are done by holding out genes from different chromosomes. Let's say we were now going to apply this to a person. So if we have kind of a different, slightly different sequence for the same gene now, so this is like the same gene as the one on the left, but a personal genome sequence that has a few mutations in kind of the promoter region for that gene. The question we want to ask, and if you want to use these models like in practice for understanding personal genomes, the question we kind of want them to be able to answer is to explain the variation across individuals. So like if this person has slightly lower expression level than another individual, can the models pick that up just from the genetic variation? And so this is a bit of a harder problem, right? So the amount of differences between individuals and expression level is usually smaller than between different genes in the genome. So it's harder. It's not what the models have really been trained on. And there's obviously some environmental factors as well that go into differences in gene expression between people. So one question you might say is like, how well could we actually do on this problem realistically just from the genome sequence? So to be fair to the models that we're testing here. And so we have kind of, for this problem, there are some baseline models that we can use that have generally been developed from kind of the TWAS community. So for transcriptome-wide association studies, people wanted to train some models to predict, like from personal genome sequences, what expression levels of different genes are. But these are just variant-based models. So it's a kind of very different conceptual approach where you take around a gene, so usually like a 1 megabase region around a gene. You look at all the variants that are present in that region, and then you just train a linear model across some training set of individuals where you just have a weighted combination of the dosages of each variant. So the dosages that that person has of each variant times some weights that are fit through some regularization. There's a commonly used version of this that's an elastic net model. This is a PredictScan model for people that have been involved in TWAS. And so this kind of model gives us some kind of baseline where at least we know that a sequence-based model or a genome-based model should be able to pick up at least as much kind of explainable variation between people as this kind of simple linear approach. But we do want to go beyond this, and hopefully the sequence-based models, the deep learning models, could do better, because these, since they're trained across observational data from individuals, they only include effects of common variants, because you can't really fit coefficients for the other rare variants. They're also linear, so there's no kind of interactions between variants or nonlinear effects. But they do have this nice property that they're useful for TWAS. OK, so this is going to be kind of a baseline model that we use throughout the work that I'm going to show. And this initial test that we did was to take kind of existing sequence to expression models and see how well do they work at this problem, and are there any kind of challenges we can highlight there. So this work was led by 3 undergrads in my lab who kind of collaboratively did all the work for the paper. And then we took this data set that's publicly available. Basically, we need variants from a bunch of individuals, and we need their paired gene expression levels. So we took the Ioannidis data set that's based on 1,000 genomes individuals. So they did some RNA sequencing of lymphoblastoid cell lines from these individuals. And they're from a few different populations, mostly European, but they also are from, there's a group from the Yoruban population in Africa. So we wanted to run like the individual genomes from these people through these models. And we focused on genes that, in the Ioannidis analysis, had been found to have a significant EQTL, so to have at least one variant that does affect expression. Again, to be kind of fair to only looking at the set of genes where we should be able to have, we should be able to pick up on some genetic signal. Okay. So this is just a cartoon of kind of the training strategy. As I mentioned, we have run each person's genome sequence through a set of sequence to expression models, and then we end up with kind of a predicted matrix of, for every individual and every gene, we have their predicted expression level from the model. And then we also have the measured expression levels from the Ioannidis data set. And so this is kind of a cartoon of the two correlations that we're running. One is if you look at a given individual, if you look at all of their gene expression predictions, you can compute kind of how well the model explains variation across genes for a given individual. And that's very similar to the original training objective for the models, where you're doing the same thing but for a reference genome. So the only difference here is that instead of a reference genome, it's a kind of personal genome with some variation. But you can still pick up on these kind of large differences between genes. And then the other evaluation is correlation across individuals. So here, it's for a given gene, how well are the kind of predicted expression levels for all the individuals correlated with the measured expression levels for the individuals. Okay, and we looked at 4 different sequence to expression models. So these are kind of a rough cartoon. It's not exactly to scale because the slide wasn't quite big enough. But roughly what the kind of input regions are around the transcription start site of a gene. And so you see there's a pretty wide variety of regions around a gene that are considered by these models. And they also have kind of very different training strategies. So we're trying to do this analysis and kind of, it's a model agnostic approach. We're checking if there's kind of any general principles that are true, like all the models that are trained in this kind of reference genome way. Okay. So the set of evaluations that we wanted to do. So first of all, just as kind of a check, we wanted to see if we do that correlation across genes that I mentioned. So for a given individual, correlation across genes, which we expect the models to do well on. The only difference is the personal genome instead of reference genome. So we just wanted to check that that looked OK. So this is kind of a histogram of, I'm going to use Informer as the example and the figures, but we have kind of similar results for all the models. So for Informer, you can see kind of this correlation across genes for different individuals. There's a pretty tight distribution, and it's all centered around about 0.55, the average correlation, which is reasonably in line with what we would expect from the performance of the model like originally reported. It's a slightly different data set, slightly different data type with RNA-seq. But this is kind of reasonable performance. OK, so then we want to do the same thing but across individuals. So now for each gene, can we explain variation across different people? And so here now when we compute this kind of other direction of correlation, you can see this is for Informer. Here is 0 in the black line. So the mean correlation is like very close to 0. So there's almost no signal to explain this type of variation across people, which was kind of surprising to us. We thought there would at least be some signal. Now it's slightly positive, right, on average. So that's good. They're like picking up maybe some causal variance. The other thing to know is there's kind of this wide distribution, which I'll get back to later. There's some tails of kind of genes that have very high positive correlation, but also some with very high negative correlations. So this is the Informer plot. And then if we kind of look at the same thing across all the models, so this is kind of summarizing those results. This is the correlation across genes for the 4 different models compared to, we also computed it just on reference genome with the Ioannidis RNA-seq data. And so that matches correlation across genes for people, matches same thing for a reference genome. But then for all of them, this kind of correlation across individuals is very low and close to 0 with a wide distribution. And just for reference, remember we had this kind of linear model based on this PredictScan approach from TWAS. And this is kind of the performance of PredictScan across genes. So it's not huge. It's around like 0.2, but we should, in theory, at least be able to get that far with this kind of sequence-based model, right? So it's like, why aren't we at least matching that performance? Okay. I mentioned the kind of tails of the distribution. So just some examples of genes that come from kind of the high positive or the high negative correlations are just shown here. So we do have these genes where you see that you have pretty good correlation between the predicted expression levels and the observed expression levels, and it seems pretty reasonable. And you see some cases, like in this negative one, where it looks like the prediction's being driven by one variant, let's say, with 3 different dosage levels for that variant. So it does seem to be like potentially picking up that there is a causal variant in that case, but the kind of direction of effect for that variant is wrong. Okay, so we wanted to dig into these results a little bit more and say, is it the case maybe that the genes that we're doing well on tend to have like stronger EQTL, so the models can pick up strong effects, but maybe not kind of small effects. So we looked at, for each of the genes in the data set, we checked what kind of the effect size and the p-value were for the top EQTL that was found by Ioannidis for that gene. And so we stratify performance by this top EQTL. Here it's by p-value. And if we look first just at the genes with the positive correlations, we see that there is a trend here, so that the variants that have kind of stronger EQTLs and stronger effects, sorry, the genes that have stronger effects, they tend to be kind of higher magnitude of correlation across the model. So that is good and shows that the models are kind of learning something reasonable about the effects of these variants, and maybe are picking up that some effects are stronger than others. But the interesting thing is that if we look at the negatively correlated genes, we also see the same kind of trend, just in the opposite direction. So there are genes that have very strong EQTL effects, but and they are getting a high magnitude of correlation. It's just a negative correlation. So this suggests that the models are kind of recognizing that there are important, kind of strong regulatory signals, regulatory variants present for these genes, but giving the opposite direction of effect to the genes, or to the variants. Okay. Then the other thing we wanted to check is whether the models are better at variant effects that are kind of close to the TSS versus far, because that's been noticed before by some other studies, that maybe these models kind of pay more attention to variation closer to the gene, to the start site. So we looked at kind of the location of the top EQTL. It's not a perfect analysis because top EQTL might not be the causal one, but on average, we expect to have like some signal from this. We don't see much of a trend. There's like a very slight trend. So this is the distance from the top EQTL to the TSS of the gene. And again, the kind of correlation that we find from the model predictions. And so you can see like very slight trend toward having higher correlations if the EQTL is closer to the TSS, but it's a pretty small effect. So I think that's not the kind of main driver of the issue, and you do have genes where the EQTL is very close to the start site where you still see this kind of negative correlation. So it's not that these incorrect directions of effect are only being predicted for far away variants, for distal variants. Okay. Then the last thing we wanted to check, because we have kind of predictions from the different genes, or sorry, from the different models, is we were wondering, so are the genes that have these negative correlations, so okay, the models are predicting, or at least Informer was predicting a negative correlation, so getting like the direction of effect of a regulatory variant wrong. Is it because there's something about that regulatory variant that makes it hard for the models to learn, in which case kind of all the models should show a consistent pattern of like which genes they have trouble with and which genes have these negative correlations. Okay, so we decided to compare all the 4 models to test this. This is just an example of one gene for 3 of the models. The Senji looks very similar to Informer, so I didn't include it for space on the slide. But you can see in this example, they're very strong correlations that are negative, for informer and expecto, but positive for expresso. So at least, in this example, the models are picking up probably a similar causal variant, or at least causal haplotype, but getting its predicted direction is different between the different models. Okay, and so this is just one example, but this seems to be generally a pattern. So we did all these kind of pairwise comparisons between the predictions from the different models. So Vicenji versus informer, expecto versus informer, and we plotted the correlation values for this cross-individual analysis for the two different models, and if they kind of all agreed with each other on the direction of effect of the regulatory variation, you would expect all the density to be kind of along the y equals x axis here. But we see this enrichment kind of along the negative direction as well, so the y equals minus x, which is kind of interesting because it could be that there was just a cloud around 0 with some enriched density along y equals x, but the fact that there actually seems to be some enrichment even for things with strong correlations, but to be these kind of opposite quadrants with opposite directions of predicted effect is telling us that these models seem to agree more with each other on the magnitude of the effect of this variation on expression than they do on the direction. So I think that's kind of one insight into what might be going on with the models and what some of the challenges might be. This is just the last kind of comparison with informer and expresso, and then again to the predict-scan style linear model, that you can see that pretty much gets the kind of positive directions of correlation because it's trained with observational data, so it should get positive for most genes. But there's kind of a relatively even split between whether informer agrees with the direction or not. A little bit of enrichment in kind of the upper quadrant, but not too much. Okay, there's a follow-up analysis that was done by 3 other students in the lab, and Aisha and Ruchi are here, so you should talk to them about this. This is the MLCB paper last year, where they trained a bunch of replicates of Vicenji models and did a bunch of analyses on the kind of uncertainty in the predictions from the different models. But one thing they tested was just, do we see the same pattern that I just showed, but for replicates of the same model, not even different models? And so these are just kind of models trained with different initializations and same architecture, same training data, everything. And you still roughly see this kind of X-shaped pattern that suggests there's some just kind of stochastic nature to what the models are learning that happens to assign a particular variant a positive or negative effect on the final output prediction that's somewhat arbitrary. Okay, so conclusions from this first part. So we have these existing sequence to expression models that are out there. If we try them out of the box on personal genomes, they have kind of limited ability to explain this cross-individual variation. They tend to agree with each other, as I mentioned, more often on magnitude than direction. So we think this is telling us that it's kind of easier for the models to recognize that there is a regulatory variant present, and harder to tell the direction. And this kind of makes sense if you think about what might be happening. If the models have learned what motifs are important, which we think they have from other kind of great work that's been done in model interpretability, they might find that there's, if there's a variant in one of those very important motifs, they realize that there might be something going on with expression, and they alter the predictions, but they're not sure, this particular C to A change, if it's going to increase or decrease expression. Okay, and to get at whether that is a challenge just because it's hard to link a local effect on transcription factor binding, let's say, with expression, because there's kind of various layers of regulation in between there, or if it's a problem just at the, even at the local prediction level, we think, one thing that would be nice to check is whether this direction of effect issue in the predictions is also true for the other molecular phenotypes, so for accessibility data or TF binding data. And there's some evidence that maybe the direction predictions are better for these kind of local properties, but it's not, we don't have the personal genome data for a lot of these measurements. We haven't yet kind of evaluated the models across the personal genome data where we don't have to worry about figuring out causal variance from QTL studies and things like that. So I think this would be a nice thing to kind of get clarity on whether there is a challenge also in predicting effect on local, these kind of local TF binding type properties, or if it's just linking those to gene expression. And depending which answer you get, I think that would kind of push future work should be more on, if you have trouble even with the local phenotypes, maybe you need to see more kind of sequence diversity during training to understand like effects of variance within motifs. Or if it's kind of this linking to genes, then we want to look at kind of hierarchical models where you're first making like really good predictions of the kind of epigenetic local chromatin effects and then linking those in kind of a separate model to gene expression. But just some kind of general strategies. Okay. So last thing is kind of lead into the next part is a kind of obvious next step for this type of analysis to say whether these models would benefit from seeing personal genetic variation during training. So it's like one way of increasing the kind of sequence diversity that they see. And what if we just kind of show them a bunch of personal genomes rather than just reference genome, because they're not currently trained on any variant data. So this is the next thing we tried. So I'm going to show kind of how well this works and also some existing, ongoing challenges that would be great for people to think about in the field. OK, so here we're going to take this personal genome data from the same kind of Ioannidis data set and fine-tune the informer model in this case. This work is led by Ruchir and Annika, who are here, so please ask them details after the talk. The way that we're setting it up is we have the Ioannidis individuals, which again were from mostly European, but also this Yoruban population. So that will come up again later in kind of different holdout splits that we do. And then for kind of computational time, we are focused on training a random subset of 200 genes taken from that 3,000 that have strong eQTLs. And so the kind of personal genome paired with the transcriptome information, personal transcriptome data for those 200 genes now is going to be seen during the fine-tuning process. But the other genes are not seen. OK, and we tried a bunch of different strategies for how to do the fine-tuning. So one thing you could do is just throw kind of every paired individual personal genome sequence, plugging in the variants, creating the personal genome sequence paired with its expression level. It could just be a separate training example. And you can just kind of throw them all together in kind of one giant batch of training data. And so that's this kind of what we're calling single sample training. Then we also tried doing a kind of pairwise training where we try to force the model to learn more about variants, like specifically than they might be able to learn from just kind of throwing everyone together. So try to force them to pay attention to variant effects. And so there we take pairs of individuals, and we try to take them, one from the highest quartile expression for a given gene, one from the lowest quartile, and set up a fine-tuning strategy where you're making predictions for both of those individuals and then comparing the predicted difference in expression between them. And then we also tried doing some joint training strategies too. So maybe if you're kind of forcing the model to learn too much about genetic variants, you start forgetting some of the things that were learned by the original informer model. So we have some versions where we train kind of alternating between the this pairwise personal genome task and then the original informer training tasks as well, just to make sure we don't forget anything. OK, so one example just for the pairwise regression approach that we have. So just to make it more concrete, what we're doing, this is a whole genome sequence data phased from these individuals. So we have kind of the 2 haplotype sequences for each person. So we take a pair of individuals, and we're plugging in, just for now, their SNPs. So just the single nucleotide variants. In the future, we'd like to consider indels as well. But for the kind of initial results that I'll show, this is all with the SNP data. So we plug in, kind of for each person, the SNPs present in their 2 haplotypes. We run, we run all the 4, so the 2 haplotypes from each individual through Informer, get a predicted expression level for the different haplotypes. And the way we're dealing with haplotypes is just then averaging the predictions. So we have kind of an average predicted output for each individual. And then we want to compare the predictions for the 2 individuals in this particular case. And so this is a regression version of the problem. We would just look at the predicted difference and the actual difference and do like a mean squared error loss between those 2. And we also have another version that's classification loss of whether the model can predict which individual is higher versus lower expression. OK. So a bunch of fine-tuning strategies. It turns out they all work reasonably similarly to each other. So I will jump into the types of analyses that we set up to test kind of with this approach. So there's a few different ways that we can hold out kind of people and genes to figure out whether this fine-tuning helps with certain tasks, not others, like what it is enabling models to do and what is still needs work. So OK, so the first thing we ask is just, if you do this fine-tuning, do the models generalize to kind of new individuals that you hold out? So it's kind of just the basic thing you would hope that the training should help with. So we have evaluation where we train, or we do the fine-tuning on these 200 genes on some of the individuals, a training set of individuals, and we hold out some individuals randomly from the data set. And we want to see how well do the models do on these held out individuals. And remember, the original Informer, right, had pretty low performance on that with this wide distribution of predictions. So just to walk through kind of these plots of what the results look like. So first, this is just on the left is the baseline Informer, so the unfine-tuned, kind of same as the original results that I showed earlier, where the kind of average performance is around 0.05, like we saw before. And then for comparison, this was the PredictScan, like ElasticNet linear model that we had as our kind of baseline in the previous analysis. OK, and then in the middle, these are going to be all the different kind of fine-tuning strategies that we tried. So there's the single sample regression, these kind of pairwise losses that we construct, either regression or classification loss, and then adding in that like joint training with original Informer tasks. OK, and we see the results for these are all like reasonably similar. There is some boost from doing kind of pairwise instead of just single sample training. And so by doing this kind of fine-tuning, basic takeaway is that we are able to kind of fix the problem with the original Informer and get it to recognize the kind of variants that are present in these individuals and match the performance of PredictScan. As you see, it's not really much better than that. There's like maybe very marginal improvement, if anything, for some of the models. So we've kind of recovered this linear kind of combination of variant effects, but haven't really gone beyond that. OK, so this is on these kind of randomly held out individuals. So the next thing we wanted to check was it could be the case that these models, even though they're kind of doing something similar to what this kind of linear PredictScan style approach is doing, that because they have this prior on what motifs and what the regulatory grammar is, that they're better able to find kind of causal variants within LD blocks, which means that if we were to hold out individuals from another population, this is kind of our best way of getting at this question of whether they are getting better causal variants. So it's been noticed that if you train a TWAS style model or like a linear model on individuals from one population and then you test on individuals from a held out population, you tend to have a drop in performance. This is because you're kind of just training. The original weights are just based on kind of observational data from the training population. There's often several variants that are in LD with each other that are very correlated that all look like, just observationally, that have some effect on expression. Only one of them is really causal, but these kind of linear elastic net regularized models don't have a good way of picking out which is the true causal one. And then when you go to apply the model in another population where the LD structure might be different, if you picked the wrong causal variant, it's no longer in as strong LD with the true causal one as it was in the original population. You get kind of a worse predictive performance then. OK, so we want to test if we run the evaluation this way, so we train only on the individuals from the European populations in this data set and hold out the Aruban individuals, do we have this kind of similar drop in performance that's seen for the elastic net models? So the results for that are that, yes, we actually do see something very similar. So again, the predict scan performance, the linear model is on the right. And you can see there this relatively large drop. So this is what I was talking about, that if you test on a different population with different LD structure than you trained on, you have a pretty significant kind of drop in how well the model can do at predicting expression in those people. So the obviously the original informer, can't do well at that at all. But then the fine-tuned versions also have this drop. We do see in a couple of cases there's a marginal improvement that it's small. It does seem to be somewhat reproducible, but it's not, I think, super clear evidence yet that we're getting more causal variance. But maybe there's some hint of that. OK, so then the next thing is we want to see whether this model is going, having been fine-tuned in this way and learning about the variants that were present in the training set, will it generalize now to kind of unseen variants and unseen genes? And we would like to ask this question of whether it's kind of learned something more meaningful now about genetic variation that's being captured in the new embedding space of the fine-tuned models that makes it more generalizable to new sequences, essentially. And we would like to be able to test this even in a given gene that we train on a bunch of individuals from that gene. So one of the 200 that were seen in the train set, but a new variant, so a rare variant in some person that variant was not seen. It's a little bit hard to run that test because we don't have many examples of people that share no variants with anyone else, right? So kind of as a proxy, we're just looking at unseen genes only and saying if the model is good at extrapolating to new variants, it should also be good at extrapolating to new sequences from kind of completely unseen genes. For those genes, obviously, it hasn't been able to learn anything about the specific variants present in those genes. OK, so we looked at performance on a bunch of genes that had not been in the 200 seen during training for these different models. And the performance is pretty similar to the original informer, which is not great. So you can see this is across all the fine-tuned models. The green bar now is the unseen genes. And it's reasonably consistent. The kind of predict scan style linear model just doesn't apply to unseen genes. Hopefully that makes sense to everyone. But if you just train a linear model based on observed variants in a gene, obviously it doesn't have any understanding of any new gene, just kind of by definition. It's only learned about identities of variants in the training genes. So, it's still, there's still this slight trend toward positive average that we saw with the original version of informer, but it's not any better after fine-tuning, which was a little disappointing. OK, so the kind of last thing we wanted to test here was whether these models that have been trained, the other possible benefit of these kind of sequence-based deep learning models for this problem over the linear approach would be that you can learn these kind of interactions between variants and nonlinear effects. So we wanted to test whether kind of any of that is happening in these fine-tuned models. And so the way we're doing that is making a linear approximation of each of the fine-tuned models. So for a fine-tuned model, you can do in silico mutagenesis and get kind of the ISM score for a given variant. So the kind of amount of delta in prediction you get from an individual variant one at a time in the input sequence from the fine-tuned model. So we can get these ISM scores for every variant that's seen in the population. And then for each person, we just construct a linear approximation of the fine-tuned model by taking that ISM score from the fine-tuned model times the dosage of the variant. So this is, for each person, just summing over all their dosage times ISM scores for all the variants that they have. And we basically get, both for the random held-out individuals and the separated Urban population holdout, we see pretty much identical performance between this kind of linearized version of the fine-tuned models and the full fine-tuned model. So at least the current versions seem to be not learning too much about these kind of nonlinear effects of variance, which is pretty interesting. So I'll give some conclusions in a second. A couple of things we wanted to check was just whether the kind of number of individuals that we're seeing, we have this data set that's like roughly 400 people, and with the test splits and everything we're training on, like around 300 people. So is that just not enough to see enough variation to kind of get the most benefit out of this type of approach? So we have just kind of a breakdown scaling of what the performance looks like versus the number of samples that we use for training. And we can see, this is blue is, again, performance on the kind of random held out individuals and the population split in orange. And the performance kind of starts plateauing, so we don't think you're going to get kind of drastic improvements in performance from adding more people. And part of the issue is you have kind of diminishing returns from when you add a new person, a lot of their variation is going to be shared with variation that was previously in kind of the training individuals. So as you add more and more people, you start getting like diminishing additional variants that are seen in the new people. So I think this is one challenge for if we're going to use kind of observational data, population-based data to kind of augment the model training. This is one challenge that we're going to run into. OK, so then the last thing we want to check is, are the models kind of just learning the effects of the common variants, right? So because this performance is plateauing, is it just that we learned effects for the variants that are common in these individuals and like not really much about rare variants? We did this breakdown where we looked at kind of the performance if you take, so in the test individuals, in the held out individuals, sorry, if you kind of plug in, so normally we're plugging in all their SNPs into the sequences that we're running Informer predictions on. If we, instead of plugging in all of the variants that they have, we plug in kind of only up to a certain allele frequency. So kind of there's a minimum allele frequency on which variants we plug in. So on the right-hand side, we're kind of only plugging common variants into these personal sequences and looking at performance. So we do see there is like a little bit of a boost at the beginning coming from the common variants, but there's still a tail of additional kind of improvement in performance from having the rare variants present in those individuals in the kind of sequence that's evaluated for them. So it's not that the models are just learning common variant effects. There's kind of also some signal being given to the rare variants, but you get kind of diminishing returns there. OK, so conclusions from this section. So first of all, if you fine-tune with this kind of personal genome, personal transcriptome data, you do improve performance on the seen genes, so you can correct this problem with the direction of effect of variants that the original models had, which makes sense because you're seeing kind of empirical evidence of what the direction of effect of a particular variant is. So you are able to correct that. So you could run this kind of model on new individuals in like a TWAS-style way and get reasonable predictions. Now, when we look at kind of the performance compared to these linear models that have been used in TWAS, the performance like only marginally better, if better at all. So we're not getting like a lot of the benefit of the fact that these are sequence-based, like full sequence-based deep learning models that should be able to pick up on kind of additional rare variant and interaction type effects. So we haven't quite realized, I think, the potential for the models. So I think this is an important area for people to brainstorm after this and like what the right way of making use of this kind of population data in training the models is to really start beating the kind of linear baselines that we have. And then this important point that this type of training, the fine-tuning does not improve the performance on the unseen genes, so the held-out genes. So this suggests that by doing this fine-tuning, even though we have now learned the effects of variance that we're seeing, that hasn't changed kind of the embedding space in such a way as to make it more meaningful to variance. So it's not that we've learned now something biologically more interesting in embedding space that makes application to a new gene do better, or like to a new arbitrary sequence do better. We're really just learning about the effects of those variants that we're seeing. And so we kind of consider this, maybe the models are essentially memorizing the variants that were present in the training individuals and not really taking advantage of that information. Final point, because these linear approximations to the models perform similarly, this is, on one hand, it was kind of like unfortunate that they're not learning more interaction effects, but it's also, if you wanted to use them for TWAS, it's actually convenient because the way that a lot of TWAS studies are run, if you have just a linear model for your gene expression predictions, it's much easier to apply that linear model to GWAS summary statistics and get kind of interesting associations with phenotypes without having to run predictions on individual level data from all the GWASs. So if we could boost the performance of these models a little bit, the fact that they have these kind of linear approximations would make them useful to be applied in TWAS. So I think that's kind of one positive thing. And if they do slightly better on the held out populations, that would make an improvement over a lot of existing TWAS models if you're going to run TWAS in diverse populations. OK, so these are kind of the main conclusions. It would be great to brainstorm with people on what kind of new, I think there's some more creative like training strategies, architectures to try to force these models to take advantage of this information. Like there is clearly information present in the population level data, right? We can see that certain variant increases or decreases expression. That information is there and like should be able to help teach these models something. But we need a different kind of, some new approaches for how to get them to use that type of information in a constructive way that is generalizable rather than just kind of memorizing the relatively small set of variants that we have in the population. So happy to talk to people more about that. I wanted to just briefly mention there's an upcoming talk from Shiron and Drusinsky from Katie Pollard's labs. They're doing very similar work to this, have some similar results, but have done some really cool analyses of kind of where the variants the fine-tuning models are picking up on, like what types of genomic annotations they're in, and have some evidence that they are finding more causal variants, which is promising. So stay tuned. In a couple of talks from now, you'll see very similar but complementary work to this. And then, yeah, last thing, if people missed any of the posters from our group, I'll just plug, some of the other projects. Students are still here, so go and talk to them about these other projects. The last one was based on another MLCB paper from last year that I didn't get to talk about in this talk, but does some cool benchmarking studies of genomic AI. Genomic AI models, both supervised and unsupervised, and trains some new DNA language models, and has some interesting benchmarking tasks that are kind of complex reasoning type tasks, like can these models predict conservation signals across the genome, which require kind of integrating a lot of information. So check that out if you haven't seen it already. And with just acknowledgements from people from my lab, and then there, as I mentioned, Katie Pollard's group and also Sarah and Maria's groups here are doing very similar work and have similar results, and we've been talking with them about this project a lot and shared a lot of ideas. So thanks very much to them as well.
Thank you so much. So just one quick question. Yeah.
It wasn't a quick question, but I will try my best. So despite the fact that we are still using really old practices for like variant effect predictions to understand the biology of variants, it still works reasonably in many cases. Like if a TF binding site is broken or if splicing is broken and so on, if the variant is kind of breaking some kind of average biology, we still see some nice results in some cases. But this is kind of a higher bar, basically resolving variation between individuals. But the community still interprets it as like, don't do any kind of variant effect prediction, like when they see these kind of results. How do you split between these two different but slightly related things?
Yeah, I think it's very challenging to reconcile, like what you're saying, that there are good predictions for individual variants and like what their effects are. I think often though those are kind of ignoring a direction of effect type of issue. So a lot of the clinical variant interpretation type models, what they want to do is just find, is this variant going to be clinically relevant? So it's just kind of a classification type problem. And it may be that here, where you're having to integrate effects from a bunch of variants, you need to know what direction of effect they have on gene expression in this case. And the model needs to be able to sum up all those effects. And if some of the directions are wrong, like even if it could correctly do that task and find the important variants, the effects end up canceling out if you're kind of assigning the wrong direction of effect. So I think that might be part of it. But also there may be like additional annotations that are useful, like conservation signals for predicting, just classifying individual variants of whether they are clinically relevant or not, that these models don't have access to, right? So they're not trained with any conservation data, and using something like that as kind of a prior to help do this kind of fine-tuning or training could be interesting.
Okay. Yeah, let's thank the speaker. Thank you for the great talk. Okay. So then now we have 3 fantastic short talks. The first speaker, where is the speaker, please? And the second speaker, please, where's Avanti? Maybe it's better to get ready there.
Hi, everyone. My name is Han, and I'm from David Kelly Group from Calico. Today, I'm going to talk about some of our recent work in doing regulatory sequence analysis of RNA-seq data via parameter-efficient fine-tuning of a multitask sequence model, Bozoi. So sequence-based deep learning models, they are, despite some of the challenges, they are still be able to accurately predict genomic profile coverage tracks. Such model has been extended to RNA-seq data recently, either as aggregated read counts or TPM values, or more recently, just as directly predicting the coverage track across genomic positions. So Bozoi is a recently proposed model from our group that takes as input a large genomic context, more than 500 kilobase per window, and then use UNet and transformer blocks to capture distal regulation and eventually predict epigenomic in addition to RNA-seq coverage tracks at 32 base pair resolution. So this model has allowed us to gain insight into both transcriptional regulation as well as variant fact prediction. However, this is kind of limited to the data that the model is trained on. So if you have a custom RNA-seq data set, what could you do? Training a new Bozoi model from scratch is very expensive. It takes a few weeks to train from scratch. A more efficient strategy is to perform transfer learning. And basically, we take the existing Bozoi model, which is trained on a large corpus of data across both human and mouse, across multiple cell types. We take this as the pre-trained model and use transfer learning techniques to adapt it to the custom RNA-seq data set you have. So the intuition here is simply that we learn most of the basic sequence grammar and biology in this pre-trained model. And in the fine-tuning or transfer stage, learning can be much more efficient. So naive fine-tuning approach is to update all the parameters of the model. However, as the model size scale up, this becomes kind of more prohibitive. So the fine-tuning approach we take here is called parameter-efficient fine-tuning, which the basic idea is to update as few parameters as possible while keeping a competitive performance. I think yesterday, some of the group has already mentioned some of the approaches they take to do this, like LoRa, which is a popular method. So many of these parameter-efficient fine-tune approaches has been proposed for these attention blocks, such as Hospi, the adapter, LoRa, and IA3. So the basic idea for these different approaches is to insert small adapter modules into the existing attention block. And during the fine-tuning stage, only the parameters of these small adapter modules are updated, while everything else are frozen. So this is usually less than 1% of the total parameters. So that's how they make it parameter efficient. So here we kind of compared benchmarks 3 different parameter efficient fine-tune approaches for the attention blocks, and kind of want to see whether they work well for our model, Bozoi. In addition to these ideas for the attention blocks, because conv layers are also quite important in our model because they pick up very important biological signals such as motifs. So you also want conv layers to be able to adapt during the fine-tuning stage. So here, taking the inspiration from the squeeze excitation network, we insert squeeze excitation blocks into after the conv layers as the way to kind of rescale the conv channels to do the adaptation. Another idea is to adapt the LoRa to the conv layer. So this is also proposed in Ye et al. Recently called low-conv, so basically LoRa for conv layer. And this kind of learning a low-rank decomposition of the kernel that's changing during the fine-tuning stage. So these are a combination of these conv layer adapters and attention layer adapters is what we are trying to experiment and see how it works out for our purpose. So next, we proceed to benchmarking some of these fine-tuning methods. Because the original Bozoi model is very large, here we constructed a lighter version, which we call Bozoi Lite. So the Bozoi Lite basically has the smaller context input size and also a reduced number of parameters. So this is mostly for the purpose of the benchmark. We transfer these pre-trained Bozoi model, Bozoi Lite model to 3 target RNA-seq datasets for benchmarking. GTEx, which is held out in the pre-training stage. HIFLIC, which is essentially RNA-seq dataset of WI38 cell line undergoing replicative senescence, and ENCODE TF knockdown dataset. These are knockdown of different TFs in the K562 cell line using either CRISPR-I or RNA interference. So these are our 3 example datasets that we transferred to. And so here is the benchmarking performance. First, by looking at how well do these attention adapters work. So to evaluate performance of fine-tuning, we look at the Pearson correlation between the predicted and measured RNA-seq coverage in the holdout test sequences. So we compare these parameter-efficient fine-tuning attention adapters to 4 benchmarks. One is training from scratch on the target dataset alone, which is the GTEx in this case. One is joint training from scratch using all the pre-training data plus the target dataset. So this is kind of a huge multitask dataset training from scratch. Then linear probing, which is the simple transfer learning idea where you just update the new head, where everything else, all the other parameters are fixed. And also, finally, full fine-tuning where you update everything. We can see that basically all the transfer learning strategy outperforms training from scratch, either alone or as a big multitask learning problem. So this shows the benefit of doing the transfer learning and kind of focusing on this target data set after this pre-training stage. Second, we see that the attention adapters approach, they outperform the simple linear probing, showing the benefit of the attention adapter. Then we can see that the model further benefit from adding these conv adapters too, in addition to the attention adapter. Here we're inserting the squeeze excitation adapter to 2, 4, and 6 conv layers, inserting the low conv adapter to 2, 4, and 6 conv layer in addition to the attention adapter. And we can see as we insert more of these conv adapters, we see a continuous improvement in performance. We also benchmarks in terms of specificity across tissues. So this metric is basically the correlation between observed and measured expression level after normalizing against the mean across tissue. So basically, we see a very similar conclusion. And here on the left, there's the benefit of attention adapters. And on the right, we see that adding conv adapters on top of the attention further improves specificity, actually closing the gap with full fine-tuning. So with parameter efficient fine-tuning, we only update less than 1% of the total parameters. So this reduces the runtime and memory cost as comparing to either training from scratch or full fine-tuning. We also benchmarked our approaches on the 2 additional data sets that I mentioned, the encode TF knockdown and the Hayflick senescent RNA-seq data set. And the conclusion is pretty similar. A combination of attention adapter and convolution adapter results in the best overall performance, while keeping the parameter number low and the runtime and the memory efficient. So next we want to see, so these transfer model can achieve a good performance, but what we really want to do with it is to, given a new RNA-seq data set with control and some perturbed condition, we want to be able to use this sequence-based model to drive some insight into biology, for example, like TF regulation, for example. So here we take this Hayflick data set as an example. As I mentioned, this is WI38 cell line undergoing replicative senescence. So at 7 different time points from proliferating to senescent, RNA and ATAC are measured. So here we transfer borzoi to the RNA-seq data using LoRa plus the low-conv method. And we hold out ATAC-seq data for kind of validation purpose. This is the predicted versus observed log fold change between the senescent and wild-type condition, time point 7 and time point 1, for the holdout genes. Overall, there is a Pearson correlation about 0.5. And when we zoom in into changes in these 2 example genes, for example, we can see that ITGBL1, this one is induced in senescence, and we can see that the predicted expression across time points also follows this trajectory, and the same is true for the other gene, which is repressed during senescence. So next, we examine this specific gene, NUP188, and from the RNA-seq track, we can see that this gene is repressed in senescence, and borzoi prediction correctly predict the decline in the expression level in the senescent condition. So then we can compare the borzoi gradient for time point 7 and time point 1 for this particular gene, and identify sequence features that actually contribute to this expression change. We call this the normalized differential saliency score. Basically, this kind of is our method to identify salient features or doing an attribution method to identify sequences that are responsible for this change in expression. So by looking at the ATAC-seq data, which is held out, and we didn't transfer borzoi to this ATAC-seq data, we can see that most of the salient features here, they actually overlap with the differential accessible regions from ATAC, highlighted in red here, and the blue are ATAC peaks that are not differentially accessible. So these are, we don't really have a real peak to gene linkage in this case. We kind of just think they are probably regulating this particular gene because of proximity, and they are differentially accessible. Yeah, so this is kind of a rough way to really check whether we are learning something meaningful by looking at the gradient of our model. Finally, in this Hayflick data set, we asked whether we can use borzoi to predict the global TF activity change from RNA-seq data alone, looking at time point 7 and time point 1. So we are trying to identify TFs that are globally responsible for the differentially expressed genes. So we use Borzoi to score the motifs by looking at the gradient over each motif instance. And Borzoi motif scores correlated well with the ATAC-derived motif score. So the ATAC-derived motif score is basically a log-odds ratio of the motif enrichment in the differentially accessible region. And we can recapitulate some of the known TFs, such as TEAT and CBP that we reported in the original paper. In contrast, when we look at, without the help of sequence models, what we can do is basically only focusing on the TSS of these differentially expressed genes and do motif enrichment. So if we apply this method, then we can actually capture the transcription factors that are responsible for the downregulated genes, but we don't see anything for the upregulated genes, basically. Most likely because these genes are more driven by the enhancer regions, and that's not captured in this particular approach. But because Borzoi is seeing a much larger context, it captures these important TFs as well. Next, we also transfer the pre-trained Borzoi model to the TF knockdown K562 RNA-seq dataset. On the left, we see a correlation heat map of the knockdown effects of different TFs with the RNAi knockdown approach. On the right, we see the correlation heat map of the Borzoi predicted knockdown effects. Here, the knockdown effects is basically log fold change of the genes before and after the knockdown. We see a very similar overall pattern, and for example, ATF3 and CAT2B have very similar knockdown effects, and these CDCF, RAT21, similar knockdown effects. So these are all captured, and we capture basically the same structure as the ground truth. So then we ask for these TF knockdown experiments, if we use Borzoi to find the motifs responsible for the downregulated genes, can we prioritize the TFs that has been targeted in this experiment using a similar approach as I mentioned before, looking at the differential gradient? And the answer is yes. So here, for example, in 18% of the experiments, the targeted TFs is ranked among the top 20, here top K as top 20, by the Borzoi prediction. And so the orange, green, and red color bar are when we're aggregating the Borzoi motif score across just promoter, enhancer, or combined. As a baseline, we look at the simple TSS motif enrichment at the promoter region using SEA. So here, information is kind of limited to the TSS only, and thus we can recapitulate only less than 5% of the experiments where the targeted TF is among the top 20. We also vary the top K, 10, 50, and 100. The conclusion is pretty consistent. So this shows that we can actually use these sequence-based models as a way to help us better analyze the custom RNA-seq dataset. And by doing parameter efficient fine-tuning, we can do this in an efficient way. So in conclusion, we can accurately and efficiently transfer Borzoi model to custom RNA-seq dataset using this parameter efficient fine-tuning approach, especially when we take a combination of attention and count adapters. That gives us the best performance in terms of balance between efficiency and accuracy. We can transfer Borzoi model to allow us to study gene regulation in custom RNA-seq data, as I'm showing before. So this is mostly focusing, which this talk is mostly focusing on. We also did some experiments to show that we can also look at varying facts and predict them at least to the same level as a joint trained model, which we'll show later on in the manuscript that's to come later. So finally, I'd like to acknowledge all the members of the Kelly lab at Calico here, manager David Kelly, Ioannidis also contribute to this project, and all the other members of Kelly lab. And thank you, and I'm happy to take any questions.
Great talk. I was wondering if you fine-tune for something that you already trained on, let's say liver RNA-seq, do you get a better like, EQTL performance or worse?
Yeah, that's a good question. So I think for the GTAX, we had a model, Borzoi model, where it has the GTAX in the pre-training, and also I did this type of fine-tuning on basically keep training on the GTAX along. There is a subtle improvement in terms of the EQTL variant effect prediction. Yeah, that's kind of what we see.
No more questions? Okay, Maria.
Could you just comment on what happens to the bias term for the conv adapter?
So I didn't really adapt the bias term to the conv adapter. So I'm mostly basically just working with the weight matrix for the kernel. Yeah, that's something maybe we can think about. I haven't like kind of evaluate whether that would give me further, because just the bias term wouldn't give me too much additional parameters. So I can also just leave that trainable. That's definitely possible.
I understand that retraining from the whole joint data set is expensive, but I was a little surprised that full fine-tuning is doing better than these adaptive methods. How expensive is it to just train the full model just from the fine-tuning data set?
You mean just do a full fine-tuning?
Yeah.
Yeah, I think the most cost is on in terms of memory. It's about 80 gigabytes of memory for training a Borzoi model. In terms of the runtime, is probably double, so it's not that bad in terms of runtime. But in terms of memory, it's kind of more costly.
I see.
Yes.
Okay, let's thank the speaker.
Thank you.
So the next speaker is Shiron, from Gladstone Institute.
Okay, hi everyone. My name is Shiron Drusinsky. I'm a PhD candidate at UC San Francisco in the Gladstone Institutes and Katie Pollard's lab, and I'm excited to tell you about work from our recent preprint titled Deep Learning Prediction of Gene Expression from Personal Genomes. So genetic variants, especially genetic variants that are in non-coding parts of the genome, can contribute to changes in gene expression and therefore contribute to complex traits in human disease. So there is like a ton of active interest in research in identifying variants and figuring out which ones are functional and the mechanism by which they function, as we all know. One research direction that's been talked about a lot is to train models that take DNA sequences input and nothing else and try to predict gene expression. Now, since we know that there is a relationship between sequence and expression, the hope is that if the model is able to accurately learn that relationship, then we can interrogate the model, figure out what it has learned. For example, just two that are relevant to this talk, we could feed it someone's personal DNA sequences to predict their expression levels of specific genes, or we could predict the effects of specific variants and try to use the model to generate hypotheses about the mechanisms by which they function. As has been talked about a lot, there are existing models that have been trained to do this already. These are typically CNNs, including Informer, which has a convolutional transformer architecture and will be a focus of this talk. These models are, they have like a ton of really impressive properties, but one thing that they don't do very well is explain gene expression variability. What I mean by that, this might remind you of the keynote earlier today, is that in a pair of papers that were out last year in Nature Genetics, the authors used cohorts of people that have had their whole genome sequenced and gene expression values measured, and used them to test the ability of these models to predict gene expression given DNA sequences that contain variants seen in these people. That leads to something that looks like this, where each point here represents a different person. The x-axis is that person's observed expression, in this case, of the gene COL5A3. And the y-axis is their predicted expression given their DNA sequence, in this case from the model Informer. And here, the story is that the correlation here is low. And since Informer only takes DNA sequence as input, and the only difference between the DNA sequences that come from these different people are the variants that they contain, that suggests that the low correlation suggests that the model isn't able to pick up on those variants and figure out who should have relatively higher or lower expression for this gene. So for this gene, Informer doesn't do a very good job at explaining variability in gene expression across people. And that's not because there aren't genetic variants around this gene that are important for its expression. And here's an example to illustrate that point. I'm using, this is an elastic net, very similar to the PredictScan like linear model that Nilah was talking about in her keynote. It predicts gene expression using people's genotype. And it can find genetic variants and pretty accurately predict who should have relatively higher or lower expression for this gene using those genotypes, even while Informer cannot. So Informer is basically underperforming expectations here. And again, that suggests that for this gene, Informer isn't doing a good job at figuring out which variants seen in the DNA sequences of these people contribute to gene expression changes. This was first reported by Sase et al. And Huang et al. Last year, where they showed that other sequence-to-expression deep learning models, not just Informer, underperform these simple linear models on most genes. So this is an important challenge that we need to resolve, and it's not limited to just Informer. And I think given this result, it's interesting to consider how these models were trained in the first place, and that is by taking DNA sequences that come from the reference genome and optimizing on the results that come from various assays, including gene expression assays that were measured in cells and tissues that do not have the reference genome. And this has led to state-of-the-art models, this scheme, and these models have, again, really impressive properties, but I've also wondered whether this could be limiting model accuracy. And that's because, in a way, the model is kind of trying to learn a relationship that doesn't completely exist. These cells do not have the reference genome, yet it's trying to learn that relationship between the reference genome and gene expression. But maybe more importantly, it means that such a model will never have had any opportunity to see any genetic variant at any point during training at any locus. And that might partially explain why they underperform when they're asked to predict gene expression from sequences that contain genetic variants. And so given this, we wanted to evaluate whether fine-tuning with personal genomes will basically improve their ability to predict gene expression from people. And so to that end, we used data from the GTEx consortium where there's paired whole genome sequences and gene expression values that come from the same people. We focused on data that come from whole blood, for which we have 670 people that have with whole genomes sequenced and gene expression values measured. We used this data set to fine-tune Informer, starting with its pre-trained weights. And each batch consists of the DNA sequences that come from different people of length 49 kilobases. And the targets are scalar gene expression values that are matched to the same people. I also want to point out the linear models that I mentioned earlier, which outperform existing deep learning models, are fit in a manner that is very, very similar to this. The main difference is that instead of sequence, they're just fit on some kind of genotype matrix that contains the counts of different observed SNPs that are seen in those people. I also want to point out that when we do this, there are two different things that we can hold out, which is genes and people. So when we're training, we use people from the train set, genes from the train set. But when we evaluate on held out people only, and you might see the acronym HOP floating around in this talk, we are using unseen people from the test set, but the same genes from the train set. And so that means that when we're evaluating on held out people, we're evaluating in a part of the genome that the model has seen, either during pre-training or fine-tuning. So it may already have an idea of what regulatory elements are important for the expression of this gene. And it can also rely on variants that it saw during training, especially EQTLs that might also be seen in people from the test set. But I want to point out that this is exactly how linear models that currently outperform deep learning models are trained and evaluated. Holding out people and genes is much harder, and to do well on this task, the model needs to be able to accurately predict the effects of variants in parts of the genome that it hasn't seen. So in the next few slides, I'm going to focus on evaluations first in held out people, because like I said, this is the same scheme that is used to fit and evaluate linear models that currently beat existing sequence-to-expression deep learning models. And I explore this evaluation first to understand whether deep learning models are even capable of competing with linear models on this task. And then I'm going to return to the more ambitious question of held out people and genes at the end of the talk. And so here's an example of what things look like after fine-tuning on personal genomes. Each point here, again, is a different person. The x-axis represents their observed expression, in this case for the gene BTNL3, and the y-axis is their predicted expression. For this gene, with Informer before fine-tuning, in green here, Informer is not very good at predicting for this gene which variants should have relatively higher or lower expression. That's reflected by the low correlation. In ElasticNet, fit on the same gene does a lot better. And after fine-tuning Informer, and we call this new model that was fine-tuned on personal genomes, Performer, its accuracy becomes competitive with the linear models. And this trend sticks if we evaluate on other genes, again, only in held-out people. Now here, each row is a different gene, and the x-axis value is the correlation taken across people that I've shown on the previous slides. The story here is that Performer in blue and ElasticNet in red typically do similar for these genes, while Informer sometimes underperforms. So fine-tuning on Informer helps us become competitive with linear models, but one thing that I also want to point out is that we don't reliably do better than the linear models after fine-tuning on personal genomes either. They're roughly on par. And we've tried training and evaluating on up to 300 genes and find that this trend holds basically over those genes. So overall, fine-tuning on personal genomes seems to improve the ability of Informer to overcome some of those previously documented issues and better explain expression variability across people. So although Performer gets similar accuracy to ElasticNet models, one reason why we might still want to use a sequence to expression model like Performer anyways, is if it does a better job at finding functional variants. And so to look into that, we took variants that were predicted by each model to cause large changes to gene expression. For Informer and Performer, that just meant simple in silico mutagenesis experiments where you take two sequences that are otherwise identical but differ by the variant, form a prediction with each, and take the difference. That difference represents the predicted change due to the variant. And for ElasticNet models, we used the learned coefficients for each SNP. And moving forward, I'm just going to call these variant weights for simplicity. And here is one example. I'm showing a 1 kilobase region around the gene ZFP57. The position is along the x-axis. And I'm showing the variant weights along the y-axis. And I want to bring your attention to this variant that's inside of this dashed box here. This is a rare variant that wasn't seen by any of these models during training, yet Performer seems to upweight it. The reference allele for this SNP is a C, and the alternate allele is a G. And we use software called Motif Breaker to see whether this SNP is expected to change transcription factor binding. This is based on experimental PWM data, not like model gradients or something like that. And we found that the C to G change is associated or is expected to increase affinity for the binding of these two transcription factors to DNA. So this is just an example of how even though ElasticNets and Performer might get similar correlations across people, Performer might still be relying on variants that maybe have more evidence of functionality. Here's another example. I'm still looking at the same gene, ZFP57. It's just a different variant this time. It's a common one. The story here is similar, though, where the alternate allele is a C, and it's expected to increase affinity for the binding of certain transcription factors to DNA. But it's not included in the ElasticNet model. So over many genes and many variants, we see that variants that have large weights from Informer and Performer are expected to alter motifs at roughly the same rates, and that's what I'm showing on the x-axis. This maybe suggests that Performer isn't just relying on sort of arbitrary EQTLs in the training data, but it's still paying more attention to EQTLs and other variants that alter expression by disrupting important motifs even after fine-tuning. And second, variants with high weights that come from Performer alter motifs at a greater rate than those that come from ElasticNet, the linear models, even though the correlations that they both get across people are very similar. One way that this could be possible is if the variants that are upweighted by ElasticNet models and Performer models are in LD with each other, but the Performer variants maybe are still more functional more often. So this, I guess, might be a reason why we might want to use a model like Performer over linear models, even though the accuracy that they see across people is pretty comparable. Now, I want to close on the much harder task of evaluating these models on held-out people and held-out genes. I think strong performance on this task maybe obviously is more difficult because the model can only really do well here by using information that it's learned from other parts of the genome. And it doesn't already have an idea of how different motifs and variants that are seen around this gene contribute to gene expression changes. Yet we expect a model that has comprehensively learned causal gene regulatory patterns to be able to do well on this anyways. So if I fine-tune Informer with genes in Informer's original train set, but I hold out 100 genes, this is the distribution of those correlation scores that I see for those genes. Each one of these dots is a different unseen gene from the test set. The y-axis is the correlation across people we find for each of those unseen genes. If instead of holding out those genes, I trained on them directly, this gives us a baseline for the accuracy that we should expect to see for those genes. And we see that relative to when we hold the genes out, or I should say, relative to when we train on them directly, when we do hold them out, we underperform relative to this baseline. So that suggests that when we hold genes out, basically, the model doesn't do as good of a job at identifying important variants in those DNA sequences that contribute to gene expression changes. It's also, by the way, not any better than Informer's accuracy on these genes originally. So fine-tuning on personal genomes doesn't lead to better performance on held-out genes relative to the original pre-trained model. In my view, what I'm sort of showing here, this story, is an important gap because we expect models that have learned causal gene regulatory patterns, causal relationships between sequence and expression to do well here, but they don't yet. So there are still important remaining challenges, even after fine-tuning existing models with additional personal genome data. So I want to point out that we've been in communication with folks from the Ioannidis and Mostafavi labs and have found that our results are mostly consistent with what they have found when fine-tuning models or training models with personal genome data or genetic variation data. And so in this talk, I focused a little bit less on the model choices and architectural choices that we made because all of our groups have made different choices and our results seem to be pretty robust against those choices. I've shown that we can overcome some previously documented issues of explaining expression variability if we fine-tune on personal genomes. We call the resulting model Performer, and it does similarly well, but not reliably better than linear models. But maybe the variants that it selects have more evidence of functionality. And that maybe represents the promise of using a sequence to expression model as opposed to a linear model for this kind of stuff. And while we overcome that previous gap, we identify a new gap, which is performance on held-out people and genes, and more work is needed there. So altogether, we think fine-tuning on personal genomes is a step in the right direction. It corrects some previously documented issues, but there are other issues that remain. So yeah, thank you. I would like to first actually acknowledge folks from my lab, especially my advisor, Katie, and the co-first author on my paper, Shen, as well as folks from the Ioannidis, Mostafavi, Troinskaya, and Capra labs for helpful discussions, and my funding sources. And I'm happy to take any questions at this time.
Beautiful talk. The publicly available data sets for which we don't have any genotypes, and the data sets where we have matched genotypes and some RNA-seq, for example, there will always be this kind of gap. So the ideal model should, I think, take the advantage of both of these kind of data sources, right? Because the biology coverage of the data sets without genotypes will always have kind of larger coverage than this tiny subset of it where we have access to genotypes too. So what do you think about reconciling these two and maybe using genotypes whenever available, but defaulting back to reference genome when it's not?
I think using the genotypes is helpful, but clearly it doesn't get us to exactly what we want. And I think using the data sets that don't have genotypes is still helpful, especially for pre-training. I don't think that if we didn't start with a pre-trained model that came from those types of data sets that we would be able to identify variants with more evidence of functionality. Because then what we're left with is a model that more or less is relying on learning the same kind of associations that a linear model learns, right? And I guess it's unclear if we would have enough data to learn anything more than that, even if we have a more complex model, if that makes sense.
Another quick question. How much do you think Informer forgets about the regulatory code that it learns in the pre-training when you fine-tune it on individual genomes?
Yeah, that's something that we're still looking into. It's unclear. There are some things that it doesn't do as well on, it doesn't predict the average expression of unseen genes as well as before, but part of that is because of the way that we're sort of normalizing the data to be distributed kind of around 0 and appear normal. We've also seen examples where the motifs that it picks up around the genes that it's seen and around genes that it hasn't seen are different from the ones that Informer picked up on, but they still look like real motifs that align to PWMs that we see in like Hokomoka databases, for example. So it's unclear if it's really like forgetting something, but it seems to be learning something a little bit different as well, but we need to explore that more.
Thanks for a really nice talk. I thought the result on like variant weights, I think that's what you call them, comparison with PredictScan, that was kind of promising, but I wonder, so for defining the variant weights for a PredictScan, I guess you just run elastic net on the same set of training individuals and take the weight from that. So it could be a little bit unstable, and I wonder if you've tried like ensembling elastic net model and averaging the weights, and like, is that still worse than your model's weights in some way? Like, I just want to know how big the gap is to understand how much mechanism that your model did really learn.
We haven't tried that. All the results that I've shown do come from different replicates of both the performer models and the elastic net models, but when it comes to the results that come from the variant weights, we didn't try averaging the weights that come from different model replicates and comparing averages versus averages. We just took the first fold and compared them to each other, but that's an interesting thing to try. But the correlations across people are all averages across different model replicates.
Okay, no more questions? Oh.
Hey, thanks for another great talk. The other thing I was struck by is that you have the same results as, for instance, Nilah presented in whole blood that she had in brain, right? Does that suggest something interesting about whether these models of cell type specific expression, like we saw in the previous talk, would be helpful?
I think if I understand your question, if we had like we don't have data where there's matched whole genome sequences and gene expression values where the gene expression are like single cell resolution, at least with many people. If we had that type of data, I think it would be better because maybe we would be able to identify genetic variants that cause changes to gene expression in rare cell types that you wouldn't be able to resolve in the bulk level data. But that might be wishful thinking. So
I mean, for common variants, you may be able to find that in cancer databases.
Oh, yeah, that's an interesting place to look. Thank you.
Okay. Great, thank you for the nice talk. Thanks. So we'll have a coffee break for 25 minutes, 30 minutes, or 25 minutes until the next keynote speech at 10:50. Okay, everyone? It's 10:55. Our keynote speaker is ready. I know you wanna chat more, but we need to really start. Oh my God, this very busy. Okay, everyone. Okay, I think we should just start. People will just come in. Okay, so next speaker is the keynote speaker, Maria Chikina from University of Pittsburgh. She's going to talk about biophysical sequence model, sequence to function models.
Yeah, you can, okay, the mic's on. All right, well, thank you so much for inviting me. It's really a pleasure to be here. I've really enjoyed the conference so far. And okay, I'm gonna start with like a little interactive thing here. So we're on the West Coast, and but last year on the East Coast, we had a solar eclipse. Who went to see it? Wow, lots of people, right? It was awesome. And we were able to do this because we got this really nice map that tells us like exactly where you have to be in the zone of totality. I had to drive quite a bit to get to it, but it was totally worth it. And really, like, we didn't think too much about how that happened, right? So what is the first time somebody in the human history was able to correctly predict an eclipse? Who thinks it's like before Common Era? Okay, who thinks it's like modern times, last 500 years? Okay, so actually you're both right. It just depends. I didn't phrase the question correctly. Like it was not a well phrased question. So it turns out like a lunar eclipse people actually were able to predict like before year 1. And the solar eclipse really was modern times. And so the thing is, there is this 18-year cycle that the 3 celestial bodies, like the Earth and the Moon and the Sun, have periodicity in, and this was discovered a long, long time ago. And both like the solar and the lunar eclipse obey this cycle, but for whatever reason, one of them we were able to predict, and the other one not. And so what is the reason? Well, this is the reason. It's just when the lunar eclipse happens, everyone can see it. Everyone for whom it is night. Right? This is where you can see lunar eclipse. This is the path of the solar eclipse. And so just the precision required to predict this is much, much higher, even though they kind of obey the same cyclical behavior. And so perspective, like of an ancient observer that doesn't go very far from where they were born. The lunar eclipse happens roughly every 18 years, so you would actually observe it multiple times in your lifespan, and like you could develop some kind of pattern matching in your head about when the next one is going to happen. From the perspective of the solar eclipse, it happens roughly every 400 years. So by that time, like your culture has turned over and you've lost all your records, right? So, and that's kind of where the problem, the prediction problem gets really hard. So this is actually the first instance of somebody correctly predicting the solar eclipse. And it was Halley of Halley's Comet, which he's somehow more famous for. I feel like this is actually really, really impressive because this was he published this map of the zone of totality before it happened. Then there was another map that was published after it happened, and I didn't put it up here because they're actually like, they look the same. It's absolutely incredible. But he had to use like calculus and the laws of gravitational motion that were pretty recent discovery. And so to kind of like put all of this in perspective, right, we have our ancient astronomers. They're doing pattern matching, and they're getting some kind of correct predictions. But really, it's pattern matching. They have no idea about physics and even like basic geometry. And then we can have kind of these what I call laws for special cases. So like Kepler laws for predicting planetary motions, assuming everything is ellipse, and these work really well. But the orbit of the moon is actually not an ellipse perfectly, and that's enough to throw the whole thing off. And in order to make these kind of predictions, we really needed we needed universal laws and this concept that you have to like integrate them over time to make predictions into the future. So why is this actually relevant? So I mean, we're also in the business of making predictions, right? And we have lots and lots of prediction problems in computational biology. The one that we heard a lot about today is predicting things from sequence. And really kind of what we want to predict is kind of how each cell interprets its DNA. So there's some kind of common germ in DNA. Each cell sees something very different in terms of its chromatin, and this dictates all of the functional output and ultimately the phenotype of the cell. So the prediction problem we're trying to solve is like predict the output of the genome conditioned on the cellular context. So we have this like specific function f like for this cell and a different function d for this cell. I just want to linger here for a little bit because I think this captures what we're doing right now, even though in theory there's actually some kind of universal function. And I think people are moving in that direction, and I think it's going to be really interesting to see what people do there. But for now, a lot of this is kind of like context conditioned. And so, so we're interested in these sequence to function models, right, where we take sequence and we predict it using usually some kind of neural network. And so this is informer predictions, right? So we have this is the measurement, this is informer predictions. These look really amazingly the same. So we're like, wow, this model has learned biology, right? And I guess to a certain extent it has, but then not to belabor this point again, but in a different perspective, it actually didn't. And in some sense, this is actually we're just not on the precision scale, we're just not where we want to be on the precision scale. We have some amount of precision, we just don't have all of the precision. It's just these personal genome effects are tiny relative to the other effects that you saw in the other plot. So you can just think about it like, well, we have kind of like a Kepler model, not a Newton model, right? I guess kind of why is gene expression really, really hard? So maybe lots of answers to this question. This is kind of the one that I've settled on. Is in some sense we actually have like a fundamental data problem. There's only so many genes in the genome, and the input for the gene expression of a gene is actually really long, right? So you have these regulatory elements and maybe very, very far away. So you need to consume like all of these different, so much sequence, most of it is irrelevant, find all the relevant parts and synthesize it into the output. And maybe it's possible that actually every gene locus is unique. And like, okay, what do I mean by that? That's kind of a strange thing to say. But maybe there's millions of biomolecular interactions possible, right? And maybe there's, for like a particular gene locus, there's one that really matters in terms of precision that is completely ignorable for every other locus, right? This is like consistent with what we know about biology, and there are like some very special loci that people list the data in detail, and it's like, I don't know if that happens anywhere else, actually. And so this is just to argue that like maybe it makes sense. Like this is the like once in 400-year solar eclipse. Like maybe like the simplest thing to do is actually develop some general, like not wait for more data or like more compute, but actually develop some kind of like generally applicable methodology that's going to improve precision. So I'm going to tell you about like really tiny baby steps that we're trying to make in this direction in my group. This is kind of generally in the sequence to function modeling space, and we've heard really amazing talks today about like just completely incredible like engineering and data efforts and people like so many GPU hours, right? Like so much work and really amazing results, like incredibly promising stuff. And so I'm going to tell you about basically none of that. We're doing like really simple things that like run on my desktop and are very easy to compute. So it's a little bit just the opposite. So I'll tell you about this work that we call the total interval sequence to function model, which was in ISMB proceedings from last year. And I'll also give you an update on a model we presented. I had a postdoc presented last year as a short talk, the differential loop extrusion model. Don't quite have a preprint, but it's coming. It is, you know, like maybe like a month. OK. All right. So for the totally turbo sequence function model, we're very much inspired by this work from Sarah's group. So often we care a lot about like the predictions that our model is giving and how accurate we are. Is like your architecture better than my architecture? Oh, I got like an extra like 0.01 whatever like performance. But we also actually a lot of times we use these models for just biological insight, right? And I think this is a really nice paper that really highlights the capacity of these models to deliver that. And so in this paper, they were looking at predicting chromatin accessibility across cell types and really focusing on the contrastive aspect, like really looking at like what makes the cell types distinct. And they had kind of a scientific objective, not like a building most predictive model, but the scientific objective was like, let's try to understand what makes these cells distinct. And the architecture is not the most complex architecture, but it was one that was optimized for the scientific objective that they were interested in. And so, so this is AITAC is actually it's a model and a kind of interpretation framework. And so, what it does is it takes convolutional kernels and it maps them to motifs. So these you can take these interpreted motifs. Everybody has done that. That's no surprise. And you can track these motifs to the final layer activations through filter ablation. And after you do this, is this hello? It's OK? Yeah. OK. So after you do this, you get this kind of it just gets basically a transcription factor to cell type map, and this map contains a ton of biological information, and we can kind of spot check it for correctness. So for example, we have like PAX5, EBF1 for B cells. this is immune cells. We know a lot about how they differentiate. So you can do a lot of spot checking. So we have like ROR-C, ROR-gamma for LC. So all of this makes sense. And from this, we can maybe trust that some of the other predictions they're making are also actually correct, which turned out to be the case. OK, so this kind of gives us like a roadmap for how to interpret these models. And so what exactly we do, right? So we have the first layer convolutions are interpretable as motifs. That's pretty straightforward. But it's not a single layer network. We have these other layers. What do? We know we need them for prediction. It makes a big difference when you put them in there. What do? So well, you can kind of hypothesize all the different things that they could potentially be doing. They probably do all of these things. And so the really kind of fun and like answer from the perspective of biology that I think we all get excited is that they do some kind of like combinatorial modeling, right, where transcription factors have to work synergistically to regulate chromatin structure. But I guess there are also what I would call like boring answers potentially. Right, so you need extra layers to do some extra motif calibration. So these kind of like right, so we have like these kind of PWM motifs. We know they're actually not accurate. The independence assumptions, like people have demonstrated, those are not actually 100% accurate. So you could do some more calibration on that. And you can also do some calibration for just like binding probability, because the motif just gives you a score. It doesn't give me binding probability, which is actually the unit that I would want. And also, OK, let's say they do actually convey some kind of TF-TF interactions, which I think definitely does happen. It's really hard to kind of tell from the network exactly what kind of interaction is it computing, right? We can think about kind of a perfectly linear interaction where the combination, like we just like linearly add the motif activations. And in fact, we saw with Puffin, like that's actually like very effective for some tasks. Like linear seems to be good. And then we can also have these nonlinear kind of functions, but we can have both like synergy and saturation. I think we get like very excited about synergy. Saturation seems really boring. I think maybe we would like to know which one of those it is. So I guess the question that we pose is like, instead of trying to figure out how to like implement a really sophisticated post hoc analysis that's going to answer all of these questions, why don't we just put a model already in this form, where it was just going to like intrinsically answer all of these questions with its parameters? So this is the model. We call it totally interpretable sequence to function model. And basically, it's still kind of like a neural network architecture in a way, but it doesn't have any uninterpretable hidden layers. So every layer is either DNA or it's transcription factor activation. And the way we went about constructing the layers is, it's inspired by what we know about biology. So I'll just quickly take you through how we did that. All right, so we have this top layer, which is just a kind of like a very basic convolution with some kernels, and we actually initialize them to known motifs. We can fine-tune it later. And so we just perform a convolution. We get this like very basic motif activation score at every position. There's a kind of a nonlinear calibration for each activation. It's trying to map it onto this probability space, which is where we want to be. And then we actually pool everything across all positions with trainable pooling. And with the trainable pooling, basically what it does is it can model both like an average and a max. And we had actually pretty specific hypotheses for what we thought the model would do. So transcription factors vary like really widely in terms of their specificity. Like you can just look at the PWM, like the information content of that PWM, like how many bases is it actually making contact with. And so like a low specificity transcription factor kind of like, it doesn't, there's not like a particular site where it's present with like probability 1, right? It has kind of like these on-off dynamics, and it can bind lots of different places. And you certainly don't want to do a max in this kind of situation, because it would just lead to a ton of false positives. Like your binding site could be so low specificity that literally like any sequence has a match, right? So a max really doesn't make sense. And then we have transcription factors that actually have like extremely specific sites. So like for CTCF, you see a CTCF site, it's like very likely there's a CTCF there. And for those, maybe actually max is sufficient, but we don't want to make these decisions like arbitrarily. We're going to let the model decide, and we can check that later. And finally, we have an interaction layer. And basically, this interaction layer is just every transcription factor gets like kind of a tuning parameter. It's like between 0 and 1 that where the tuning parameter is a function of the presence of the other transcription factors. And that actually explicitly parameterizes this like and or additive contribution, where like 0.5 is actually perfectly neutral. If like all of the interaction coefficient is 0, you get 0.5. And so that's the model. And then through all of these steps, you actually maintain TF identities. So every layer in this network, all the rows they have names, right? They're just whatever transcription factors we used to start with. And so the final output is actually just a linear prediction for open chromatin activity. And it actually is immediately interpretable as a TF to cell type map. And just before I show you the results, I'd just like to point out a few things about the data set that was used. So we're actually using exactly the same data set as the AI attack. And it's a really amazing data set where we have data across an entire developmental tree of immune cell types. And, but it is classified in 7 main cell type lineages. And generally, as like we see and was reported in the original paper, the lineage prediction is much, much easier than the within lineage. Like you can just kind of visually assess like a little, I zoomed in on a little heat map from the original paper. Like if you just focus on a particular lineage, there's all these places where it seems kind of the predictions I'm making are constant, even though the data has variability. So, so for most of the talk, I'll just focus on the lineage prediction tasks. And so the way we kind of, we actually assess these results is we assess them as improvement. So we compare like a convolutional tower, exactly the same one that was used in the paper, though we retrain it with our folds, like so all of the training is exactly the same. And the way I report the results is just improvement over convolutional tower. And so we can see that, in fact, the full model does improve over the convolutional tower. And we have kind of this ablation study where we take out different kinds of different layers from the model. And it seems that the interaction is actually making, our interaction layer is making the most difference. And we can now, the final layer coefficients is just like, that's it. That's what they are. I can just immediately plot them. Nothing else has to be done. They just already look like this. And the results are extremely similar to what you get with AI tag post hoc analysis. For example, B cell, we got literally the exact same result. And similarly with Broer gamma. OK. And all right, so let's look at some of the other parameters, right? This is the final layer. What about some of those internal layers? So for the trainable pooling, we had this extremely specific hypothesis that it should kind of track with motif specificity. And in fact, it does. So this is the information content, and this is the pooling parameter. It's like learned from scratch, and goes between average and max. And they're very well correlated, though not perfectly so. And we can also look at the interaction layer. So this is just kind of a global view of the interaction. So we can look at kind of the final layer coefficient. So this is what gives you all the cell type specificity, just aggregated over cell types. And we can look at the total contribution to the interaction layer. And so one thing to notice is that these are actually quite different. So we have like different TFs that are high for interactions and then don't contribute to TF specificity. And so a really key one here is CTCF. That makes sense because it's like a broadly expressed factor. It doesn't, it doesn't determine cell identity. All cells have it. So and this is a kind of a fairly general trend where a lot of these factors were not necessarily expressed in every cell type, but just generally broadly expressed. These were the most interacting ones. And one thing I said is the way we parameterize the interaction layers, we try to kind of explicitly capture, like, do we have like and or linear? And so just kind of like it's kind of color matched. We have blue would be saturation. There's a negative coefficient. And this was maybe at first a little bit surprising. It's mostly blue. Right? Especially like something like CTCF has like a ton of interactions, and they're just all blue. So basically, it's capturing saturation. Maybe a little disappointing, but actually, in retrospect, it makes a lot of sense. And it has a very simple kind of biological interpretation. So if I'm trying to predict open chromatin, I see like a strong CTCF site. I don't care what else is there. Like that is already highly informative. Right. And I just want to kind of go back to the full prediction problem with the full tree. So I'm not going to talk too much about that. There's more results in the paper. We did actually, at the end, we tried to use all the data. And I think we did do kind of like an interesting thing here that in general is just, I think like a good technique for trying to deal with these multitask problems where the amount of there's certain contrasts in the multitask problem, they're like way easier than others, right? And how do you make sure that you're actually finding like the subtle interesting things and not just like getting better and better doing just the lineage prediction? And so in this data set, there was actually like a pretty natural way to transform the problem that made it more interesting. And so we made a new prediction task, which was child versus parents, right? Like you're trying to predict the transition across the lineage tree. And then you're still using all the data, right? So you don't have to like somehow selectively throw out the data or make kind of like a new data set, but it's just been transformed this way. And I just like to point out, like even though we have these kind of parent-child relationships here, this is always possible because you can always like make up a parent by just taking an average. And this way you can form a bunch of kind of interesting contrastive tasks. And so it turns out, yeah, OK, this is really hard. So basically, everything that's colored is a place where we had a good prediction, and that is just very few edges on this tree. So really hard to make these kind of parent-child predictions all for a few places. And as you might expect, they correspond to like major lineage transitions. And interestingly, doing it this way does all of your B-cell predictions, turns out they are all concentrated in a very specific developmental stage. There was one exception here where we actually were seeming to get quite good predictions for this transition. So double positives to CD4 and CD8. And I don't know, if you've ever looked at like data from naive CD4 and CD8 cells, they're incredibly similar. I mean, very, very difficult to distinguish with single cell ATAC-seq or RNA-seq assays. Not so much once they're activated, but in naive state, they differ very little, mostly by the expression of the CD4 and the CD8 genes. This is really subtle. But it turns out not only were we able to predict this well, we actually found the correct biology. Won't go into too much detail here, but for this transition, which has actually been kind of studied in depth. So I want to kind of summarize this with all the things that we tried, we didn't work, because the model is really simple, and I know all of you are why didn't you do this? Why didn't you do that? And the answer is probably we did do that, and it just didn't work. And so one particular kind of really major criticism, which still bothers me, and this is something we're actively working on, is this is not a grammar model. This is the billboard model, right? So we at some point, at a very early point, lose all positional information about where the motifs are, and that is not because we didn't try to make it a grammar model. So what I mean is it doesn't work, because certainly we have these convolutional towers, and those are grammar models by their construction, and they do work. So what you're saying, it didn't work. So we kind of had some different metrics for what we consider working. So obviously performance, right? That not too difficult to make a grammar model work just in terms of performance. We had another metric, which is parameter stability across folds. Now, that is more difficult once you start making the model complex. And finally, we had our most important task, which is how well did we recover, just without any post hoc analysis, just in the final layer from that final heat map, how well do we recover in the known cell type specific transcription factors? We actually had quite an extensive list because the system is so well studied. And so when you make the models more complex, these just go away. Your performance will increase. But the interpretability and the stability, which actually go hand in hand, because an interpretable model should ideally return very similar results across training folds, right? If it's finding out something real. So those tend to go away pretty rapidly. So still working on kind of trying to resolve that and make more complex interpretable models. I think this is actually totally possible, and we're making progress towards that. So, right. So this is that was a totally local model that just looks at a very small window and tries to predict open chromatin. And of course, it's just a small piece of the regular code. And ultimately, what we're interested in is gene regulation, because that is what is going to give us phenotypes and diseases, right? And gene expression is really complex. It's much harder to predict. We've heard many talks about that today. And it's so complex because just so many things have to come together. And you have all these distal elements that come in contact with the promoter. And this is like a very complex process that's orchestrated over like mega-base scale. So we have some data that kind of tells us a little bit about what's going on at the correct scale. And so we have these kind of high C, micro C data sets that capture some of this 3D folding that we think is important for orchestrating gene expression. And so this is an example of, hopefully this is familiar to everyone, this is an example of a high C matrix. So where does the high C data come from? So I think when people first looked at this, there were maybe quite a number of theories about what is actually going on at the molecular level that is giving rise to these patterns. And so when you, obviously, like we observe these kind of diagonal square structures, which are now are known as topologically associated domains, or TADs. And one thing you can imagine is like a TAD is like just kind of like a bundle of DNA that is like all in close proximity in the cell, but that is like a model that mostly we've moved away from. And so now what we understand is happening is that this pattern is orchestrated with what we call a loop extrusion process. So instead of this being the picture in one cell, in a single cell, actually what we have is a bunch of different states that just in aggregate produce this. So the loop extrusion hypothesis hypothesizes that, I was going to say it's a hypothesis because actually not everyone believes it. But I mean, so the idea is that you have the cohesin complex, which is going to bind at some particular point, and it's going to start like here. And it's going to start extruding a loop, and the loop just progressively gets longer and longer. And the cohesin has two legs, like a left and a right, and then they can both move. And the rates at which they move is variable across the genome. So when you see like a boundary like this, that is kind of a control point where the cohesin leg seems to get stuck and not move across that position as efficiently. So the important thing is to realize that what we now understand is the contact map is like, yes, it's an average across cells, but a really important aspect is actually an average across time. That even if you had like a super homogeneous cell population, you're still somehow not seeing the full picture. So we've been working with kind of this idea. So our model is actually very heavily inspired by this kind of like 1D loop extrusion simulator by Jeff here. And so this is a really simple model that models this process, but it never instantiates like the 3D chromatin structure. It actually just adds context to the contact map. So the way we can look at this model, so we have like a cohesin, and at first it binds, and it binds right like at the same location. So it binds somewhere on the diagonal. So it's making a contact very close to the diagonal. So just maybe like a kilobase away, and at any point it can just maybe do nothing. One of its legs can move forward, or it can completely detach. So now, if you simulate this process, and for all of these like time steps, you just add a add a count to the contact map, you get something like this, right? So the cohesin moves along. Sometimes it just stays in a particular position a little bit longer, so you get more counts, and it like kind of randomly moves its right and left legs. Sometimes it gets stuck because these are actually convergence CDCF sites, so it's really stuck on one side here for a lot. It only moves in one direction, and eventually it falls off, right? So this is one cohesin complex. That's its lifespan on this piece of DNA. So now you can add another one. So now there's two, and you see like they actually get stuck at two different positions. And you can do this a lot, and then like really a lot, and it starts to look like a high C map. Okay. So what we decided to do is actually just consider just the steady state, right? So this is, like factoring out all these stochastic processes which are known what's happening. Like, what is it going to look like when you just take the average over all time and all of the cellular states? And this is what we call the differential loop extrusion model. And basically, we formulated as as the concentration, we consider the concentration in a particular cell, which corresponds to a particular loop. It's just related to how much things are flowing into the cell, which is like how much of the loop is being extruded, and how much things are flowing out of the cell, plus the term for the cohesion coming off entirely. And so so this is now just like we can actually get like this completely closed form expression in terms of these quantities, and this now is not a stochastic thing. It's just a closed form expression for the concentration of cohesins in the cell. So that's why it's differentiable. You can fit it, and you can fit it to data. And so before I show you how we fit it to data, there's kind of a subtle, like seemingly completely kind of pointless discussion, but we actually had very long and lengthy discussions about this aspect. So probably you're familiar kind of with looking with high C on high C in two different ways. So this is like the raw data. It's actually log transformed, so you can visualize it a little better, some of these like interesting looping features. And this is the diagonal normalized view. So so we just subtract the overall diagonal value, so it's 0. And this is why you actually see blue is negative, right? You're just like, what does it mean to have negative contacts? Well, it's kind of, it's relative, like it's a log fold change. And so, yeah, right, which of, like, how do we fit the data? What is more interesting? So in some sense, the raw data is clearly, like, we're trying to fit a biophysical model. Raw data is the physical model. Contacts cannot be negative. Right, but on the other hand, most of the variance here comes from the diagonal decay. And so your model is going to work really hard to fit the diagonal decay before it starts doing anything else. Like it's not going to do any, like many iterations, it just won't do anything interesting. On the other hand, in this model, we see like a lot of really cool structure and like promoter-enhancer interactions. We see them right away. We do actually work in this space. And the way that we do this, so our model is a biophysical model. It outputs like contacts, it outputs concentrations. So it kind of runs in real units. And just before we compute the loss, it self-normalizes to this view. And I'm not going to go into a ton of details for why this is true, but it turns out that this has like a really nice side effect where we don't have to worry about some of these like terms that come from polymer dynamics, because like we actually have no polymer dynamics in our model. We just have the loop extrusion, and modeling those is actually quite hard. Okay, so this is the DLM model, and so for we have some kind of initial concentrations in the diagonal, and actually like in reality these are all constant, but you can think about them like potentially variable. And starting with these initial diagonals, we have some parameters that describe how likely you are to move left or right at a particular location, which is these p's, and we have like a off rate. And so we can just go diagonal by diagonal and calculate all of the other values. This is a completely closed form. You can do it for the entire, like if I gave you these parameters, you can just fill out the entire contact map. So one thing you notice is this gets like really ugly really quickly because it's actually fundamentally recursive, so you get these very ugly equations. Another thing I wanted to point out, just like when people are thinking about architectures or what architectures like are biologically meaningful, which is a totally separate question, like what architectures work well and are trainable, right? But if you think about what architectures are biologically meaningful, this is not a transform, this is a scan, right? This is like every computation is actually totally local computation. So in fact, right, there's a closed form for the entire thing. You can like fit it directly to the data. It will work, but it is slow, it is not super stable, so there's actually a better way to fit this. And so this is, I'll just tell you a little bit about how we actually fit this. So we think this is actually now the data, and so we're gonna, the decay parameter, we don't worry about it because everything's diagonal normalized anyway, so we're just gonna fit the left and right slowdowns, which tell you how likely it is to go left and right. And so we can just look at the perspective from the perspective of like a single square. So we're going to make a prediction for this particular square. And this is going to be made in terms of the inflow to the square, which depends on these kind of slowdown parameters for moving left and right, times kind of the concentration of cohesins at the previous looping step, at like the slightly shorter loop. So that's the numerator. And the denominator is just the outflow, is just like how likely you are to outflow from the cell by moving either left or right. So we can think about, right, like we want to fit these parameters for like a left and right flow. We have this kind of functional form which predicts the concentration at the next diagonal in terms of concentration in previous diagonal plus these parameters. And so the way we do this is exactly like that. So we have a model that takes the previous diagonal and tries to predict the next diagonal in terms of these parameters. And compare it in terms of just MSE loss with the actual data. Right. So, okay. So it goes kind of diagonal by diagonal, and if you look at like the individual fits, like they're amazing. Like they look exactly like the data, but it's kind of a stupid thing because, well, you use the previous diagonal to predict the next diagonal. Maybe you like didn't even have to do very much. Maybe you can just like average the adjacent cells or something, and you will do a good job. And, right. So, so what does it mean to actually make a prediction in this case? So the way we make a prediction is we actually start with the first diagonal is constant, right? So now we don't use the only thing we can use is these like left and right parameters that would fit from the data. And so the model kind of unrolls in a recursive fashion from a constant concentration in the beginning. And I feel like on this patch, I think this is pretty amazing. I think you're going to have to look hard to find where it's actually different. It's different. Right, so this is like this nice example patch. And so the way to think about it is like we have this data. And what we're actually just performing is some kind of very like compression operation. So I'm taking this data, I'm saying like, no, I'm going to represent your data just in terms of this. This is much now very fewer parameters. This is like, this was 2D, this is now 1D. So this is your representation, and I just give you this decoder. And this gives you your data back approximately. So I'll show a lot of plots that look like this. The top's the data, the bottom's the prediction. So in this particular case, they look very, very similar. It's hard to tell which one is which. On like a more real case, you're not going to have so much trouble telling which one is which, because one thing, the data is like very noisy, and this ends up looking quite smooth. But we make a lot of plots that look like this. And so this is the data. This is the prediction starting from a constant and just using these left and right parameters, which are plotted alongside here as the representation for the data. So you can actually get pretty good fits from this model. Okay, but actually this model is like not really, I mean, of course it's not close to being complete. There's lots of things we're not modeling. But actually there's a very simple thing that we know is present in the stochastic model, but we're completely missing in this model. And the thing that's present in the stochastic model is when you run the stochastic model, you actually you have more than one cohesion on the DNA, right? And which is realistic, right? People know like what the concentration of cohesions are, and you really should have more than one. And so they have an opportunity to interact. And okay, what happens when they interact? And this again, we take inspiration from Jeff's model, which dictates that when cohesions bump into each other, like they're trying to move past each other, they can't. They become more likely to detach completely, right? So. So we made this what we call like the cohesion stalling model, where actually it has exactly all, it doesn't have any more parameters. It just has a different functional form. So it has this kind of implicit stalling term on the bottom. And it turns out the stalling model actually fits the data much better. So and there's an interesting phenomenon where, so a bin is 1K actually, so this is 20K bins. It's actually exactly the same patch, just like at different resolutions. It's a biophysical model, so it should kind of scale nicely to different resolutions and behave exactly the same. But we found that the basic model really actually kind of breaks down at the finer resolution scale. But the stalling model actually can still recapitulate a lot of the data. And in particular, what the stalling model is really, really good at is creating these, like, what I would call like these passive TADs at all the different resolutions. And this is just kind of like an overall plot where we look at like like the very fine resolution and the more coarse resolution and across different cell lines. And overall, this blue stalling model is actually definitely fits the data better. Has no more parameters, just different function, right? So I think this lets us learn something about what the realistic biological model formulation is. And OK, and so finally, I guess these kind of models over here. So we call them like direct fits in the sense where all we're really doing is data compression. It's like not a predictive model at all. Like I've taken this patch and I've like generated this very small representation for this patch from which I can get it back. But I haven't learned about anything else that's happening anywhere else in the genome. Like I can make no prediction. It's just nothing. Right? So this is the direct fit model. And ideally, we would like to use it in a predictive context. And so the way we use it in predictive context is now this is kind of a sequence to function model. So we have a reference genome which has sequence features. So the CTCF positions left, right. And we want to do this in like a cell type specific way. So we have some like a context vector, which is the open chromatin for that cell. And we take these and have some kind of fairly small neural network that just outputs our left-right parameters. And from the left-right parameters, we generate the data so we can compute kind of the error on our data, and this whole model can be fit end-to-end. And now this is actually a fully predictive model. Here's a bunch of patches that this model has never seen before. And OK, so it is not Orca quality or Akita quality, right? Like, no, it's not. If you're like squinting at it and like, is this actually good? It's not that good. But we only used 536 parameters. That's it. And we also didn't, like, we used very little of the sequence, in fact. So like, we had a lot of discussions about data leakage. Like, oh, is this a homologous region that you have seen before? Like, what happens if you left that out? We actually really don't have to worry about this because we literally just use the CTCF sites, and that is it. There was no actual sequence went into this model. It was already pre-featurized in this way. So yeah, this is, I think, like it's pretty promising. We're working on getting like a full sequence model, and of course, like one advantage of this model is this is a thing that can just train in a few minutes on your computer. You can really play around. Like you can play around with the parameters, the model formulation. It just lets you do so much. As opposed to like the full sequence model actually takes some time. So yes, it's more accurate, but you can explore less, and there's definitely a trade-off. And so I'd just like to finish with thanking the people who worked on this project. So the total interval sequence to function model was led by a student in my group who actually just graduated. You want a postdoc? This is Churl. He's amazing, and he's done so many diverse things and really excelled at all of them. And the DLM model is really a collaboration between Tina, who actually presented the idea for this model at MLCB last year, and Turl as well, who's working more on the sequence to function aspect of it. And I also I like to thank all the other members of the group. All of the great collaborators have been indispensable with their insights and advice, and I'll take questions.
Thank you for the great talk. Very energetic, too. Okay, let's see if there is any question.
Hi, very cool talk. I was wondering if you looked at if you found sequence features that are present but are missed by your model, like that, say, there's an absent loop or an absent TAD that is present in the real data that's driven by something that's perhaps not CTCF?
Yeah, I mean, that's certainly like a direction that we are going, and we haven't looked as much in depth into that issue as the reverse issue. And there seems to be like an interesting trend, because as you move away from diagonal, you have very few reads. But the biophysical model is really like not bound by that. It still makes a prediction, and so it predicts all of the structure. And I'm like, I don't know if it's there or not. But actually, like people have made kind of like more local data sets that are have more reads. And actually, if you look at that, it is there. So so I think that's kind of one aspect. I mean, and the reverse, like we haven't looked so much at it, but it's definitely there. I mean, right now we're modeling like very, very few things. Of all the things that I know matter, we don't even model all of those, and I know there's more things that I don't know about. So, yeah.
Do you want to give them? Alex and then Max.
Very nice talk. I was wondering, so how to sort of like interpret these vectors that you get, that you kind of create here with the left and right transition rates. Basically, if you had basically two different conditions, two different cell types, you would get like two different vectors from that. Like if there's like a difference in your IC map, I'm wondering how you would interpret that kind of knowledge that you get now from these this new representation basically of the data.
Right. So yeah, you do. Actually, I don't have any cell type comparisons, but I do have the plots, so I can show them. So yeah, you do get things that are different, and they're kind of subtly different. And I mean, the high C itself is also subtly different. The features are mostly conserved, and you can predict the cell type differences, like in this like sequence way where you just use CTCF and some data like CTCF, like a site whether or not it's present, and you have some kind of cell type specific data that tells you like, oh, is the site occupied, right? So it could be like CTCF or like a TAC-seq as a proxy. And the occupancy of those sites is variable between cell types for reasons that I think are not super well understood, having talked to experts about that. But yeah, so there are places where the cohesion complex can't proceed in one cell type because there's a CTF there, but in a different cell type it's missing for some reason, and extrusion just proceeds. So you can capture that, a lot of those elements quite nicely with actually this very simple model.
A very neat talk. So with regards to the first project, there was a paper from Wyeth Wasserman's group that I think was presented at MLCB a couple years ago that was suggesting you learn a complicated 1D function related to each motif, but didn't do any of the interaction modeling. I'm wondering if you have comments about.
Anand.
That's right.
I'm not the best with names. Yeah, so I think I mean, this was almost, I guess, like a similar idea, but like with like completely orthogonal parameters, I would say, right? Like, 'cause the things that we like held constant, they fit, and then yeah, I mean, I think like I thought that was good work, and I think like trying to make the simplest model possible is super informative, and we should all be trying to do that. And and really trying to understand like what are the super important features to get like, even if you get like 90% of the way there. So and I think like, I mean, yeah, somehow the main like in a TAC-seq, like a major nonlinear thing is just the saturation effect, which is like, oh, chromatin can only be so open, like it's open, right? So but for some things like, some of the MPRA people argued that linear models fit great. So, right, and it's almost like it's almost just like down to your opinion whether or not you want to care about kind of higher order terms. So yeah, I think it's really nice complementary work.
There's one more question there.
Great talk. I have also one question on the first model, on the interpretable CNN model. It's really interesting to see the saturation effects. I wonder how diverse are, because the motifs are predefined, are they? So how diverse are these motifs in the model, or are there similar motifs? Could that kind of lead to potential saturation effect? Thank you.
Yeah, so our motifs were like cis-BP because we really like almost followed AI TAC like line by line, and that is what they used. So in some sense, like the total input to our model is exactly the same as the total input to their model, including the input you need for interpretation. So we actually have a ton of motifs in there, just like way more than we actually need. And in the paper, one thing that we do that I didn't show here is we can make the whole model sparse, so like have a lot of the parameters actually go to 0. Which is a little tricky in this kind of like non-convex context. Yeah, just overall, like there's way more. For any given thing, there were more saturation interactions, and overall there were more saturation. Even most of that is like explained by CTCF, but just like saturation was dominant.
Okay, last question.
So I guess for interpretability, this is, I guess, a bit of a broader question, but I guess in the LLM space, there is this sort of movement and research direction of mechanistic interpretability, where you take these like really complex models and try and enforce some sort of constraint on them, like sparsity or something, and then try and identify individual neurons that correlate to some sort of features. What do you think are the trade-offs like between that direction and instead, I guess, making a model as simple as possible?
Well, yeah, I guess I don't have a ton of evidence for this, and I think, with time we will know more. That's why I kind of started with a solar coast example, like as a kind of inspiration. And I think you can think about okay, what would it have taken for pattern matching to succeed at this task, right? Like in the 1600s, you were so far from being able to do that. Like there's no computers, no data, the instruments are all terrible. Like everything is terrible. And yet, you have this one nice, simple model that does amazing things. Can we have something like that in biology? Because we have all the same problems. Like the data is terrible, actually, right? We don't really have great quantification in any of our assays. And we have computers, but I guess we always want more. And yeah, maybe just maybe, I don't know. But maybe I think you can get somewhere very far by just formulating things in a general way.
Let's thank the speaker. Okay, so the last talk will be given by Jianshu. Jishnu, yes. Jishnu.
Okay, good afternoon, everyone. I'm Jishnu. I'm an assistant professor at the Center for Systems Immunology at Pittsburgh. So I'm a computational systems immunologist. I run a computational systems immunology lab where we think about two things: how you can integrate high-dimensional multi-omic data sets in the context of the immune system using predictive and interpretable machine learning, and how you can also use biological networks to integrate different kind of omic data sets to uncover molecular phenotypes of immunological disorders. And we do this across disease contexts in infectious, autoimmune, and alloimmune diseases. But what I'll be talking about today is some of our interpretable machine learning work that we did recently. And I mean, this was really, really interesting. This is an absolutely fantastic conference. But one theme that has kind of emerged is we have all of these deep learning methods, and they focus very, very deeply on prediction, but not necessarily inference. So what are we actually learning? So what I'm going to present today is on the other end, a statistical machine learning approach, which prioritizes inference. We still do very well on the prediction front, but what are we actually learning in the context of explainable AI or interpretable machine learning? And in human systems immunology, when I wear my statistics hat, I say, okay, prediction and inference are both important, but when I wear my biology hat, we really need inference beyond prediction. So this is something that we published in Nature Methods earlier this year. This is following up on a larger body of work with essential regression and other papers, but I'll be focusing on our Nature Methods paper and then show you some downstream applications. We call this slide Significant Latent Factor Interaction Discovery and Exploration. And this was done in conjunction with key collaborators, Alok, Bob, and Amanda, and the work was co-led by two awesome graduate students in my group, Javad and Hanshi. So what is the slide model? So this is a latent factor regression model where rather than moving from the x's, the features, to the y, the response, I mean, all of you are familiar with regression, we first move from the features to the latent factors, the z's, and that's an unsupervised step. And then we move from the latent factors to the y. Now, the biological intuition is very, very simple. Biology doesn't care about what we can measure. In 2024, we have all of these different data modalities: single-cell RNA, single-cell attack, proteomic, metabolomic, but biology doesn't care about what we can measure. Clearly, the true answer lies in something that is hidden, hence latent factors. Now, this is not some great insight. Latent factors have been around in the statistical literature for 2 decades or more, but there were some very nice, interesting properties of what we did with latent factors that allowed us to do this regression model. So step one is we set up a latent factor discovery framework with identifiability guarantees in parameter space without making any assumptions regarding data generating mechanisms, without making any assumptions regarding orthogonality of the latent factors. So we solve this. So other methods, let's say non-negative matrix factorization methods, are non-identifiable. On the other hand, if you have methods like principal components regression, obviously the principal components are orthogonal. So we can prove, this was work done in conjunction with theoretical statisticians at Cornell, we can prove unique identifiability of the latent factors by allowing them to be overlapping without making assumptions regarding data generating mechanisms. The way we do this is we solve this as a constrained optimization problem where we anchor each latent factor based on what we call our two pure variables, two variables that belong only to that latent factor and no other latent factor. So of thousands of features, let's say you have 100 latent factors, so 200 would be, so each factor would have two anchors. Everything else can be shared or not shared in a data-dependent fashion. This is done using the underlying covariance matrix. Now that we have identifiable latent factors, we regress to outcome using these. So here regression makes sense. In an NMF setting where the latent factors themselves are moving around, regression does not make as much sense. But these are stable latent factors, so we can regress to outcome using these. And here what we did, we can prove guarantees on the regression coefficients, but remember that the latent factors themselves can be overlapping. That reflects true biology. So they can be multicollinear. So rather than using the p-values on the individual regression coefficients, we use an overall method for false discovery rate control developed by Emmanuel Candace and colleagues called knockoffs. This is obviously very, very widely used in a number of different contexts. So we control overall false discovery rate. The key adaptation was knockoffs was originally in the observed space. We adapted to the latent space, to the Z's. So we identify what we call significant standalone latent factors. Now, using hierarchical OR logic, we test nonlinear up to pairwise interaction terms and apply knockoffs again. So we identify significant standalone latent factors and then significant interacting latent factors. And we have a model where we come down to a necessary and sufficient set of latent factors to understand the response or infer the response. Note that I have never used the word causality so far, and I'll come to that. As a statistician, and we've actually couple this with causal graphical modeling, you have guarantees even in terms of causality. But as a biologist, I would never claim causality without showing it in a perturbation system. So I say inference or putative causal. So SLIDE has many unique aspects, including identifiability, no assumptions regarding data generating mechanisms. It's not restricted to any single cell or bulk data modality, can handle nonlinearity, correlated latent factors, FDR control, inference beyond prediction, and I'm like comparing it to other methods in this space, including variational autoencoders, partial least squares, principal components, lasso, and so on. So now let's actually look at what we did with this method. So the first data set that we started off with, this was in conjunction with Bob Lafayette's lab, who's a local scleroderma expert. We had single-cell RNA-seq data from skin biopsies, and we asked what are the significant latent factors underlying disease severity. So here disease severity is scored by punch biopsies on the skin, where the skin is scored on a scale of 0 to 3. 0 means completely normal, 3 is the thickest, so it's a surrogate measure for disease severity called the modified Rodnan skin score. And we said not only can we predict, but what can we actually infer or learn about underlying disease severity from the single-cell RNA-seq data. And of course, the first thing we looked at with k-fold cross-validation and permutation testing, how well do we predict? This is kind of the necessary, but to me, the less interesting piece. So SLIDE and essential regression, the two methods that we developed, predict this as accurately or better than state-of-the-art methods. But what exactly are we learning? What is the interpretable part? So this is the first example that I'm showing of the kinds of latent factors that we get. So this is a busy slide. I'll walk you through it. So each box that you see is a latent factor. It's a context-specific group of genes. This structure is inferred using the underlying sample covariance matrix. This is not a predetermined gene set or pathway. We do not have any biological priors. We are simply doing this in a data-dependent fashion. So now, within each latent factor, you have these genes. The color represents the cell type of origin. So you have keratinocytes, fibroblasts, T cells, and so on. Left of the dotted line, you have genes associated with higher disease severity, right of the dotted line with lower disease severity. The green boxes are the significant standalone latent factors. The purple ones are the interaction terms. So without using any biological priors, we were able to recover, for example, in the top right, the pro-fibrotic fibroblast differentiation. This is a special subset of SFRP2 fibroblasts or myofibroblasts known to be involved in scleroderma disease pathogenesis with CCL19, IGF-BP5, well-known markers. We also see crosstalk between myeloid inflammatory signaling and interferon signaling like thrombospondin, CCL19 again, ISGs. So these were well known. Without using any biological priors, we were able to recover. This is what's well established in the field. But we converged on two very interesting things. The first was the crosstalk, not just of fibroblasts and myeloids, which in the field is believed to be the true driver of disease pathogenesis, but also a role for keratinocytes. So we see these interaction terms with keratinocytes. And that was very, very intriguing. And that is something we actually validated in downstream. I'll show you one slide on that. That's a new mechanism involving crosstalk of fibroblasts, myeloid cells, and keratinocytes that we validated. The second was the T-cell responses, which I won't talk about. So slide latent factors we rationalize are meaningful with robust effect sizes. Here I'm showing you like the empirical null. That's the blue if you just pick a size match number of latent factors. And then the green is if you fix the standalone but you shuffle the interactors, the model is at the tail end of that. So you can calculate tail probability, a p-value. So we then said, well, how good are we overall? So we are giving you specific vignettes, but let's say we see how many known genes do we recover. We do better than MOFA plus or VAEs, and in terms of effect sizes of each of these latent factors, we again do better than these. And the latent factors are meaningful. This is one example of where we validated the keratinocyte signature using protein staining. So in the scleroderma, you have these patients with higher levels of keratinocytes. Now we said, let's push the bar a little harder. So are we actually getting something mechanistic? So this is a human perturbation experiment. So a subset of these patients were treated with tofacitinib. That's a pan-JAK inhibitor. So we had patients who were responders and non-responders, and we said, can we infer latent factors that discriminate responders and non-responders? And without using any prior knowledge, the key latent factor that the model found was this IL-6 STAT signaling-centric mechanism, reflecting that depending on how well you respond to the pan-JAK inhibition, you respond or do not respond. So this is recapitulating a known mechanism of action. This is another piece of evidence showing that we are actually capturing something meaningful. Now we switched systems. That was a organismal disease phenotype. So now we went to a NOD model, this is in conjunction with Alok's lab, of type 1 diabetes, where we looked at a cellular phenotype. We said, well, we have paired single-cell RNA-seq data and TCR-seq data, where each cell was labeled based on the extent of clonal expansion of these CD4 T cells, so unexpanded single, low expansion, or medium to high expansion. And we said, can we now understand this cellular phenotype in a disease context? And again, we can predict very well. So the top left plot is in a k-fold cross-validation setting. Slide and ER are predicting clonal expansion very, very accurately, but that's not really the main piece. The main piece is what we learn, what is interpretable. So we see three key latent factors, one involving markers of naive CD4 T cells, some well-known markers of naive like L-selecting or CCR7, celebrated activation markers, inhibitory receptors like LAG3, PD1. But we see these context-specific group structures emerge. And why are they important? They are capturing higher effect sizes than any univariate canonical marker genes. So this is kind of a thought experiment where we said, well, if I talk to my immunology colleagues, they will come up with LAG3, TIGIT, PD1, CD200. These are well-known inhibitory receptors, markers of activation. They are all somewhat higher in the expanded relative to the unexpanded. L-selectin, CCR7 are higher in the unexpanded, but it's not a slam dunk. On the other hand, if we look at these latent factors, they clearly stratify based on extent of clonal expansion. So we are really capturing robust, explainable effect sizes using these context-specific groups. Now, I made the claim across biological domains. That's a big claim. So have we really done this across biological domains? So we've been talking about single-cell data modalities. Now we switch data modalities completely. So this is in the context of schistosomiasis infection, where we have this platform technology called antibodyomic profiling, where using a Luminex-like assay, we can quantify the response of a polyclonal pool of antigen-specific antibodies. So for different antigens, we are measuring the isotopic isotype subclass profiles binding to Fc receptors, and we are saying, using this interpretable model, what exactly do we understand of the latent factors, humoral latent factors driving schistosomiasis infection? This was done in conjunction with Anirudh's lab, co-led by Trirupa in my lab and Anushka in Anirudh's lab. This is now in press, should be online in a couple of weeks. So I'll keep it very brief just to show you high-level vignettes. The first is people know in the context of helminth infection that you have higher IgG4. So if you do the standard DE kind of analysis, the only thing that you see is IgG4 and some Fc receptor binding. But when we ran the slide model, we see a very, very significant model. So I'm showing you the model performance with k-fold cross-validation and permutation testing, but it's not just IgG4 and FCR binding. We also see a crosstalk between IgG1, Fc binding, as well as an IgA1 dominant latent factor. So we are finding novel mechanisms or inferring novel mechanisms. But how real is the inference? And we wanted to put it to test. So we said the first thing is let's validate this in an orthogonal cohort. So we discovered these latent factors using a cohort in Brazil. We tried to validate the signature using a completely orthogonal cohort, Kenyan cohort, and the model still does well. I mean, it goes from 0.9 to 0.7s, but the key thing is both the structure of the latent factors. We discovered the unsupervised structure discovery as well as the prioritization were only done using the Brazilian cohort. We did not see the Kenyan cohort at all, and we still have signal. So we are capturing something that is extrapolable across contexts. What we also were able to do is we were able to look at different mechanisms of active, passive, and prior infection, and I won't go into details of that, but all I'll say is it's not just IgG4 in classical helminth infection. We find a key role of IgG1, and also the unbiased profiling tells us that CD63 and calumenin are the two major antigens that people focus on, but actually find this SM25 specific responses are a critical discriminator, not of active and prior passive, but within prior and passive, and that can help early detection. So this is really, really important. Again, switching gears, this is also in press now. We've worked with Amanda Poholik's lab to identify spatial micro-niche specific cellular programs in a murine model of allergic asthma. I'm only showing you a couple of slides. There's a lot of data in this. I'm focusing only on the spatial data. This is 10x Visium as well as slide-seq data. So these are mice exposed to intranasal house dust mite. That's the allergen. They continuously inhale the allergen, and they have basically a severe asthma phenotype. They are profiled at day 3 and day 5. So we asked this question, which a lot of people in the spatial field are asking, what are these spatial environments or micro-niche specific cellular programs? And what we find is at day 3 and day 5, spatial regions enriched for Th2 and TF8 cells, so these are primary cells, this is in a lymph node, so you have B cells at the boundary, T cells at the center, including TFH and Th2 cells, have divergent cellular programs. At day 3, for the TFH cells, interactions with the B cells are critical to maintain the TF8 cells. This is known biology. But the intriguing part was we found this in this latent factor Z13. The actual pure variable, and it's a key driver, was IL-2RA. And the IL-2RA, IL-2 pathway, we hypothesized this was not known, is critical for Th2 differentiation. At day 5, the TFH B cell signature still stays the same, but the Th2 cells are now engaged in migration to the lung. And with murine models, it was actually validated that spatial niches of IL-2 synergize with IL-10 to drive lung migratory Th2 cells in asthma. So again, this is the kind of mechanism that we are inferring. And for the last piece, I'll talk about characterizing dynamic gene regulatory networks in primary human B cells. So we are a part of the IGVF consortium, so it's a multi-PI grant that Harinder and I have. So this was work co-led by multiple people in our labs, where we are looking at B cells, so naive B cells. This is an in vitro system, but with primary cells, they are differentiated into activated GC-like extrafollicular like cells. And we said, can we now identify cellular programs and gene regulatory network circuits that are discriminating these? So the slide model learns de novo cellular programs discriminating granular cell states. Here I'm showing you the GC versus PB model using really one standalone significant latent factor and one interaction. We can almost completely separate these. This is very nice properties. We tried this like learn from a male donor, predict for a completely orthogonal female donor, learn across dataset modalities from single-cell RNA alone to multiome. It all works well. In a lot of cases, DE analyses completely fail for this. But what is more biologically interesting is slide captures combinatorial control of transcription factors in state-specific GRNs. So we can superimpose these context-specific cellular programs that we learn with inferred gene regulatory networks to identify combinatorial control of transcription factors. I'm just showing you one example in the PB state how BLIMP1, actually PRDM1, works with multiple different transcription factors to control the PB state. This is unpublished work, so I'll not talk about it anymore, also in the interest of time. So in summary, SLIDE can integrate high-dimensional multi-omic data sets with no assumptions regarding data-generating mechanisms to identify significant latent factors underlying immunological outcomes. These are putative causal, and some of the inferred mechanisms have been validated using perturbation systems or model organisms, comes with rigorous statistical guarantees regarding identifiability and corresponding inference, FDR control, and the discovery of nonlinear hierarchical relationships. Using SLIDE, we have identified causal factors of a range of phenotypes from organismal to cellular, molecular, across contexts in health and disease. I talked about scleroderma, type 1 diabetes, asthma, schistosomiasis. We also have other work in myocardial infarction, COPD, that I didn't talk about. And finally, I would like to thank my lab. None of the work that I do would be possible without my lab, and of course, my funding sources. And thank you very much for the invitation to present here. I'm happy to take questions.
Any questions? Yes.
Thank you. Very nice talk. Could you please elaborate a little bit more, like about the idea how you create these latent factors? You know, why 2 anchors, not 3 or 4? What's the biological idea behind this?
Great. So I'll answer it in 2 parts. The first is we create the latent factors based on essentially the underlying covariance matrix, and we are clustering into overlapping clusters the underlying covariance matrix. Getting to Y2, that was the least stringent assumption. So we basically said we don't want to make any assumptions. We cannot prove identifiability without making any assumptions if we allow them to be orthogonal. So 2 was the very least. So you could absolutely have 3 or 4 or more, but 2 is the least that we need to prove identifiability guarantees of the latent factors. And biologically, what we are saying is each of these context-specific groups, cellular programs, humoral profiles, is anchored by a couple of things that belong only to that latent factor and no other latent factor. So we are allowing, let's say, to use a concrete example, let's say you have a certain cell type, let's say in CD8 T cells, you're looking at 4,000 or 5,000 genes, you get 100 latent factors. You're only assuming that 200, so for each of the 100 latent factors, for each factor you have 2 genes. So 200 are locked into each individual, all the other 4,800. So more than 90 to 95% of your data in practical examples can be pleiotropic, reflecting the true underlying biology.
Okay, thank you. Let's thank the speaker.
Thank you.
The food is outside, so we'll have a lunch, and then the afternoon session will begin at 1 o'clock.
Hi, everyone. So I'm going to get started. My name is Philip Fradkin, and I'm going to be presenting Orthrus self-supervised evolutionary and functional objectives for RNA foundation models. And this work was done with my wonderful co-first author, Ian Shi, and Karen Asayev. And then it was supervised by Quaid Morris, Brendan Fry, Leo J. Lee, and Bo Wang. And so I'm going to provide a little bit of like a high-level overview about what genomic foundation models are, although extensively talked about here already. So conceptually, these models undergo some sort of pre-training to leverage unlabeled or, in some cases, labeled data. And at a high level, these SSL objectives work by applying some sort of augmentation to the unlabeled data, and then the model is asked to reconstruct the original input data. And minimizing the error in reconstruction has been shown in other domains, such as NLP, to allow the model to capture the underlying structure and semantics of the data. And the core sort of idea here is that the knowledge contained in these pre-trained foundation models should allow for faster convergence and better performance compared to simple supervised learning. And a couple of examples that could be applied in genomic data sets is, for example, training on mRNA sequences to predict properties such as like mean ribosome load, or non-coding sequences to predict RNA secondary structure, or DNA sequences that we've seen for like promoter or enhancer detection. So there are two core strategies that have been borrowed from NLP for self-supervised learning. The first of which being MLM, where the training consists of reconstructing a percentage of the missing data. And the other is next token prediction, where the model is required to reconstruct the next token in an autoregressive manner. And existing models have implemented these strategies for genomics data. And the core sort of decisions here are, in addition to the objective, is what is the data set that you're going to train on, and what is the underlying choice of the architecture used. So a number of works have used the transformer architecture because it's been so successful in NLP. But you'll notice that these models have actually quite limited context length. And in genomics, we know that there are these long-range contexts that are often so important. And due to the limited context window of these models, they're unable to capture this long-range information. And a work published last year was able to overcome this by using an alternative architecture that's a subtype of deep state space modeling, which increases the context width to 1 million nucleotides, which is quite nice. But sort of taking a step back, we can ask a broader question. Are there self-supervised learning objectives that are maybe better suited for learning RNA properties, which is what we're interested in here? So existing SSL genomic foundation models focus on DNA, and we'd like to model RNA properties because of nice, interesting properties such as like predicting RNA half-life in different applications. So a natural question that I think we can ask is, can we use DNA foundation models for RNA tasks? And this has been brought up during this conference a couple of times. And our sort of thesis is that current DNA foundation models are trained to reconstruct tokens from the entire reference genome, and exons that capture information for this mature RNA are actually much more information dense as measured by constraint compared to intergenic regions. And they overall compose a small percentage of the reference genome, which usually is the data set that these models train on. And so this results in MLM and NTP reconstructing sort of random or uninformative tokens for mRNA property prediction, which we see results in sort of bad, not great performance for these tasks. So we propose Orthros, and we dub it an RNA foundation model, and hopefully I'll do my best to convince you in this talk that it can be built upon. So we pre-train it on mature RNA transcripts to focus on high signal regions. And then alternatively to NTP or MLM, we propose a contrastive learning self-supervised objective. And for the architecture, we use Mamba, which is able to handle these long-range transcripts. And it's a type of deep state space model that actually can be interpreted as having these global convolutional filters that don't have as high of a memory cost that is usually present with transformers that scale quadratically with sequence length, instead scaling with OL log L in terms of memory costs. And so first I want to provide some sort of high-level intuition for contrastive learning, which typically utilizes two related samples by an augmentation, and it minimizes the distance between these samples in the model latent space. And so something we would want to find out is, like, what are the augmentations that we'd be using in genomics? So we have sort of two main augmentations that we use, and the first of which
as being orthologs. And the hypothesis here is that orthologous transcripts that are generated by these speciation events, these sequences are related through evolutionary distance, and they preserve RNA function while non-constrained elements are subjected to genetic drift. And then the second class of augmentations that we use are alternative splicing isoforms that are generated from the same pre-mRNA but are spliced differently. And then the intuition here is that these sequences are more likely to share function rather than RNA transcripts sampled from different genes. And so the central sort of intuition here is that we have these processes that generate sequences such as speciation and alternative splicing, and you can sort of assign positive relationships to them. And they differ in sequence, but most of the time they would have similar function. And by learning to minimize the distance between these pairs of transcripts, we hope that the model would learn regions that are preserved in these, which are then responsible for regulating RNA properties. So I'm going to walk you through what a forward pass of the orthers model looks like. And so first we sample a pair of positive samples identified from these augmentations. And we use standard one-hot encoding of the full mRNA sequence. And we add these additional tracks corresponding to splicing and coding sequence, which have been identified in previous works to be helpful in predicting RNA properties. And our architecture consists of a mix of Mamba layers and layer norms with an additional MLP projector which is commonly used in contrastive learning. And we use a variant of this contrastive loss function, which structures the model latent space, which I will now expand on in a little bit more detail. So we'll represent each sequence as one of these circles with the color encoding the sequence relationship to a reference transcript. And our objective is really to maximize the representation similarity between splice isoforms and orthologous pairs. And you can imagine prior to training, these sequences are maybe uniformly distributed in the model latent space. And during training, we want to maximize the similarity between positive pairs while minimizing that of negative pairs. So we'll go through each element of the loss function and describe some intuition behind it. So we sample these two transcripts ZI, 1 and 2, which is going to be our positive pair. Then we have the unrelated transcripts, maybe from different genes or different species. And to measure similarity between embedding vectors that the model provides for each of the sequences, we use cosine similarity. And so this decoupled contrastive learning loss can be broken down into two terms, the first of which will maximize the similarity between the positive pair of embeddings, while the other will minimize the similarity between this one positive reference transcript and all the other unrelated samples in the training batch. And to sort of summarize, our pre-training data consists of alternative splicing annotations from 10 different species, orthologs identified by the Xenomia project, which collected data for over 400 mammalian species. And this actually results in quite a sizable data set consisting of 50 million unique transcripts with 850 million positive pairs. And another thing that we empirically find is that introducing some noise into our training data in the form of independent masking of two sequences improves performance, which you can sort of think as MLM, but without the reconstruction component. So I guess it's not reconstruction, but we introduce another form of this augmentation. And then we evaluate this model on a couple of mRNA property prediction tasks, such as RNA half-life, mean ribosome load, protein localization, and gene ontology term classification for molecular function. And we do so in two main sort of methodologies. The first of which is linear probing, where we freeze the pre-trained model weights and train a linear classifier on the embeddings to predict a downstream task, And you can sort of think about it as measuring the quality of the model latent space. And the other is fine-tuning, where the model is trained end-to-end on a downstream data set, where we update the pre-trained weights. So, and another important note to point out is that homologous transcripts are kept in the same data split to avoid information leakage. So to evaluate the linear probing, we compare orthros to other self-supervised learning methods. And on the x-axis here, you have different self-supervised methods that we can compare to that I mentioned earlier. And on the y-axis is Pearson correlation for RNA half-life. Another thing to note is that the dashed line up there represents a supervised model baseline, which is Saluki in our case. And we see that the larger orthros actually matches the performance of the supervised model. And this trend is consistent across other data sets where we evaluate performance, where orthros consistently outperforms other models while matching or outperforming, in some cases, the supervised baseline with just linear probing. So this sort of means that we're able to achieve deep neural network performance while just fitting a linear regression on top of orthros embeddings. So the number of trainable parameters here in the linear probing is actually very few. In addition, we can compare by fine-tuning the pre-trained model, updating all the model weights instead of just freezing them. And so we compare orthros to Saluki, a model pre-trained trained to predict RNA half-life, against the dilated residual CNN, which is sort of a classical model that's been shown to work really well in these domains. And then actually an architecturally equivalent Mamba model with no pre-training and sort of this darkish color. And these experiments sort of directly validate the value of pre-training by comparing an architecturally equivalent model, whether this additional pre-training adds information to predicting downstream properties. And we find that pre-training improves performance compared to other supervised methods in all our evaluation tasks. And this sort of directly validates the value of this contrastive pre-training. Another property that we would sort of want our foundation models to have is data efficiency. And this plot, I'll walk you through how to read it. So the outer rings demonstrate the percentage of the training data used for model fine-tuning, while the number of samples in brackets underneath is in the parentheses. And the model performance is indicated by the corresponding line around the plot, and a high volume of a corresponding color would indicate that the model consistently performs well in across all data regimes. And here, authors maintain 73% of original performance using just 45 samples, as compared to supervised models, which are clearly unable to learn with using so few data points. And we find that it's able to effectively operate in this low data regime for most data sets, sort of opening up the door for few-shot learning, which I think can be pretty exciting, where maybe you have an experiment that is expensive, or you have just a few occurrences that in the genome that you want to learn from. And finally, we're able to evaluate inter-sequence similarity in the model latent space by computing similarity functions. So here we're using log of L2 distances from the learned embeddings. So another way to say it, like given a pair of sequences, we can sort of evaluate how similar are they based on the model embeddings. And sort of consistent with the training objective, in yellow here is the alternatively spliced transcripts. So they're quite similar to one another. So if we sample pairs from the same gene, we can calculate their similarities. And this is the distribution in yellow. But you'll notice that there is actually quite a bit of a tail on the low embedding similarity score. And this means that there exist transcript pairs in the same gene that are as different to one another as transcript pairs that are sampled from random genes, which is this distribution in blue. And sort of consistent with our intuition, if we measure distances between transcripts from different genes, but from the same Go class, we find that they have average than higher similarity, which is this red distribution in the middle. And also we find that transcript similarities correlate to domain presence in RNA sequences, sort of hinting that Orthrus learns maybe some sort of underlying RNA functionality. So to conclude, Orthrus is this RNA foundation model trained using contrastive learning. Latent space is well structured for RNA property prediction tasks, and fine-tuning Orthrus on downstream objectives outperforms supervised learning. It's pretty effective in a low data regime, and something that I find exciting is that contrastive objective can be applicable to other foundation models. So you can combine this with like NTP or mass language modeling, and the updated preprint and code and weights will be out soon. And this is like a big many labs were involved in this, and all the individuals are really great, and it was really fun to work on this, and I'll be happy to take questions.
Two related questions. Oh, there it goes. One of them is for the contrastive loss. Maybe this is a standard thing, but to me it looked like that second term has this huge summation over the whole data set, right? Do you actually compute that, or is it some approximation?
Yeah, so the way it's actually done is on other samples that are in the same batch. So yeah, within batch. Yeah.
And then the second question was, did you do a comparison of Orthrus just with pre-training with your contrastive loss and pre-training using a standard masking approach? Just exactly the same model, two different pre-training regimes?
Yeah, we didn't do that.
I think that would be cool to see.
Right, yeah.
Really cool work. I wonder if you try the same here in this domain. There aren't that many good models, perhaps, but like in the protein world, that there's a lot of models. If you could do the same approach and compare with like ESM or other models there.
Yeah. So I guess these tasks that we evaluate on primarily are RNA-specific tasks where like these UTRs are pretty important. I think we could definitely compare it with like ESM embeddings, but I suspect it wouldn't do very well because I think a lot of this regulatory language is encoded in the UTRs. Yeah, but it's something that we could do.
Yeah, totally, but I guess I wonder, like on protein tasks.
Right.
Like just compare the general approach.
Yeah, maybe like gene ontology where maybe it's not as important. Yeah, that's a good point.
Hi, great talk. I have a question. So I noticed in the input here, so the model is actually partially annotated because you have the codon track as well as the splice side track. So I wonder, in terms of comparing to the master language model pre-training, how much of the benefit comes from these additional annotation? How much of it comes from the contrastive learning part?
Yeah, that's a great question. So for the supervised models, the baselines have the 6-track encoding here, but other self-supervised models don't. But we actually have an ablation where we evaluate what is the contribution of all the different augmentations that we use. And I mean, it's hard to compare here, but if you were to use the line where there's no 6-track, but we use all the other augmentations, it outperforms the self-supervised models that are just trained with NTP and MLM.
Thanks again. We're going to move on to the next talk. So the next talk is a remote presentation.
Hi, are my slides visible? All right. I think we can begin. All right. Hello, everyone. Thank you so much for being here. My name is Rishabh, and I'm a grad student at Yale. Today, I'm going to talk to you about RNA FrameFlow, a generative model for de novo 3D RNA backbone design. It's something that I've been working on with amazing company for the better half of 1 year. This was for my undergrad honors thesis. And I had the chance to work with all of these amazing people from the Cambridge Computer Laboratory in Pietro Leo's group. I think there's a Q&A segment at the end, and I'd love to answer questions along the way, if any. Yeah, let's get started. So just a fresher on nucleic acids, or specifically ribonucleic acids, RNA. They're super important. They facilitate protein synthesis, gene regulation. They essentially make you. And over the past few years, there's been a lot of interest in RNA biomedicine. We've heard a lot of action in terms of CRISPR and, because of the pandemic, mRNA vaccines and a whole bunch of other related therapeutics. What's interesting is that from an AI standpoint, most of these RNAs that are important, they have some sort of important structure. For example, riboswitches, ribozymes, they're just examples of many types of ribonucleic acids that can be sort of hijacked or modified to act as really important biomedicine in the body. So essentially what our goal is here is we want to build tools that can design custom new novel RNA and eventually figure out what function it would serve in the broad scheme. So I think in terms of generative modeling for structural bio problems, there's a lot of emphasis on proteins. It's seeing a lot of action. But RNA is pretty similar and pretty complex as well in terms of being a modeling problem. We know proteins to have 4 backbone atoms. The RNA nucleotide has 13 backbone atoms. So in terms of complexity, that's slightly higher in terms of modeling. Proteins have 20 amino acids and RNA have 4 bases, A, U, C, G. And protein structure, the tertiary structure that drives protein folding or protein structure, it's driven by H bonding, hydrogen bonding along the backbone. But in RNA, it's between the bases themselves. And this gives rise to really important or interesting phenomena like base pairing and base stacking that give you that really globular 3D shape. So this work, we wanted to present a geometric generative model for this task of 3D RNA backbone design. And as you can see, this graphic, we want to go from complete noise or like nothing, and we want to generate these RNA structures that are novel and nice and have desirable properties if you want to use them for something else. Just to get the crowd excited, these are some samples generated by our model, and I'll eventually get to how we were able to generate these and some other topics worth considering in this project. So I think since this is mostly a machine learning / bio audience, I think data is such a huge important factor. So for us, we wanted to find a rich enough source of RNA structures, 3D RNA structures, and we were able to come across RNA Solo. It's this database of cleaned PDB structures, and it features these structures from several sources. They could be solo isolated RNAs, they could come from complexes, and altogether, in total, there's about 15,000. And when you do the filtering and when you actually look at things that would be considered useful or nice to generate, we sort of filter that even more and look at any RNA between 50 nucleotides and like 250 nucleotides. And that really gives us a really diverse set of RNA structures of varying length. It gave us about 3,000 structures that we were able to play with. So next, an important task here is I mentioned that RNA backbones, every nucleotide has 13 atoms. Modeling each and every of those 13 atoms would be very computationally expensive. So we wanted to find alternative methods to be able to represent them easily so that in terms of modeling or generation, it's simple. And maybe you could retrieve the full atomic scale and atomic resolution structure easily as well. So in sort of protein land, there's a lot of excitement about using these things called frames, how you could parameterize the backbone using just a rotation and a translation from some global origin. And you can think of these frames as representing each nucleotide. And the RNA structure or the protein structure could be a collection of these frames arranged in 3D space. So that's what we're dealing with. And we essentially take these three anchor atoms, C4 prime, C3 prime, O4 prime, and we perform the Gram-Schmidt operation to get this frame T, which comprises or encapsulates this rotation and relative rotation and absolute position in 3D space. And of course, you might be wondering, hey, there's 10 other atoms you might be forgetting about. How do we get that back? So for us, we auto-regressively place them in. We fill them in one by one using these final output frames after the model has done its thing. And we're able to get back all these atoms by modeling it as these angles, these bond angles that you can easily predict using some sort of MLP or auxiliary network and just sort of place them for each nucleotide in the backbone. So sort of the main workhorse of the engine of our generative model, it relies on this very recent, relatively recent paradigm called flow matching, and this specific variant called SE3 flow matching. SE3, it's essentially the group of all rotations and translations, which basically is the sort of set of rotations and translations that you can sample from to create these diverse backbones or to place these atoms or nucleotides in 3D space. So we do flow matching on this set, and we want to generate a set of these rotations, translations. We want to predict those angles and get the other remaining atoms, and that allows us to recapitulate or retrieve the full atomic resolution structure. So this is just a TLDR on flow matching. It was devised by these two papers specifically in the context of protein design or just relying on things that are from SE3 or any sort of manifold. So it starts off with rotations and translations. You can consider them separately. The math works out really nicely, and you can sort of independently do or sample along the rotations and translations, and that allows you to get those frames. And essentially what flow matching does is it starts off with random noise, both the translation and the rotation aspect of the frames, the nucleotide frames. And essentially we take these steps, these T number of steps, and it allows us to update the atomic positions or these frame positions in 3D space. So you can think of it as starting off with this really, this gas of frames, this really just random point cloud. And eventually you iterate and refine this structure. You update these frames, and these frames are eventually converted to all atom coordinates. And then you finally get the very nice looking RNA structure over here. Hannes Stark has a very interesting flow matching visualization in his video from his recent paper. I'm not entirely sure of forgot the name, but it has a very nice visualization. Maybe I'll include it into my slides later when it's shared publicly. So essentially we're doing the same thing, and in this step where we're denoising or we're updating the frames, it passes through this neural network, which is the denoiser neural network. So between any two intermediate states, we pass it through this denoiser multiple times, and you can think of it as a refinement of frames. This denoiser that performs these updates, it is based on the AlphaFold 2 structure module. It contains very nice components like invariant point attention that allows you to do geometric modeling, geometric attention in a very nice way. And it allows us to, allows the network to be expressive enough to really learn the distribution of 3D RNA backbone structures. So this is just a quick animation of what exactly is happening. You can see that it starts off from this random gas, and eventually you iterate and you update these frame positions. Each of these bubbles that you see, so the color is sort of irrelevant, but you can see these bubbles, and each bubble is a frame in itself. And you can see how these frames are arranged and updated so that they've come to form this very nice looking RNA backbone. So I think now that I've talked about the generation aspect, how exactly are we evaluating it? we were hoping to do something similar to the protein design literature. We wanted some local metrics to show that, okay, if you sort of zoom in, it looks like RNA, not sort of gibberish. At the same time, we wanted to see, okay, the RNA that we're generating, how does it hold up against RNA that exists in the wild? Is it good enough, essentially? So we've come up with a few local and global evaluation metrics that help us see or test, evaluate our model. So evaluation, local evaluation, we hope to retrieve sort of physical quantities, physical measurements like atomic distance, bond angles, dihedrals. And I guess coming from a computer science background, the Earth mover's distance is a very nice measure of similarity. So we're essentially computing this for every single structure in the training data set. And we do the same for our generated RNA. We generate a bunch of them, and we see whether these measurements match, whether we're seeing similarities in the distributions. Next, we want to globally evaluate our RNA, and this is very similar to what we've seen in the protein design literature. So we want to check some sort of validity. So if I generate a backbone, is there an AUCG sequence that can eventually fold into our backbone? This is also called self-consistency. And in the protein design literature, sometimes it's called designability. And for us, we measure this using self-consistency TM scores, self-consistency RMSD, and self-consistency global distance test, which is used in the protein design literature very commonly. Next, we want to check diversity of all the samples that are so-called valid. How different are they from each other? Are they diverse, or are you seeing some sort of sad mode collapse issues where it's just generating one single type of RNA? And of course, we want to check novelty. Compared to everything that's out there in RNA solo or in the wild, what's the new stuff that we're cooking up in our generative model? So here's a note, a sort of word of caution, that unlike proteins that have gone through experimental validation, there's no real notion of designability for RNA yet. We haven't really seen AI-designed RNA go into wet labs, and at least publicly, we haven't seen it being done. So that's why we call it validity or self-consistency. And we use a TM score threshold of 0.45. And the medicinal chemistry literature says that RNA with a TM score above 0.45, they sort of have the same global fold, given how flexible these RNA are in general. So let's get started with some very interesting results that we've seen. Right off the bat, we can see that our generative model can successfully retrieve these physical or geometric quantities. You can see that we match the training distributions between the data set and our generated samples in terms of bond distances, angles, and dihedrals. And if you compute the equivalent of the Ramachandran plot for RNA, which you can think of it as, since there's phi and psi for protein backbones, you can create pseudo bonds between those 13 atoms in a nucleotide, or pseudo bonds between these nucleotides, and compute similar angles, theta and I think that's nu. Yeah, theta and nu. And it allows you to relatively retrieve the underlying angular distributions. And I think in terms of global metrics, we hope to show that our method, if you compare it to existing baselines like MMDiff, which also came out recently, we're able to get a validity score of 40%, and MMDiff being a diffusion model, or like a vanilla diffusion model, we weren't able to really squeeze enough performance out of it, which is why we left it blank. We weren't able to get any valid structures from this model, even after retraining. So I think this is one of the key results that we'd like to highlight, that in terms of validity, in terms of self-consistency, TM score, SETM, we're achieving about 41%. And you see similar very nice values for diversity and novelty. We present further ablations in our paper, so definitely do check that out. We try out different settings for RNA frame flow. We play around with the hyperparameters, and we try to give you the best model, which is open source, by the way, and I'll get to that soon. Next, we want to look at how realistic or how valid these generated backbones are. And you can see that simply because of the structural diversity or the current structural diversity in RNA solo, you get this sort of bias, length bias towards certain sequence lengths like 70, 80, 120. Essentially, we want to be in this zone here where the self-consistency score is very high. And you see low PDB-TM, which means it's pretty novel. You don't really see similar structures in the PDB. So how exactly are we measuring this SETM? I want to go back to this sort of global flowchart that we have. You have our generated backbone, and we pass it through this pipeline where we inverse fold the backbone to get the AUCG sequence. Then we pass it through this model called rowfold, also called E2E fold recently. There was a name change. And that allows you to do structure prediction on top of that generated sequence. And then you would sort of superimpose and RMSD align these, or TM score align these generated backbone and predicted backbone. And if there's a high TM score, you would say that, hey, and if it's above 0.45, you would consider this generated backbone as valid. So that's how we compute SETM score using the grenade and rowfold pipeline. So yeah, TLDR, we can see that our model is able to generate some valid structures, some novel structures. But of course, there's still a long way to go. And it's limited even by the structure predictor's performance. And in our paper, in fact, we highlight that rowfold may not be the best structure predictor out there. So there's definitely a lot of work that needs to be done. So in our current model, we sort of make a trade-off between novelty and validity. We've observed that valid structures, simply because of the diversity in RNA solo, the valid structures, they look like they're from the PDB, or they look pretty similar. They have relatively high PDB-DM scores. But then we can see, on the other hand, that the novel structures, the ones that our model is able to uniquely generate, rowfold is not able to, the grenade and rowfold pipeline isn't able to retrieve it properly, which might be an issue given the current state of tools for RNA design. So again, these are just select samples, and with these metrics, you can see that we've, for certain sizes, certain sequence lengths, we see that trade-off between validity and novelty. Otherwise, the scores usually are pretty decent in terms of the RNA looking locally realistic or locally plausible. And that's one of our strengths in the paper that we hope to highlight. Of course, there's the other side of the coin where our model still is prone to some limitations. We end up getting RNA that where the strands of the RNA are sort of entangled with each other, or they might have these sort of stray loops in the middle of nowhere. That means the frames corresponding to the nucleotides along this stray strand, it was just generated and placed over here, and it has no bearing on any of these, the broader structure of RNA. And you can see that it also generates these really, really highly coiled RNA structures, which could be an artifact of the structural diversity in RNA solo, even with a bit of cleaning and processing. So we present methods in the paper that curb and control these issues. So we include data augmentations of different types, structural clustering, length-based clustering that allows, or length-based sampling, all these tiny tricks that we've tried to really squeeze the juice and really get the most out of RNA frame flow. And again, that's all open source for everyone to try out. And with that, that brings me to the end of the presentation. Thank you so much for your attention. If I had to really conclude, we've seen that these current generation of protein design tools, they are expressive in recapitulating, retrieving RNA structural biology to a certain extent. We've seen that lack of data diversity, it's a broader problem in AI for science, but specifically for these very niche, relatively niche biomolecules that are not proteins like RNA and DNA. And we hope that that's something that the community gets to work on together. So over here, we've just sort of randomly taken frames and we sort of update them to create these very nice looking 3D structures. But then now the next steps would be being more principled, sort of sticking to the science and trying to incorporate notions of base pairing and base stacking to ensure that the diffusion process or the iteration, the flow matching, it's a lot more realistic, and you get structures that don't face these limitations like coils and stray strands. You see something that would mimic something you'd see in the wild. Of course, we want to bring in improved evaluation metrics, maybe bring in secondary structure-based evaluation. So far, we've been evaluating the 3D structure, but I'm sure there's a bunch of exciting things that can be done in really showing that the generated RNA are both locally and globally realistic. And of course, I think naturally, you'd want to extend this to more conditional design settings. So that would come in the form of aptamer design, RNA aptamer design, where you have some sort of target protein and you want to design a binder RNA that has high affinity, has nice properties. And of course, now, given the recent surge of these biological LLMs, very recently, there was an RNA language model called Rhinalmo that came out of a lab in Singapore, and that's really exciting because it gives us nice sequence embeddings to play with. And those would be natural extensions to this project. And with that, yep, thank you. I'm open to any questions. And one last thing before I end, you can check out our paper on arXiv over here, and you can check out our GitHub. I promise our documentation is pretty good. So the paper's over here, and our code base is, it's fully documented for you to hit the ground running. And with that, yeah, thank you so much.
Thank you so much. So we have time for maybe 2 quick questions.
Thanks, Rishabh. Great talk. I had a question about how you parameterize the parameterize the torsions of. Yes, exactly. So do you just have a prediction head that predicts those torsions from the diffuse structure at the end? Is that right?
Yes. So once the frame, the central frame, these anchor atoms go through the whole diffusion process, we have an auxiliary network, much like RF diffusion, or even frame diff, that predicts these individual angles, 8 to be precise, and then we'd autoregressively place these atoms based on these angles.
Got it. Cool.
Does that clarify?
Yeah. And then did you look at diffusing over those torsions as well during, or I guess, I don't know what the analogous word to diffusing is for flow matching, but did you try it all flowing over those torsions as well?
We actually did, and we've come to find that using an auxiliary network is a lot better because in terms of the diffusion, sometimes you run into these issues like the lever arm effect, where maybe if you update these angles a bit by bit, you could have this OP1P phosphorus bond sort of pointing here, right? So we found that MLPs are a lot simpler, a lot more robust to these sort of global angle orientations, and it was just able to recapitulate the angular distribution a lot better. We didn't really study the how close or how similar the MLP distribution of angles was to the ground truth angles, but I'm sure that an MLP would suffice here. Much like, is as similarly used in these protein design models as well. But you can definitely also do angular flow matching on like the hypertorus or something like that, and it would get you these very nice angles. But maybe it might take a bit more optimization, I think.
All right. Thank you. We're going to move on to the next talk.
All right. Thank you, everyone.
Okay. I'm Gokul. Today I'm going to talk about our work on estimating mutual information. So first, I'll give a little roadmap of how I plan to spend the next 15 minutes or so. First, I'll go over the basics. I'll tell you what MI is. I'll explain why we care and why it's hard to estimate. And then I'll talk about our approach, which we call LMI. And then I'll go over some empirical results where we try to understand what regimes LMI can be useful in. And then I'll look at a real problem. So we'll study sulfate decisions in hematopoiesis. And then lastly, I'll just show you how you might be able to use LMI through our Python package. Okay, so first, what is MI? There's a lot of ways of thinking about MI. Today, I think the most useful way of framing it is that it's just
Recording stopped.
It's just a really general measure of statistical dependence. So you can imagine you could have like two variables with a kind of complicated relationship. Maybe it's nonlinear. Certain correlation coefficients might not capture it, right? So Pearson only gets linear ones. Spearman only gets monotonic ones. MI is special in that it can capture any type of relationship. So yeah, I think it's useful to think about it as just a very general measure of statistical dependence. So I'll go over very briefly just a few basic properties of MI. So unlike a correlation coefficient, it's not like -1 to 1. It's bounded below by 0, and that's it. So there's like no negative information is one way of thinking about it. It identifies independence. This is a really important property. So the MI of two variables is 0 if and only if the two variables are statistically independent. And the idea is that the higher the MI is, the higher the dependence is. And then one more property that's really important and really useful is that MI satisfies something we call the data processing inequality. And the idea here is that you can't increase the MI between two variables by transforming one of them. So what I've shown is like not actually the full formal statement, but you can think about it basically as like whatever function f you pick, you cannot make I of fx and y greater than I of x and y, right? Okay, so beyond this, there's a lot of really, really nice properties when working with MI. So first is that it generalizes not at like across continuous and discrete variables and even mixtures of the two. It generalizes to vector, like multivariate random variables. Another really nice property that we won't really get into today is that it can be interpreted in terms of bits. So like the bits on a computer, like for storage. But for today, we won't really talk about that. And lastly, it can decompose into pointwise contributions. So you can imagine that like for two variables, maybe there's like a certain part of sample space that has like dependence enriched there. We can quantify that enrichment of dependence in certain parts of sample space by decomposing MI into its pointwise contributions. Okay? So because of all of these really nice properties, MI is used super widely across like basically every field of science. I picked a few here because they're amenable to nice pictures. I think one application that might be familiar to people in this audience is like for network inference, for like GRNs or signaling networks. You can imagine quantifying the strength of a connection between two nodes as like the MI between those two variables. But one thing you'll notice is that in like basically every application of MI that you see in real world with real data, people mostly only use one-dimensional data. I think a big reason for this is like a technical limitation, which is that it's super hard to estimate MI for multidimensional variables. So a lot of times this is like implicit, right? But it's sometimes written explicitly in papers. So here's one quote from a paper from Alon Klein's lab, which we'll revisit shortly, but it basically says they wanted to estimate MI between single-cell RNA-seq states, but the dimensionality, which is like thousands, is just way too high, so they couldn't actually do it. And instead they resorted to heuristic methods. We'll revisit this one in a little bit. But the reason MI estimation is hard in high dimensions is kind of like the classical reason density estimation is hard in high dimensions. It's like the traditional cursive dimensionality. In practice, what does this mean? So there's like two kinds of popular estimators that people use for MI. There's like an old school, like 2004 non-parametric estimator called KSG, which is really, really powerful when you have small sample sizes. It works really well, but it kind of breaks down after 10 dimensions. It doesn't work super well after that. More recently, people have developed variational lower bound estimators. Two notable ones are MINE and InfoNCE, sometimes used in contrastive learning. And these can work, they can like scale up better to higher dimensions, but they tend to be super data hungry. So if you're like in a regime where you have say like 100 or 1,000 samples, it doesn't matter what your dimensionality is, you're really going to struggle to get a good estimate out of the variational bound. estimators. Okay, so this is kind of the landscape, and the problem we want to solve is basically we want to have a sample-efficient estimator that works well in high dimensions. But at the same time, we can't really beat the curse of dimensionality. It's pretty fundamental. So the approach we're going to take, which we call latent MI approximation, is basically to make just a super strong but potentially reasonable assumption about our data, which can let us estimate MI. So the assumption in particular is that our high-dimensional data has like some meaningful low-dimensional structure. I think this is a really common implicit assumption in basically all deep learning methods, maybe. But OK, so you take your two high-dimensional variables, super hard to estimate MI. What we're going to do is just super simple. We're going to try to learn compressed representations of those two high-dimensional variables to turn our hard problem into an easy problem. And the question is just how do we learn those representations? So one sensible thing to do would be to use like the classical approach of autoencoders to compress our data. So you could imagine if you have two variables x and y, you just have two autoencoders, you learn two compressed representations, then you estimate MI. This seems reasonable, but there are some situations where this won't work very well. So we can look at one example. You could imagine both of your variables have a bunch of independent dimensions, but then they have like one really rare pair of strongly correlated dimensions. If you just have these two independent autoencoders, you have to throw away some information in your representation. Maybe you'll throw away that one strongly correlated component, and you'll end up with this idea that the two variables are independent, when in reality there is some dependence there, it just got lost. So the thing we want to do is we want to try to like take this framework, but we want to like add a regularization, which allows us to like preferentially retain the mutually informative structure in our data. So there's a number of ways you could do this. One really simple way is to just try to predict one variable from the latent representation of the other. So predict y from like zx, if you will, and x from zy. So this is a super simple approach where now you just have 4 decoders instead of 2, but same setup. I think intuitively it might make sense that this will retain mutually informative structure, but we can show this mathematically as well. Optimizing this with like MSE error on the 2 decoders is going to be equivalent to minimizing an upper bound on the difference between the latent MI and the true MI. Okay, so I won't go over the details of how that's done, but you can look in our paper. It's quite simple actually. Okay, so what's the workflow now? So what we do is we take half the data available to us, and we use that data to train or to learn those compressed representations with mutually informative structure. And then we're going to take the other half of our data, put it through the encoders, and then use our favorite non-parametric estimator, KSG, to estimate the MI. OK, so in what regime is this useful? The way we answer this to begin with is to look at synthetic Gaussian data because it has analytically tractable ground truth. So what we do is we just sample multivariate Gaussians, and we control the covariance structure of the Gaussians such that there is a low-dimensional dependent structure between our two high-dimensional variables. So we have like an ambient dimensionality we can call D, which is like the actual like real dimension of the vectors. But then we have some lower number K, which is the intrinsic dimensionality of our dependent structure. And we can change D and K and see how well our estimator works for different K and D. And we can compare this to three popular estimators. So what we do is we study specifically this like low intrinsic dimension, high ambient dimension regime. So we go up to K equals 9 and up to D equals 5,000. And kind of arbitrarily, we choose 2,000 samples for each problem. So we think this is like pretty sparse, especially in this dimensionality. And we use LMI with 8 latent dimensions. So most of the time in this benchmark, we'll have more latent dimensions than the intrinsic dimensionality of the data. But sometimes, for the last case of K equals 9, we'll have latent representation which cannot possibly capture the truth structure. OK, so if we do this, the 3 like popular methods, info, NCE, KSG, and mine, do really well at the top when we're in the tens of dimensions. But then they kind of fall apart as you go further down, especially to the hundreds or thousands. LMI, however, stays consistent all the way down the chart, and this makes sense because it's actually just basically solving the same problem every single time. It's reduced the 5,000 row to the case of the 10 row. But the flip side of this is that as the representations get worse, when you increase the intrinsic dimension moving right on the plot, the performance gets slightly worse. So that's the trade-off there. But we can see, like in this low intrinsic dimension, high ambient dimension regime, most of the time it's just not possible to get a good estimate out of other estimators, and we kind of rescue this behavior with LMI. Okay, so this is nice, but it's like a really, really favorable setting, and also it's Gaussian data, and you might think, well, okay, if it's Gaussian data, we don't even need MI. If we want to capture dependence, we just use CCA or whatever, or like whatever linear method. So what about non-Gaussian data? So it's actually kind of non-trivial to benchmark MI estimators on complicated distributions, because analytically, most distributions are like not tractable to actually know the ground truth MI. So instead, we came up with a trick for creating artificially complicated distributions where we can get the ground truth MI. So I think the easiest way to demonstrate how this trick works is to just show you. So we start with some bivariate Bernoulli distributions. So we have like just a discrete binary here. And what we do is we drop in pictures to replace our binary labels. So instead of a 0, we replace it with a picture of a 0. And instead of a 1, we replace it with a picture of a 1. And we can show under like really, really weak assumptions that the MI between x and y in both cases should be identical. Okay? And here we do it with pictures of 0s and 1s because it's easy to understand, but we could in principle do this with like any data that has like really nice clustering properties. Okay. So we essentially use these types of data sets to benchmark. So here on the left scatter plot, the x-axis is like the ground truth MI of that simple distribution, and the y-axis is the estimate that we make from the complicated distribution. So in the existing estimators, you see two types of failure modes, basically. So for the classical like KSG non-parametric estimator, you see like really, really low variance. It looks quite nice. The only issue is that it systematically underestimates MI. And for the other two estimators, you see they get much closer to the ground truth, but they have this like crazy variance. And with LMI, we kind of reconcile both of these views. We have something with relatively low variance that's also relatively close to the true value. And one nice perk, which we didn't plan for, but it turned out that the runtime of LMI ends up being faster in our hands than the other three existing methods. Okay, so now we'll talk about like a real problem that we try to answer using our MI estimator. And specifically, we'll look at, I guess, cell-fate commitment and hematopoiesis. So we'll work with this really, really nice data set from Alon Klein's lab from the paper that I showed the quote from, where it's single-cell RNA-seq profiles of differentiating mouse hematopoietic stem cells in a dish. So one really, really powerful thing about this data set is that it's lineage traced and sampled at multiple time points. So what happens is the cells get a clonal barcode, and then after 2 days, some of the cells are sequenced, and the remaining cells are split into 2 wells and allowed to differentiate until day 6, and they're sequenced again. So typically in single-cell RNA-seq, you can never track the dynamics of a cell because you have to kill the cell to get its transcriptome. So here in this lineage tracing setup, you do the next best thing, which is you sequence a cell, and then you also sequence its clone, and you know that they're clonally related, right? So what we can do is take cells from day 2 and match them to their twin cells from the day 6 conditions. And the reason that this like little like split well experiment is done is so that you can test essentially the presence of hidden variables in this system. So the idea is that if single-cell RNA-seq captures all of the variables relevant to cell fate, then once you split the cells at day 2, you shouldn't have any increasing information about fate commitment between the 2 wells, right? So the idea is if you know everything about cell fate at day 2, then looking at a day 2 cell should tell you just as much about fate as looking at a day 6 cell in a different well. Another way of saying this, maybe more formally, is this is a test of whether or not the dynamics are Markovian. And ideally, we would do this by estimating mutual information between the samples at day 2 and the day 6 well, and then between the 2 day 6 wells. Okay, so this is what they wanted to do in the original paper, but it turned out MI estimators don't work for this data, so they use some heuristic approaches to try to get at the same thing. But here what we can do is just take LMI and revisit the same problem and just make the estimates. So if we do that, what we find is that the mutual information between the day 2 and day 6 is 0.3, but then between the 2 day 6 wells is closer to 1. So we've seen that the information about cell fate increases between day 2 and day 6. So this says the dynamics are non-Markovian, and the likely reason for this is that there's some hidden information about cell fate that doesn't appear in the day 2 transcriptomes that does appear in day 6. And so this could be that there's like some chromatin state that's seen at day 2, or that exists at day 2 but is unseen by the single-cell RNA-seq, that emerges into a transcriptomic change by day 6. Okay? So with the MI estimate, one nice thing we can do is we can go further than just saying there's more cell fate information at day 6 versus day 2. We can try to pin down when exactly the cell fate information emerges. And the way we can do this is by decomposing our MI estimate into its pointwise contributions from each cell. So here what I'm showing is for all of the early time point cells, their contribution to the overall MI. So purple means low contribution. So at the top, you see essentially no contribution to the MI, or mostly purple cells. And then as you start to go down the differentiation trajectories, you get more yellow. Okay? So we can try to look at this a bit more quantitatively. So you could pull out the neutrophil trajectory, which is like the orange one. And we can infer a pseudotime along this trajectory, which kind of just says how far down the path are you. And then we can compare this to the pointwise contributions at that point. And what we find is that they're kind of flat, and then at some point they just dramatically increase. And if we look at where that is on the UMAP, we see that it's right around this branch between monocyte and neutrophil. So it kind of intuitively makes sense that like once you go past that branch, you start to have like a dramatic increase in cell fate information. So this like intuitively makes sense. Okay, so here I showed one example of using LMI to study cell fate decisions from lineage trace single-cell RNA-seq data. But LMI is just a way of measuring statistical dependence, and it should work in principle for any high-dimensional data with low-dimensional structure. So in our paper, we looked at images, and we also looked at protein sequence embeddings from protein language models. And in some B-sides that didn't make the cut for the paper, we also looked at like estimating the mutual information between modalities of single-cell data, like the chromatin states in the RNA, or RNA and like a site-seq profile. And we also looked at chemical fingerprints, like in the context of small molecule drug screens. But there's really, probably many other interesting questions that you could ask with LMI, and I'd be super curious to hear people's thoughts on things that this could be useful for. And if you're interested in using LMI, I've tried to make it easy to use. So there's a documentation website, which is, I think, a bit sparse, but definitely workable example usage and API are there. And you can install it from pip, and then the estimate can be made essentially with one line of code. There are some like parameters that you can optimize if you want to, but every single estimate in our paper is made with this exact line of code, lmi.estimateXY. So you don't actually need to optimize too much if you don't want to, but you can always get like a tiny bit of better accuracy if you do. Okay, and if you run into any issues using LMI or any questions, feel free to reach out. And with that, I'll wrap up. I'd like to thank my co-authors, Peng, Alan, and Xiaokang, and the Wyss Institute for supporting me, and all of you for paying attention.
That's really interesting. I look forward to trying it out. I'm curious. I think I missed part of the sort of the concept when you were describing this idea that you could potentially have a correlated variable that gets lost in the reduction, and then you showed an example of how you basically have some kind of optimization to make sure that doesn't happen. But presumably you have to do that iteratively over all of the different possible features. Like the optimization is like a min or a max over all the features. Is that how it works? To ensure that no individual feature gets lost, right?
Yeah, so what we do is we actually just predict the entire vector from the entire latent representation. So we don't consider features one at a time ever. We're just basically learning representations which are useful for predicting the other variable.
How is that different from an autoencoder?
So I would say it's an autoencoder with 2 extra pieces. It's like you take 2 autoencoders and then you add 2 extra decoders crossing across the...
I guess I'll just also build on that question. If I understood correctly, you're learning the latent representation at the same time that you're trying to not lose any information, and then you use the latent representation to compute the MIs. Do you have like a double dipping problem that then you have to compute the MI on the test samples?
Yeah, so okay, so I guess a secret, not a secret thing, but a thing that's unwritten here is that there's actually 3 partitions of the data set. So we learn the representation on like 40% or 35%, or I forget what the default is. but we have like a test set within that half of the data. And then we use the other half, which has been never seen by the network.
Yeah, that makes sense.
Thanks. That was really interesting work. So for some applications, people are also interested in calculating stuff like conditional mutual information. So I wonder, like, have you thought about whether this framework could also be applied to like conditional mutual information estimation?
Yeah, I think that's a really interesting question and definitely something we've thought about. And I think in principle, it does fit into this. So there's like one very lazy way of doing it, which is to take these representations and then if you have like a separate z, if that's like a low dimensional z ideally, you can just throw that into the non-parametric KSG estimator, which estimates conditional mutual information. But then there's maybe a more clever way of doing this where you add like a third encoder to support like a high dimensional z, if that makes sense.
Hi, yeah, this is beautiful, and I really look forward to trying it out. Thank you for making software, too. So if I'm understanding right, it's sort of this two-step estimator to get the mutual information. And I mean, that runs into the, the data splitting problem at first. But the other assumption is that doing this cross-modality reconstruction maximizes the mutual information of the latent representation as well. And I'm wondering how you think that compares to something like, if you did have like a one-step estimator for just maximizing the representation, like, is that possible or?
Yeah, yeah. Like so one step is definitely possible if you basically if you like instead of regularizing with cross prediction, if you regularize with like a differentiable mutual information estimator. That's something we explore. It's like buried in the appendix of our paper. But I think the fundamental reason why we don't do that is because those variational estimators tend to be very sample hungry. So if you're ever in the low sample regime, like 2,000 samples or under, you really want to be using the non-parametric estimator because it's just a lot more sample efficient in that regime. So that's the reason we have the two steps. But it's definitely possible to have a one step, and we do empirically compare and show that the two-step works better.
And then the cross-modality, like reconstruction, like it seems empirically this maximizes mutual information, but is there any like analytical?
Yeah.
Okay.
So we have so basically one way of thinking about prediction from X to Y is to think of it as minimizing the conditional entropy of X given Y. And this is a classical result in one dimension. Like you can open a textbook and find this. It's a bound, like upper bound from MSE to conditional entropy. And if we just like stack together this result a couple of times and then like add a few words in between, we get that maximizing the mutual information or minimizing the MSE is the same as maximizing the mutual or equivalent to maximizing minimizing an upper bound. For information. Yeah.
Okay, let's thank the speaker again. So we have a 20-minute coffee break, and then we'll be back for the last session of today. All right, so we're going to start this last session with our keynote talk from Ava Amini from Microsoft Research on a little bit different topic, biophysics and protein design. So it'll be really nice to see her talk.
Great. Thank you all so much for being here and for the invitation. Hopefully we can keep the energy high and enthusiasm high for this last session of the day. And it's my pleasure to present some of the recent work in our group in biomedical machine learning in Microsoft Research based in Boston, specifically focusing on our efforts at the intersection of generative AI and protein design. So to ground ourselves first, I'd like to take you down to the world of the nanoscale, where here what I'm showing you in this image is an illustration from scientist and artist David Goodsell. And what you're visualizing here is the boundary of a nuclear membrane in a cell at 1 million times magnification. And I think oftentimes as computational biologists or computer scientists, we maybe lose sight of what we're talking about when we think about the world of individual molecules and how they interact and function together in concert in a cell. And so what you're seeing here, as you may have guessed, is that these individual molecules that you see colored are proteins, right? As we all know, biology is actuators. And so our real question that's motivated a lot of work in our group is this question of generative design, specifically asking whether we can harness the power of computation to design new proteins to unlock new functional or therapeutic capabilities. This generative ability would unlock new opportunities across industries, applications from basic biology all the way to ultimate therapeutic implementation. And so to highlight kind of what we think about when we think of these applications around protein design, 3 examples that I'm showcasing here. The first is the creation and engineering of new molecular tools, such as gene editors or base editors that can really transform and advance synthetic and molecular biology, to the design of new enzymes that can catalyze new chemical reactions that are unseen before in nature. And ultimately, where a lot of my interest lies is in the design of new therapeutic agents, biologics or even therapeutic enzymes that could ultimately be deployed therapeutically, such as the enzyme asparaginase, which with some clever protein engineering has actually been clinically approved as a treatment in certain leukemias. And so really our first question is, okay, if we are going to set off on this task of trying to build AI to understand and design proteins, we have to think about how we represent a protein and what scale or what modality we want to tackle this problem at. And of course, right, in this community, there's been tremendous work across individual pillars of this pipeline, right, from protein language models such as the ESM family or the CARP family, to structure generation models like RF diffusion and others, as well as sequence to structure prediction models like Rosetta Fold or AlphaFold. And ultimately, where we in our group like to think about this problem is, can we integrate across modalities and leverage principles of biological grounding to build new AI models for protein generation in a multimodal fashion? And so today I'm going to focus on our efforts towards this end, speaking a little bit about some work that we've done in biophysically informed methods for structure generation, but really spending the majority of the talk and the majority of our focus on sequence generative models. And so thinking first about structure, what we were really motivated and inspired by was to think about how proteins naturally and biophysically fold to adopt a final structure. And as you may know, as a protein is being made and translated in the cell, initially it can adopt kind of this random unfolded configuration. And during that folding process, you have this sampling of different conformations to arrive at a final 3D configuration that's going to be energetically favorable. And what's really beautiful about protein structure is that we can very precisely and perfectly define the backbone structure according to these bond angles between the atoms that comprise the protein backbone chain. And if we think about folding as a high level of abstraction, you can think of it as this analog that's connecting these unfolded states corresponding to a random or pseudo-random set of angles, and a folded configuration where we have an inherent structure to the set of angles that define the ultimate 3D geometry and shape of a protein's backbone. And so looking at this process, a student from James Zhou's labs, who interned with us previously, Kevin Wu, had the concept of thinking about this native biophysical process of protein folding and using it as inspiration to define a new generative model for protein backbone design. And the concept here was, if we can define this process of going from an unfolded state to a structured and folded state as a stepwise denoising, if you will, that we could perhaps pose this as a diffusion problem to now build a generative diffusion model to do exactly this unnoising process framed as a design one. And so that is exactly what Kevin spearheaded and that we implemented in a model that we call folding diff or folding diffusion, where you can see some samples from generation stepwise from this model here on the right that effectively learns to unnoise from a random configuration of angles to learn the patterns of a structured state. And really the backbone to our method is a powerful class of generative models that are known as diffusion models that have been introduced previously in a couple of the spotlight talks, where the basic concept is you have some data distribution. You're going to perform a deterministic corruption process stepwise to now progressively add noise to your data samples and use this to constitute a training set of examples from which you can learn a model to iteratively learn how to remove the noise by looking at these pairwise consecutive steps in this noising process. And so the concept is, right, you have this controlled corruption scheme, and you can then parameterize a neural network to learn how to iteratively remove noise from these examples to then be able to generate new samples at inference. And so with this backbone and methodological framework in mind, what we did was pose this problem of structure design over these sets of protein backbone bond angles. And the concept here is, right, we define these angular sets. We can corrupt the data progressively over a fixed number of time steps until we get to a state of being indistinguishable from random noise. And then we can learn a neural network model in a generative fashion to be able to reverse this noising process such that we can generate realistic structures. And what I'd like to focus in on is kind of how the parametrization of this occurs with this angular representation. What's really beautiful and elegant about this framework is because we are defining our data set over the bond angles of the protein backbone, all the invariances and the geometries that we want to preserve about protein structure are inherently represented in that angular representation. So what this means is that the neural network that we learn as the denoiser can actually be quite simple, a vanilla transformer without any specific equivariance constraints or geometric constraints. So the key aspect of this formulation is kind of visualized here, where in folding diff, we've taken the equivariance requirements and baked that into the data representation itself, in contrast with some other approaches that have been proposed to now diffuse over 3D coordinates, where you have this diffuse point cloud of the atom coordinates in space, and then need to integrate these equivariance requirements into your neural network architecture itself. So I'm going to be very, for the purpose of time, really focus just a highlight of sampling of some of the empirical evaluations we did. We considered, okay, first of all, how well does our folding diff model actually recapitulate the natural distribution of these internal angular coordinates? Can we actually recapitulate structural motifs and key elements in protein structure that we would expect to see? And can we assess some of the generations for designability and diversity criteria? Now, I will say that, you know, as you may know, right, this space of protein structure design is a very, very active one. And I think what was inspiring about working in this field and about this project in particular was we proposed an alternative formulation that has simplicity with respect to the efficiency and simplicity of the architecture, kind of inspired by the biophysical constraints of the problem and the actual biological question we're posing. So we really think that the open sourcing of our code, of our models, pre-trained weights and everything, really hopefully provides a building block for the ongoing work that has gone on to expand diffusion models for protein structure and now move to new classes of generative models as well. So so far, right, we've just given a taste of the work that we've done on structured design. But really, right, this is one part of the picture. And I would argue that ultimately, if we want to think about the general capabilities that we want for functional protein engineering, the real question we're trying to ask is, can we learn what amino acid sequences can give us a desired protein function? And so motivated by this question, our next step and our inspiration here was to ask, OK, what if we think about removing structure entirely and approach this design problem from the lens of sequence first, and pose the question of how can we achieve these principles of controllability by learning over protein sequence space? And really our underlying hypothesis was that sequence could constitute a universal design space for proteins. And the motivation behind this was that we have a tremendous scale of sequence data available. And furthermore, that when we want to engineer proteins, ultimately we realize them as their amino acid sequences. Whereas with a structural approach, there's this need for an indirect step to infer a sequence that would then fold and generate a particular structure. Furthermore, biologically, we know that many, many proteins fall outside this traditional structure-function paradigm, namely proteins that have intrinsically disordered regions that function very critically in a variety of cellular activities, and these traditionally have been inaccessible by structured design methods. And perhaps most importantly, from a data perspective, the number of sequences that we have available across data sets like UniRef greatly outnumbers, and by orders of magnitude, the number of structures that have been experimentally solved. And so this was really our inspiration and motivation to now think about building out classes of models that could learn from protein sequence data to be able to effectively generate new proteins well unconditionally, and ultimately give us a good foundation to be able to move towards this ability to prompt the model with different forms of conditioning to now be able to move towards this idea of functional design. And so the model that we have developed towards this end is a model that we call EvoDiff, which stands for Evolutionary Scale Diffusion. And this is work that has been really spearheaded by an amazing applied scientist in our group, Sarah Alamdari, together with a really world-class team of complementary researchers and scientists at MSR. And the concept is, right, we want to be able to learn from this natural sequence diversity to be able to now generate new protein sequences that can hopefully execute desired functions. And like folding diff, the backbone modeling framework we've adopted is, again, a diffusion-based framework. But now we have a critical difference in that we're no longer in a continuous scheme of thinking about protein angular coordinates or 3D XYZ coordinates or other approaches. And really what we want to represent and learn over is this discretized representation of proteins as amino acid sequences. And the concept here is now, instead of having some noising scheme where we're adding Gaussian noise, we need to define a way to corrupt or effectively noise our source data to be able to train this diffusion style model. And what we adopt is a framework called the order agnostic discrete diffusion framework, where each step of the noising process is defined by the masking or corruption of these individual amino acid tokens in the sequence. And we perform this stepwise to be able to arrive at the final corrupted sequence, and as before, train a model to now learn how to undo this corruption iteratively. To train the EvoDiff model, we leverage a data set of on the order of 50 million unique protein sequences and a baseline architecture of a 640 million parameter CNN. Now, methodologically and from a machine learning perspective, what is really interesting about this discrete diffusion framework from a more theoretical grounding is that it actually provides a generalization of two other common classes of language models that are out there, namely mass language modeling and traditional left-to-right autoregressive language modeling. And so in this order-agnostic framework that EvoDiff adopts, we actually perform this corruption in every possible order, and each step of this iterative process is learned from a denoising perspective. This contrasts with traditional autoregressive models where you have a defined start token, you are tasked to predict the next token, and you perform this stepwise decoding in a single decoding order. While this gives us really strong performance in unconditional generation and actually learning the data distribution, the limitation, as we'll see later on, is that it's less flexible in terms of how you can try to condition on subsequences or parts of the input that you may want to hold fixed. And in contrast, right, mass language modeling, another common language modeling framework, amounts to learning the decoding over a single step of this objective, right? You have a fixed set of mass tokens, and you learn to predict the correct identities of those tokens at those mass positions. Inherently, this is not a generative task in and of itself, and it requires to actually sample from the trained model to now produce new sequences. And so diffusion gives this generalization over both these model classes, which lends distinct advantages. So in practice now, how do we actually generate new sequences at inference? With our trained model, we can now define a starting state of entirely corrupted tokens, and the model is effectively stepwise decoding and producing new amino acid tokens in this iterative fashion. And so in this visualization on the left, you see the outputs of our EvoDiff model stepwise to generate a new sequence from scratch. And on the right, what I'm showing you is a visualization purely post hoc in prediction of the resulting structure from that generation. And so importantly, what I hope you can appreciate is that this learning is entirely over sequence space, right? There's no notion of explicit structural information that's provided to the model. So with these two elements in hand, right, our discrete diffusion framework and this expansion over these evolutionary scale data sets, we wanted to focus our evaluations of the generations across these three axes, thinking about, okay, are these generations even biologically plausible? Are they designable? How diverse are they? How well do they cover the natural space of protein structure and function? And ultimately, what capabilities can we start to move towards from the perspective of conditional design? And so our first evaluations were really thinking about, okay, for an individual generated protein, how plausible are these? And so we adopted a framework where we think about generating a sequence, looking at its associated structure, and thinking about, okay, how plausible is that structure? And moreover, how consistent is that structure? Can it be realized by an amino acid sequence? And so in these plots, what I'm showing you is a pairwise correlation of the structural confidence of the predicted structure from samples from the independent test set against the sequence-wise self-consistency. And so ideally, you want to be in this lower right quadrant of this plot where you have high structural plausibility and confidence and consistency from a designability perspective. And what we can appreciate is that EvoDiff's generations kind of span this natural distribution fairly well. And if we look at some individual protein sequences generated by EvoDiff and look at their predicted structures, they're structurally sound and plausible across a range of amino acid lengths. So this evaluation is more on the level of individual generations. But now, what if we think about the overall distribution of samples that EvoDiff is producing? So thinking more now from a distributional lens, what we considered is how well our model could recapitulate the natural space of protein function and structure. And so in this analysis, what we did is we took test natural sequences, used an independent protein language model to generate embeddings for those sequences. And these embeddings have importantly been explicitly benchmarked to show separation across different functional classes of proteins that exist. And we can look at and compare the distribution across this embedding space for natural sequences compared to generations from EvoDiff, and compare these and visualize their overlap to see how well qualitatively EvoDiff is recapitulating this functional space. We can also try to measure this quantitatively by looking at a distance metric that measures the similarity across this embedding space. Ultimately, though, right, this plot is not super meaningful without understanding how other approaches or baselines are performing. And so that's exactly what we did next, comparing the generations from EvoDiff with samples from a traditional autoregressive model, which does slightly better in capturing this distribution unconditionally, but has less flexibility in terms of its power for conditioning. We can look at samples from a masked language model like ESM, where now we see a stark difference in the quality of the generations produced. And importantly, by the state-of-the-art sequences that are inverse sequence designed or inverse folded from a state-of-the-art structure generative model like RF diffusion. And here you can see that in a structural method, there's a strong bias towards a particular slice of this functional space. Now that may inspire the question of, okay, well, if this is over functional space, how well can these methods compare in their coverage of structural space? And again, this is exactly what we did next and performed a similar spirit of analysis, where now we're looking at an amino acid prediction of a secondary structure element and looking at the distributions of that analysis for generations from EvoDiff, as well as comparing to the test distribution. So first, if we compare now independent sets of natural sequences together, what you can appreciate is that there's this distribution over both helical and strand secondary structure elements. But if you'll notice in this bottom left corner of this plot on the right, there's this region that appears to be neither helical or neither strand in natural sequences that corresponds to regions that are intrinsically disordered and don't have an explicit secondary structure. And what is really exciting and encouraging to see is that with generations from EvoDiff, we recapitulate this same distribution of structural elements very, very well. And this is in contrast most starkly to structural generative methods that are quite biased towards preferentially generating structures that are biased for helical elements and missing this element of disordered space. And so the bottom line here is that by looking at these generations at a distributional level, that we can begin to understand how well our model is able to capture these elements of secondary structure that mirror those of natural proteins. So now, perhaps most excitingly, is, okay, now this gives us a framework and a basis for generating these unconditional samples, just generating samples from the model. To be able to move towards a paradigm to now think about functional protein engineering from a sequence-first perspective, we really need to think about our abilities to do conditional generation. And what's particularly beautiful about the discrete diffusion framework is that it gives us a very natural and inherent way to do some forms of conditioning inherently. And the concept here is that because we have done this discretized stepwise corruption and learned over all possible corruption orders, what that means is that at inference, we can fix some subsequences in a sequence and inpaint or generate the remainder of the sequence. What this amounts to in practice is masking those sequences that we want to generate over and decoding those portions, right? And that's, again, owing to the fact that this learning objective has learned over all possible decoding orders. And so in our work, we explored 3 different approaches to leverage this conditional generation capability. The first was to think about now informing a more stringent evolutionary constraint on the generation itself, where we could supply an alignment of multiple protein sequences of orthologs and now learn over this MSA rather than over a single protein sequence. And the concept here is that the entirety of the MSA, except for a single reference protein, a query reference, can serve as a form of conditioning, where now, given that context of the remainder of sequences in the alignment, we can generate a new query. We can also do kind of inpainting or scaffolding tasks, again leveraging this capability to mask these arbitrary subsequences in the overall sequence and be able to decode accordingly. And so I'll give you a taste over the next 10 minutes or so about how we evaluated and generated new protein sequences over these 3 types of application tasks, thinking first about this MSA-guided generation. So what is really beautiful about MSAs is that this is kind of a compressed representation of evolutionary information, where we can perform homology search to construct these alignments. And now the only difference as before is instead of giving a single protein sequence as input to the model, we can now leverage the entirety of the MSA and again perform this type of iterative masking task to now train a model that we call EvoDiff MSA, which is very similar in spirit, but now capable to operate over entire multiple sequence alignments of proteins. And again, we draw inspiration from this evolutionary perspective to curate a data set, to leverage a data set that spans MSAs across many different organisms and many different biological functions. What this capability to learn over an MSA in a single generative model gives you, and this is also in line with some of the work Sergey presented yesterday, is that traditionally, right, if there's a particular protein family that maybe you really care about, if you wanted to train a local model over that, you would have to curate that data set and learn the model to now be able to sample from it to generate hopefully new proteins of that family or class that you care about. But by leveraging the evolutionary information inherently encoded in an MSA as context, what this means is that we can effectively do an amortization of these local models over a single unifying model that's learned across all these different MSAs. And to showcase what this kind of enables, learning over these multiple sequence alignments to now, given the context of a set of aligned proteins, be able to generate a query sequence conditioned on that information, we see that we can actually improve the quality locally of these generations to now produce generated protein sequences with higher structural plausibility and better and lower self-consistency. And importantly, we're able to tread this balance between maintaining structural conservation where we care about it, meaning that we are able to match the constraints of the multiple sequence alignment provided as context, while still generating sequences that are novel with respect to the original information that was in that MSA. And so we think that this gives a flexible framework to be able to now condition on families of evolutionarily related proteins to balance this line between structural conservation and sequence novelty. So now thinking about, okay, right, the whole premise of this sequence is all you need hypothesis was that it would give us flexibility to do access regions of protein space that fall outside the traditional structure-first paradigm. And so what we were really excited about was exploring the possibility to now generate regions that could be contributing in intrinsically disordered proteins or intrinsically disordered regions. And again, to remind you why we had a hypothesis that we could achieve this, drawing back to these secondary structure analysis results, what we had observed was that in generations from our EvoDiff model, we were able to cover this secondary structure space, including this region of not inherently helical or not inherently strand-like elements. Meanwhile, structural approaches like RF diffusion are completely missing capitulation of this region of secondary structure space. And so, motivated by this, what we set out to do was now to inpaint or generate intrinsically disordered regions within a protein of interest. And so the framework we set up was either using the single sequence EvoDiff model or given information and context from a multiple sequence alignment, could we mask out the region corresponding to an intrinsically disordered region within a protein, and then ask the model to, given the context of the surrounding regions and optionally the information from the MSA, generate or inpaint that region that we had masked out. And we can do some in silico analysis to now score that generated region for the likelihood that it is actually disordered. And so when we did this evaluation, what we were really excited to see was that across several examples, we were able to do this quite effectively in silico, as measured by these in silico evaluations of disordered regions. And so what you're seeing here on these bottom plots is now in green highlighted that masked portion that we masked out as conditioning to the model, corresponding to the disorder prediction score for the generation on the top versus the true intrinsically disordered region on the bottom. And what you can appreciate is that these sequence disorder scores correspond quite well across these examples. And so right now we're in the process of validating this approach experimentally to now look at some of these generations, particularly focused on proteins with intrinsically disordered regions. So finally, right, the kind of a big paradigm in this realm of protein design is this notion of conditioning on a motif to now scaffold or generate a protein given that information as context. And biologically, this motivation is very grounded in the fact that protein function is often mediated through these interactions where there's a particular functional motif stabilized by the structure of the protein overall, and that motif is what's mediating the function or the interaction that the protein is responsible for. And so we pose this counter-paradigm question of whether we could achieve success in scaffolding these canonical structural motifs using our model that has only learned from sequence information. And the short answer to that question is yes, that we can, specifically by this perspective of leveraging this subsequence context and masking as a form of conditioning. And so in this example where we can see that principle kind of come to life, what I'm showing you here is the structure of calmodulin, which is a calcium binding protein responsible for calcium transport and signaling in the cell. And in green is that calcium coordinating region that is structurally and functionally responsible for that binding function. And here in the bottom is the amino acid sequence of calmodulin with the residues corresponding to that functional motif highlighted in green. What you can appreciate, right, is that these are discontinuous in sequence space. Even though structurally we have this continuity, they're discontinuous. So what we can do with the EvoDiff is leverage these fragments, right, of these functional residues as a sort of biological prompt. And now, given this context of the motif, design a brand new protein sequence around that part, step by step from the bottom up. And so at the bottom is the visualization of the EvoDiff generation, and at the top is the predicted structure for the design resulting from this process. And again, at the top is purely a visualization, right? There's no structural information that's provided explicitly to the EvoDiff model. We were able to run this evaluation and analysis over a suite of different examples and benchmarks provided by RF diffusion. And what's really interesting is now when we compare the fractional success rate of generations from EvoDiff relative to RF diffusion, we see a comparable success rate in the terms of how many designs we can successfully scaffold. Digging a little further and deeper into this analysis, if we look at the relative success rates of the sequence-only model versus the MSA model, we see that they're not perfectly correlated across these scaffolding tasks that we evaluated on. And very interestingly, that if we compare success rates for RF diffusion versus EvoDiff, they're very divergent, suggesting that there could be complementary strengths comparing a structure-based model versus a sequence-based model. A couple of noticeable examples amounting to instances where the sequence-only model or RF diffusion has a relatively high success compared to an MSA model. In fact, this design has very few homologs that exist naturally. And from a novelty perspective, as perhaps you could expect, we can achieve higher novelties with a sequence-only model versus when we give that alignment information as context. And so from this perspective, what we're really excited about is that this is just one first step of a form of conditioning that we can leverage with this flexible diffusion framework. And ultimately, we see EvoDiff and this suite and class of models as a foundation to move towards this principle of controllable protein sequence generation, both from the lens of unconditional design, but also from across these types of in-painting or scaffolding types of tasks that are flexible across structural problems that we may care about, and also problems that fall outside this traditional structure-based design paradigm. So with that, I'll close by mentioning that our efforts in our group around protein engineering and protein design are just one piece of the broader effort across BioML at MSR. And all the code, model weights, data for both EvoDiff and FoldingDiff are open source and publicly available. And really what we see is the coalescing of efforts across these scales of biology from the level of individual molecules all the way up to the patient level, such that we can build generalizable systems that can reason across these modalities to try to power relevant tasks in biological design and discovery. So with that, I'll conclude and really thank all of those in our group at MSR BioML based in New England, and happy to use the remaining time and for any questions and to talk further. Thank you.
Hi, thanks for your talk. I think, to me, beyond improved performance, the EvoDiff MSA unlocks completely new functionality. I think what protein engineers can use being conditional generation of variable sequence length. by just introducing the knowledge and understanding of this gap token, which many of the sequence models don't have. Did you run any kind of ablation on in-painting gaps or diffusing two gaps?
Yeah, it's a great question. So this is actually something we're actively working on as we speak to give the flexibility across indels to account for that gap information. So hopefully stay tuned. Yeah.
Hi, great talk. I noticed that during like the functional scaffolding portion of it, there was still like, it seemed fixed length of amino acids in the sequence. I was wondering if you guys were looking at maybe making that a little bit more dynamic, because some smaller peptides might be useful for some therapeutic applications or like manufacturing purposes.
Yeah, absolutely. So here in the evaluations over that scaffolding portion, you have to pre-specify that overall sequence length, right, for which you want to generate over. And what we did in this first pass of evaluations was, in the motif examples, conserve the residue-wise distance in between the discontinuous segments of the motif, and then given a fixed overall length, experiment with different placements of that motif and sample over this possibility as well. So you're absolutely right that thinking about more clever ways and just evaluating over toggling that length in between the elements of the motif will give greater flexibility there. Yeah.
Thanks for the great talk. I'm wondering that so the secondary structure or this 3D structure can be also tokenized, right? And we can also do mask diffusion. Do you think that's useful, or yeah, I want to know your opinion about that.
Yeah, absolutely. So recently there has been a surge of this concept of tokenizing structure and then trying to even predict it, generate autoregressively according to that. I think, so the structural perspective is that you can never do as well with tokenization over structure than a pure, continuous or frame or angular representation that inherently represents structure natively, because of the fact that the tokenization is going to be some form of compression, right? So there is going to be some element of information loss. I think I'm eager to see what are the relative trade-offs. My intuition and hypothesis is that the approaches that that do not take a structured tokenization approach will yield better results in terms of designability, consistency, et cetera, from a structural evaluation point of view. Now, there's a separate question of if you want to integrate across sequence and structure, what is the best way for doing that? Diffusion flow matching gives you a framework for thinking about cross-modal learning as well, and you can think about that in different ways. I think it remains to be seen. It's still an open question, but I have my opinion and happy to talk further on some of the things we're thinking about as well.
Okay, thank you very much.
All right. Hi, everyone. My name is Romain Lopez, and I'm a postdoctoral fellow at Genentech and Stanford, and I will start my lab next year in September at New York University at the Current Institute in Computer Science and at the Department of Biology. Today I will talk about optimal transport and perturbation screens. So I will not talk about sequence models. I hope that's all right. All right. So this was a work, a team effort that was led by Jiang during her internship at Genentech. And this was also a collaborative effort with Charlotte, Luca, Pinello, and Jiang and Luca are both at Harvard Medical School, and also with Aviv at Genentech. And this work was, we were pretty humbled to receive the Base Paper Award at the ICML workshop in AI for Science earlier this year. So I will start by motivating why optimal transport can be important in this specific context, and I will talk about the specific problem I'm interested in here. Then it will motivate the method, and I'll go through some of the algorithmic advances here. Talk about the baseline that's pretty largely and broadly applicable and that we could try here. And then we'll see on some multimodal data how this performs. So let's get started. So high-content CRISPR screening have been very valuable and has really transformed how we think about functional genomics. When I talk about high-content CRISPR screens, I usually think about cell lines or primary cell that express CRISPR. And we transduce, we deliver guide RNA to the cells to be able to edit the genome and basically have those knock-in, knock-out, or knock-down some genes inside of the cells. We obtain, it's called a pool screen, so we obtain a bunch of cells with different perturbation inside of them. And then I write here complex phenotype, meaning there are different ways of then measure some biological insight in those cells, and I will go through different of those measurements, and we'll think about them as modalities. So this kind of framework is very powerful and has been applied for understanding genetic interactions, understanding mechanisms of cancer resistance, immune cell function, as well as cellular differentiation. It's important to recognize that, so there are different ways, again, of profiling this pool of perturbed cells, and each modality has its own strengths and limitations. I will talk about two specific modalities. One that's very popular is called Perturb-seq, in which we will basically perform single-cell RNA sequencing on this pool of cells, and we can also retain the guide information, so we know what we perturb and we know what is the outcome in terms of gene expression. We can say that the readout is unbiased because we observe all the genes in the transcriptome. It's relatively safe to say that it's interpretable in terms of outcome, because if we put a few genes together based on pathways, we can have a broad sense of which pathways are up and down regulated after those perturbations. And it's also very safe to say that it's relatively expensive and difficult to scale. So there is an alternate type of modality, which is based on microscopy imaging. And this assay called cell painting consists in taking those cells, dyeing with chemical dyes the organelles, or some organelles with some broad set of markers, and putting them under the microscope to see what the morphology of the cell looks like. And we can somehow featureize it. And it's interesting to think about it as also an unbiased readout, because even if you have enough of those organelles, even if they're not very specific in terms of pathway, they still give you a broad sense of how the cells are changed after perturbations. It's very difficult to interpret them. For example, if you think about immune cells and then you have those broad markers, you will not be able to read off directly what you're interested in, but it might still contain that information. So let's leave it at, it's not super interpretable, but it's very scalable in terms of number of perturbations we can profile, and potentially we can do genome-wide screens in this kind of setting. It's not very difficult. So this motivates the idea of can we get put this data together to try to get in silico Perturb-seq, for example, for many more perturbations by diminishing the cost of those experiments. So as a mental thought, I guess, that would work as follows. For many perturbations, we can profile single-cell RNA sequencing as well as imaging. We learn a model that will help us translate from one modality to another. So here, for the exercise, I will pick predicting gene expression from imaging. And once we have trained that model on the data that we have, we can basically pick the other perturbations for which we have only one modality and try to predict and fill this in. And we can call that in silico Perturb-seq from the imaging data. So the most important and critical step here is how do we build this modality translation model. And let's try to, again, go through a very cartoon type of example. What's happening here is basically we measure for several cells some features or some cloud points. And we have two modalities, so two different spaces that are broadly incomparable. They live in different scales, etc. Each point is a cell, and we have colors that correspond to somehow labels or perturbation. And so I can take a particular cell, I can have some kind of model or a neural net that helps me pass, translate, and go through the other space. I arrive somewhere, I don't really know where I am, and the reason is I don't really have paired measurements. So these methods are destructive in a sense, meaning that I can never recover the exact cell that is in the matching modality. So let's say I arrived in another cell, and that measurement is called yj, starting from cell I. And I can't really use that for a supervision signal, for example. But maybe if I have an oracle or somebody that's really nice to me telling me, hey, I think those cells are actually related, or maybe those cells are completely unrelated, and I have a matching matrix, T, then I can somehow still, as a starting point, write an objective function, which is a weighted nonlinear least square type of problem. So I try to predict one modality based on the other and kind of weighting those pairs of observation based on how relevant they are. And I'll call this T matching matrix. I want T to be doubly stochastic, meaning that it kind of is a probability distribution for every cell. I associate a probability distribution over the other cells on the other modality. So this is kind of the setting. Now, everybody will look at me and ask, how do we get this matching matrix? So I don't know yet, but what we can first do is we can go through the exercise of thinking, what if I have paired data? So if I have paired data, I will pick the identity matrix because I know how to pair the cells. And then I know that my model F will be basically learn with supervised learning, and it's kind of the best thing we can do, and there is not much interesting things to be done here. Now, if it's not the case, maybe we have some labels, and we can say, when I pair my observations like this across modalities, I want to make sure that for every cell, I try to predict the sequencing based on the imaging of only the cells in the other modality, but that have the same label. And when I do these kind of things, I'm kind of doing some shrinkage. So implicitly, I will not be able to distinguish at the single cell resolution anymore, but I will be able to perform some kind of pseudo-bulking. So I will lose resolution, but at least I have some information. And the point is, can we try to do better than that? Okay, so this is a minimal kind of thing we can do. And so we need to estimate a matching matrix for doing this. And optimal transport in general is a really nice framework for learning matching, except that in general, optimal transport thinks about, or it's in most it's easier formulation, usually cares about matching within the same space. I'm going to first go through this for a few minutes. We have two probability distributions that live in the same space, mu and nu, and we want to know how to fit a coupling matrix, a pi, or a transport, or a matching, that will tell me how to move mass from one part of the space to another based on a certain cost. So if I don't care about the cost, the problem is defined as follows. I can pick any admissible matching matrix that basically matches, that corresponds to the right marginal distributions. I want to make sure I move all the mass from one place to the other place. But now, out of all these possible matrices, coupling matrices, I want to make sure that I find the one that minimizes the cost. So I can write it as an optimization problem, and that's usually how optimal transport is formulated. We obtain this optimal plan or optimal matching, and the value of the cost, the minimal cost, will be called the Wasserstein distance. And usually we don't quite run it like this. We will solve a regularized problem with entropy, and that allows for going into the dual space, making it strongly convex as a problem, and having much faster inference procedure. So this has been applied very widely to single-cell data and has been presented in this kind of venue many, many times. So I'll go through it quickly. This is not useful, again, because we cannot use that when it's across different spaces. So people have worked on the Gromov-Wasserstein optimal transport, which allows for matching across comparable spaces. The way that this works is basically, instead of having a cost function across two spaces that we do not know how to construct, we built a cost of cost functions. So basically, we look at the distance matrices within each space, and we try to minimize displacement of the cost matrices. And intuitively, what these kind of algorithms are doing, they're doing shape matching. They're trying to make sure that using the singularity in the shape, somehow we can kind of learn a decent matching. And so this has been applied also to single-cell data in the context of matching RNA-seq to ATAC-seq and other modalities like this when you have completely unpaired measurements, which looks like what we're trying to do here. But not quite, because we have some extra information, and that's the purpose of this presentation. So we're trying to get to the point where we can actually include information from the labels. And so that's what we dubbed label-constrained optimal transport. And the key idea is, instead of just doing naive shape matching, can we use the perturbation labels as anchors across multiple modalities? And in the context of optimal transport, if we look at the mathematical framework, maybe we can somehow just constrain the optimization problem to match samples that are only within the same label. So it doesn't sound too hard written like this. And that's our contribution. So we extended two frameworks for matching across modalities, Gromov-Wasserstein OT and co-optimal transport, for doing this kind of label-constrained method. So what we're interested in Fine is actually to understand, if we have a better matching, can we predict better across modalities, right? So I'll get to that throughout in a couple of minutes. So to finish, for this part of the talk, it's important to think about what's our setup. Our setup is, we have, again, two modalities. First, we will run this label-constrained optimal transport procedure that I haven't yet described. And then we will use that to learn a predictor and do the modality translation. All right. So now we can look into the specifics of how this label-constrained optimal transport can work. And so first, you might wonder, can I just split my data sets per label and then run number of label times a Gromov-Wasserstein optimal transport? And do I get the same even? And isn't it what you want to do? The key answer is no. So it's the optimal transport problem could break up like this, but Gromov-Wasserstein is a bit different, and this slide is meant to explain why. So if you only consider per label or per perturbation Gromov-Wasserstein, I'm showing you here two cloud of points, and the distances or the arrows that you can see are kind of like what you would do for the what you would compare across modalities for the Gromov-Wasserstein matching. And here it looks like the cell I have on the corner is kind of matching really nicely the distances of other cells picked in the other modality. So I would be very tempted to somehow put a high matching probability between those pairs of cells. But if I had other labels, maybe perturbations that affect another pathway or downregulate the same pathway, I'd be able to kind of figure out that actually that cell is very close to cells from another label in one modality, but very far from the same cells on the other modality. And in that case, if I just split the data set, I kind of lose the information. All right, I hope that makes it clear. If you look at the math, it's also clear, but I think that the drawing makes it nice. So now let's try to get into how the algorithm works and how we can quickly change it for solving that particular problem. So when you think about entropic Gromov-Wasserstein optimal transport, Gabriel Perret and other authors in their paper explained that you can solve Gromov-Wasserstein OT under some assumptions about the cost function that we will assume are fine here. You can break down this problem into a sequence of optimal transport problem across the same space by kind of changing the cost function. So you have a iterative dependent cost function, and you solve like this a sequence of OT problems. And this is equivalent to some kind of projected gradient descent. So you can write the algorithm like this, basically. The algorithm would look like I calculate a cost function or a cost matrix. I solve optimal transport with this synchron iteration. And then I go back, calculate the cost again, and perform synchron iteration and iterate until I converge. And I know I solved the Gromov-Wasserstein optimal transport. So this is for the unlabeled setting. And our contribution is just to say, all right, because somehow what we want to do is make sure that I cannot match things when the labels are different. I just need to do an extra projection. My problem is convex, so it's nice. My constraints are convex. I take the intersection of things convex, it's still convex. Things work out. And somehow, now I can just need to think about how do I do this extra projection. And it turns out we can still solve the OT problem with the same sinkhorn iterations. They're just slightly different. So you can see here we need to kind of like multiply by this matrix of compatibility that tells you if you're in the same label or not. That's doing the projection for us. And the cost function is also a little bit different and has a better runtime, but I'll leave it at that for this short presentation. We were able to implement, so Jiayong implemented this in the OTT JAX framework. It was really nice that we were able to work with Charlotte Bune because she implemented part of that package, and she's been a postdoc with us at Genentech and now started her own group at EPFL. We get this type of methods to work. So I'll present a natural baseline that everybody would try to apply to this kind of problem with a variational autoencoder. So, and we've seen, I guess, some of those models in different contexts maybe today, but you can use those autoencoders with different heads. So basically, to resolve this shape matching problem. So here I'm going to propose one way, but there could be many. You have two autoencoders that are trained at the same time, one for reconstructing one modality, one for reconstructing the other. And then you learn them jointly and force the bottleneck layers, or the latent variables, I would rather say that, but the language is fine. You use those bottleneck layers and you kind of force them to be undistinguishable in distribution from each other. So you could use an MMD, or here you can use an adversarial classifier. And if you train this jointly and you force things so that you cannot distinguish one modality from the other in the latent space, then you basically solve this problem. And it's been applied in recent papers working on also imaging and sequencing in a different context. So this is a nice baseline to try, but it's not label aware. The way to make it label aware would be to have a different adversarial classifier per label, which adds a lot of parameters. So to kind of avoid doing that, we just added the label of the one hot encoding of each perturbation label into the classifier. And it's crude approximation, but it's a baseline that we're trying to apply here to get a sense of how much better things get with adding labels. Right, so now I'm going to discuss our application to the real data. The bad news is we don't have real data of images and sequencing. Also, either way, it wouldn't be paired, so it would be tough to assess a matching algorithm. So I'm going to use a surrogate type of data. So this is data produced in the context of a collaboration with Professor Kelvin Shen at Osaka University. This is a chemical screen data set where we take T cells and activate them with anti-CD3, anti-CD28 antibodies, but in the context of small molecules. And the assay works like this. So we take those CD4 T cells, put different drugs, activate them, and measure protein and RNA sequencing. We filtered a few drugs that have the highest effect to test the method. We also have dosage information that's kind of helpful. It will come in later to visualize and make sure that the algorithm is behaving properly. And we're trying to predict gene expression from proteins. So I will argue that it's quite different from the original setting about imaging and sequencing, and it's much simpler, but it's a first step into evaluating this kind of method. So if you look onto, I guess, some kind of UMAP space, I'm plotting here different drugs and the gene expression space and protein space, and you can see that when you increase the dosage, things are moving along in latent space, which is kind of giving you the idea that the drugs are having an effect on the cells. I'll leave it at that for visualization. So the experiments we wanted to perform was, first we want to run the OT-based methods. Those methods are trained on the 50 PC latent space for both gene expression and proteins. We run optimal transport, Gromov-Achterstein optimal transport, and this other framework I haven't discussed called co-optimal transport. For all of them, we run the per label, no label, and label constrained. And then we train a multi-layer perceptron to predict the gene expression from the proteins using the same formula I've discussed. And then we also compare to the adversarial VAE. The architecture was based on multi-VI. And there are some hyperparameters you need to optimize. So for the optimal transport, usually the entropic regularizer is critical. So and for the adversarial VAE, you need to pick the adversarial weighting of the penalty in the elbow. And so what we did was folded 5-fold nested cross-validation, in which we picked the best result by splitting and average the results across all the folds on the test data, of course. And so to evaluate, we evaluate the matching because we have paired measurements, and we look at the fraction of samples closer than the true match. We also look at the dosage matching. So the dosage was not given to the algorithm. The labels were only the drugs. And we look at whether the model can figure out that cells from the same dosage should be matched to each other more closely than otherwise. And we look at how well we can translate those modalities, so based on fold change of gene expression and correlation also across cells, and the mean square error. So the results look as follow. First, we have the baseline, where we have the one-to-one mapping, the per dosage mapping, and uniform per label, which is what the algorithm only sees the labels. We can see that the coarser information as expected, and the worse the results get. We can also see that the entropic optimal transport, because it doesn't operate in incomparable spaces, doesn't perform well at all. But we see that the variants that we propose with the labeled data actually have a decent performance and are the best contenders in this study, both for Gromov-Wasserstein and Coyote. And the domain adversarial VAE performs decent matching, but has really poor performance for prediction, which means likely there are some. These methods are really tough to make work. There are a lot of not just maybe the lambda was not the only hyperparameter we should have tuned, and also the number of epochs you train when you train the adversarial classifier, etc. So it's but this is the first try. So as a conclusion, I would like to say that those labels constrained version of OT are interesting, and they improve prediction results for this cross-modal perturbation data translation project. What's important to think about is what is the input space for those models. So in general, you really need to, because we're doing shape matching, you really need to have the latent space in the right form, meaning that if somehow you have technical noise on the imaging and technical noise on the sequencing, those induce completely different shapes when you're going to apply those methods, things are going to fail pretty hard. And then the last thing I want to say is we're looking into amortization and neural optimal transport for increasing the scalability of those methods as well. And we're excited to apply them on this problem with real data of imaging and sequencing. And thank you very much.
Hey, thanks for the nice talk. I'm curious, going back to your original problem of mapping perturb-seq to self-painting, how do you compare your general class of methods to methods where you just featureize by aggregating over cells? Is there obvious evidence that sort of you need to model that individual cell level?
Yeah, so that's a good. I think that's a good point. I think if you start having enough perturbations and many contexts, maybe at some point you start picking on variation that will even be inside of the bulk variation of the control cells, meaning like cell cycle and all this stuff. So it's possible that with enough data and enough perturbation, you start getting into that. But I think the way I think about the single cell variation is that a lot of it could also be like confounding factor that could lead your algorithm astray when you're trying to make predictions. So the images across the cell cycle look very different, for example. But the genomics data as well. So if you can kind of condition on this and pick it up from an optimal transport like model, you might reduce your variance and have better results in maybe in a lower regime of less perturbations. So that's how I'm thinking about it. Yeah.
Hi, great talk. How would you suggest somebody scale it for multiple modalities, let's say like 4 or 5? Would it be just pairwise for each, or is there a more optimized method?
That's a good point. I didn't really think about this. So if you start having, so yeah, you could either do all the pairs. The way that would make more sense is to, once you start having many more modalities, is to think about some kind of common space, I guess. But in the context of optimal transport, I don't know exactly what it would mean. You need to define some kind of, that wouldn't fit in that exact framework. Maybe with autoencoders, I think people have already done these kind of things.
Okay, thank you so much.
Thank you.
No? Wait, okay. I took that photo, and I'm very proud of it. Wait, why is it not showing up? Okay. It's a little primitive, but let's start from here. Hey everyone, thanks for staying around at the end of these sessions. And as just like the last speaker said, I'm also not in sequence modeling. I'm going to talk about optimal transport. So we're going to talk about how do you infer trajectories when you have multiple time snapshots, and you could use this multiple time snapshots to actually give you some information about some underlying dynamics. I want to mention that Yunyi, okay, I'm Yunyi, forget to introduce myself. And this is a joint work with two awesome people from MIT, my advisor Tamara and Renato, our favorite Italian. And it's really nice to be here. So given we're all biologists, I guess it's very easy for me to state these motivations that we want to infer unseen trajectories. For instance, you can see cell differentiate, and you want to sort of understand how like stem cell become fibroblast, or stem cell become whatever other cells, or you are trying to understand how a heart cell become cancerous, like it started to become cancer cells, and understanding their transformation will hopefully give us some future understanding of how we deal with cancer, or how we maybe reverse the differentiation and go back to stem cell. So when we say transformation, what do we really mean? It's like you can probably, if you're in a Petri dish, you can probably see the form change. But in at least in this talk, we'll say this transformation are measured by their gene expression, and then further measured by, say, mRNA level change over time. And then in this kind of systems, like stochasticity is one of the inherent part of it. Like after all, they are chemical reactions. Like two molecules has to collide together to make another molecule or something like that. So stochasticity is just inherent in the systems. And that's part of the reason why people tend to use stochastic differential equations as a model. It's not the model, but at least is a model for this type of systems. So this will boil down to how we infer SDEs, or how we infer a solution of SDEs. However, in our setting, how we measure mRNA level make this kind of inference extremely hard. For at least two reasons, at least it's not only these two reasons, I believe. So first of all, we don't really have continuous time measurement. If you want to use the traditional or most classic inference method on SDEs, hopefully you will get the entire functional measurement, but we don't. We probably 6 time points is already luxury. I do see a poster with probably 20, but that's super luxury. Sometimes only 2, and there's a still thing you can do. And the most another probably even worse situation is we have to kill the cell. So if you see what's inside, you have to kill it. Then you never see one cell twice. That's a curse and a blessing in some situations, but that's definitely a problem. So how do we deal with it? It's definitely not a new problem, and people have thought about it for a while. At least one of the current solution is the so-called Schrödinger bridge. It's a optimal transport. It's not the optimal transport, but it's a optimal transport solution. It's a dynamic optimal transport regularized by entropy. So it's an entropy regularized dynamic transport problem. And however, there are some problem with it. First, the most naive Schrödinger bridge is an optimal transport only between 2 time points. And actually, if you think about diffusion models, it's one of the special cases, starting from pure Gaussian noise to your target distribution with only 2 time points. And the other problem is, as we just discussed, it is an entropy regularized optimum transport. The regularization is by entropy. And another way of stating that is trying to find trajectories that pass through the two distributions, but most close to a Brownian motion. So you need to choose such a reference measure, usually chosen as a Brownian motion, but you can choose other things. But still, you need a single point reference. This could be more than a luxury for a biologist. We can think about, like yesterday we heard about a perturb ODE talk, which is awesome in Spotlight. We can think about, okay, I maybe want to use that as my reference, but there's no way for me to figure out every single parameter out of blue just before I do anything. So that's too much. And using Brownian motion is a default, but is Brownian motion a good reference that you want your trajectory to be most close to? Not necessarily. So what we do here, not only us, and apparently I figured out yesterday, Stephen, I don't know where he is, he also come up with this idea, and we almost did it at the same time. So we provide a extension of Schrodinger bridges. We handle multiple time snapshots, and we choose the reference on the fly by iterative procedure, not only fix that reference in the beginning, so that this method could interpolate, and it's beyond piecewise interpolation, and it showed better performance in both simulated and real data. So let's think about the setup. We all okay with me saying, we never have continuous time measurement and we kill cell. But what that really means mathematically or schematically is, let's think about we don't sequence at all. So if we don't do any experiment, and each cell will has, for instance, you have only think about 2 genes. It has 2 each cell will have a trajectory, like from this expression level of gene 1 and gene 2, and eventually it goes to these places. For instance, it's a differentiation. Might go to another cell type or become cancer. I only show 6, but in data, hopefully we have more than 6. And to see the first batch of sequencing at time step 0, we kill the first 2 trajectory. We see these 2 cells. And we'll never see the trajectory ever again, because they're dead. And then in the first time step, you see another 2 cell, and you kill the trajectory again. You never see it once more. And then in third, you kill it again. So if you think about it, just look at this figure, you probably could tell, yeah, there is some curvature there. So our goal is really to infer these trajectories you didn't see, or actually you will not be able to see because you kill cells. But we infer these trajectories or sample these unobserved trajectories. More practically, beyond schematic, what we do is we have to still tell that whether we work really well is we would retain some validation time steps. For instance, we have 5 training snapshots here, and we would retain something in between to see if at least our infer trajectory would pass through this retained data. And we would say we'll say we're successful if we at least see our trajectory pass through this retained thing, and we do some numeric measurements on that. So this would hopefully be a successful example, that we infer these trajectories going with this curve, and these points are retained as validation. I probably get it wrong, but we retain some validation data and to see if we pass through it. So as we just had a talk about optimal transport, let's have a quick review of Schrodinger bridge as optimal transport. So it is a optimal transport. It's a dynamic optimal transport. And the intuition is you want to find those paths that pass through the 2 distributions, if you start from the most naive Schrodinger bridge, 2 distributions, and most close to a reference measure, which is usually chosen as a Brownian motion. Then if you chose Brownian motion as a reference, as entropy regularized. But the issue is, first, you really need, if you have multiple time steps, you really want to use them. And actually, people shown that if you do it naively, it becomes piecewise interpolation. Like multiple marginal Schrodinger bridge is essentially doing it piecewise in consecutive pairs. And then it requires a fixed reference point, usually as Brownian motion. Actually, I took some time trying to figure out why it is a Brownian motion, and it actually dates back to Schrodinger, because it's called Schrodinger bridge. And his original problem is, okay, I see a whole bunch of gas molecules and move from one configuration, which is not uniform, and to another configuration, which is also not uniform. So how do these gas molecules move? In their situation, Brownian motion is a correct reference because it's gas molecules moving under thermal fluctuation. They are moving according to Brownian motion. However, if Schrodinger himself is here, he would say, don't use it for biology systems, because we are not in a closed system. We have energy intake, and we don't really respect entropy laws. And he himself even wrote about it, saying what organisms feed upon is negative entropy. Like, we don't respect those. But beyond that, beyond this optimal transport viewpoint, there's another useful viewpoint is it is also a projection method. Think about your data or all the possible trajectories that pass through your data distribution, live in a family. And you start with a reference family, a single reference family. And then Schrodinger bridge solution is a projection, is a specific type of projection to the family that passed through all your data. This is where we start to think about how we would extend it. So let's still start with a family that we ask this family is the family that all possible dynamics that pass through all your marginals or snapshots. And then Schrodinger bridge would be, let's start from a single point. Let's find that projection, and that's our solution. So what about we start with a family of reference? For instance, I really enjoy that spotlight, so I'm going to use that as my example. Your perturb ODE as your model family. And we choose the initial point. It could be Brownian motion if you move all the dynamic parameters to 0. Then we could do this first step Schrodinger bridge. It's the usual Schrodinger bridge. Start from a reference, go to your family. That's our first step of iterative procedure. You gave the current reference, find the Schrodinger bridge solution. But we can move back to our reference and find the next better reference point by projecting back, which we showed this is basically boiled down back to a regression problem. And then, after finding this better reference, we can do our Schrodinger bridge again and do that until until we're satisfied or converge. Sometimes they converge. It might not necessary, but until we're satisfied. And by these iterative minimizing, this distance, actually measured by KL divergence, will always be reduced. So we have to test it in practice, in both simulated data and real data. I will show some simulated data here, but feel free to check out, or please check out our paper for some real data situation. So we compare with two baselines. The vanilla Schrodinger bridge is you just do piecewise interpolations. And one thing called deep momentum Schrodinger bridge by Chen et al. In 2024. Their idea is basically, let's think about my particles, single cells, to have inertia, so that it will overshoot a little bit. Let's think about not only position, but also their momentum. And the metric we use to test success is first visual representation. It should at least pass through your left outs. And we also measure Wasserstein 1, which is also called Earth movers distance, between the samples of our inferred time step, inferred trajectory at each holdout time step, and our holdout data distribution. Well, let's went over this first set of experiment. So our method is in the middle. Hopefully you can see my mouse. And these are holdout data. Every other steps are holdout. So our method seems to pass through most of the holdouts. And if you only do piecewise interpolation, the method will be, oh, sorry, those people cannot see it. But the naive Schrodinger bridge doing piecewise interpolation, they will just overshoot. It will miss the second point because it just doesn't know there's a curvature there. DMSB sims did reasonably well because you have this inertia behavior that it will overshoot a little bit. But this is not the end of the story. We also tested, this is one example, as probably if you're a systems biologist, you're very familiar with this example as one of the minimalist example of a biological clock, having three mRNAs that suppress each other, and then you will see this rotation behavior. So this is a very good test bed because it has rotation, and if you do, if your method is unaware of these curvature behavior, there's no way you really do it. So our method sims being able to sort of infer that we have this rotations because you have multiple time steps, so that by having this interpolation, well, by choosing the best reference, it learns the curvature and was able to pass over this holdout site. And similar behavior as the last example I showed, if you do this vanilla piecewise interpolation, it will miss all these validations because it's just piecewise connecting. And does the deep momentum thing did better? It's not piecewise connecting anymore, but still, it's still after all, it's a Schrodinger bridge. It has this Brownian motion reference and is still trying to connect dots. And for a summary, that we propose a new method based on Schrodinger bridge that first learn the unobserved trajectories from sample snapshots across multiple time points and requires a family reference instead of a single point. And we validate the experiment, we validate our method with a simulated and real data experiment. Since our method outperform our baseline both in visually and in Wasserstein one. And please check out our paper. And if you have further questions, feel free to email me. And I'm looking for interns if you want. Thank you. Internships.
Questions?
Thank you. That was a really nice talk. I was very interested in, maybe you have explained and I just didn't get it, but what is the class of model that you're considering when you do your refinement in your experiments? And how do you design these kind of models? Like how much biological knowledge can you add? Yes. Can you transfer this across data sets? I mean, if you had some different data sets, or how much parameter sharing could you somehow get from that? Or, yeah.
Yeah, that's a great question. So I'll answer it as two parts. So first part is how we choose the reference, and second part is if we have multiple data set, how do we share? So first part is harder than the second. I would say for the real data experiment in our paper, it's cell differentiation, so we end up using a gradient field. That's because of Waddington's landscape. And if you have better sense of what the systems look like, that's why I always use the example of the perturbed ODE yesterday, because it seems to be a good choice, or at least a starting point. I don't think I have a very good D file to give you, more than yeah, you use your biological knowledge, and I hope there is a better way to choose this reference, but at least it's not one single point. And to the second part, I would say the first step projection, the Schrodinger bridge, once you fix the reference family, you can do it separately. And then the second step projection is essentially a regression problem. Then you just pool the likelihood, then should be fine.
Okay, let's thank the speaker again.
Thank you.
He took away. Oh, yeah. Great.
So hi, everybody. My name's Hao. Oh, yeah, great. My name's Hao. I'm a final year PhD student at Tufts. I think we're a little bit behind time, so I will try to speed up a little bit. So today, I'm going to introduce you to reg diffusion. It's a new class of a diffusion model to do very fast gene regulatory network inference from single-cell data. So gene regulatory network, so gene network, sometimes we call it, is a kind of directional network we use to describe how genes place with each other. And there are many ways to get to a GRN, but in this talk, we focus specifically on just inferring GRN simply based on gene expression data. This field has been actively researched in the past 30 years. We have seen a lot of methods being proposed, ranging from Bayesian network to decision trees, and also Bayesian networks. And in recent years, we are exploring this option on using deep learning methods to speed up this process. And on the other side, on the source of data, we also see a lot of advancements, ranging from microarray to RNA-seq and to single-cell RNA-seq. So one of the problems we're facing today is like a lot of these network inference algorithms, they are very computationally extensive. And right now, today, we have exposed ourselves to a lot of data with a lot of genes and cells. These algorithms, they don't really scale that well. A few years ago, we had this method called DeepSum published. Essentially, this is an autoencoder-based structure equation model where they are trying to reconstruct the gene expression profile under the constraint of a parameterized agency matrix. People find that it's outperforming other methods based on the B-line benchmark, and it runs much faster than for example, like decision trees methods like Gini3. And one of the previous work that we have been doing is like we proposed a method called dropout augmentation inside this model called DESL, where we are trying to solve this, the dropout problem in single-cell data by introducing some extra amount of dropout noise. And we find that it's actually helping improving the model's robustness and stability. Before I move on, I would like to point your attention to this highlighted area where to model this agency matrix, we have to use this agency matrix on both sides of the autoencoder, both the encoder and decoder. So on the decoder side, we actually need to do this matrix inversion on the agency matrix. Unfortunately, this matrix inversion step, it's usually solved by Gaussian elimination, and it runs in cubic time. That says that if we have a single-cell gene expression data with N cells and M genes, the runtime will at least be n times m to the power of 3, right? So in this talk, we're going to show you how we actually managed to reduce the runtime all the way down to m squared. Again, we are using this diffusion model. I'm very excited to see so many diffusion model talks this afternoon. So basically, just a recap, a very quick recap. Diffusion model is widely used in those image generation apps, and it has two processes. In the forward process, you're gradually added some Gaussian noise into the image, and in the reverse process, this is actually where the modeling process is happening. We're trying to predict the added noise from the noisy input. So as you can imagine, the reverse model, where we create the model, it takes two kinds of input: the noisy image and the diffusion time step t, which kind of tells the model how much noise should it expect. So the reverse model's objective is to predict the added noise, such that if we want to get the original image, we can simply subtracting the noise from the noisy input. In computer vision, people usually solve this with UNet, but we actually don't have to do this, as what you will see in this talk. So in reg diffusion, we solve this problem by making this a linear additive assumption. Essentially, here, imagine we have 4 nodes, A, B, C, and D. And here we use X to denote the log of the probability of seeing A, B, C, and D. Okay? So the linear additive assumption just simply say that the XA will the expected value of XA will simply be the weighted sum of XB, C, and D. And because we normalize the data, we want to make sure all these values are at the same scale. We assume the expected value of the weights to be 1 over, well, it should be minus 1. Yeah, 1 over the number of genes. If we write this equation in the matrix multiplication form, we have this very simple equation, x equals to xa. This is a matrix multiplication, and it's a very typical structure equation model equation. So then we ask this question: what will happen to this equation when we introduce some Gaussian noise? So we did some math, and we are very happy to discuss at the end. But here's the conclusion. So if we use x prime to denote the noisy input, then x prime a, well, when your expected value of the weight is small, then x prime a will be, in fact, be a close approximation of the original input x. So given this assumption, given this conclusion, we can see that if we want to calculate the added noise, we can simply subtracting this x prime a from x prime. Remember, in the diffusion model, I mean, those vision folks, they are using UNet, but the goal is to predict the added noise. This is actually exactly what we need. Here's an overview of the RAG diffusion workflow. As you can see, we start with raw counts. We did some pre-processing, and after some pre-processing, we get this x0, and we send this x0 to the forward process to noise the data, to noisify the data. And we send the noisified the data, xt, in the gray box, together with a diffusion time step t into the RAG diffusion model to generate the prediction of the added noise z prime. And the benefit of what I have been talking in the last 2 slides is that, as you can see right now, there's no need to do the matrix inversion. Here's an overview of the actual model architecture. It looks a little bit complicated, but it's in fact just a series of MLP. So we start with feature embeddings, MLPs, do some matrix modification, and then linear prediction. So no UNet, just MLP. OK, so that's the end of the model architecture. And to validate our idea, we tested it on three data sets. We tested on the B-line single cell benchmarks, which itself has like 7 small, fairly small single cell data. But it also comes with 3 sets of ground truth networks. And we also tested on 2 microglia data sets at different sizes. The first one, which we call it Hummel microglia, is from the Best-Stevens lab. And it's these microglia cells, they are experimentally obtained. And the second one, I think, is from Bard. It's an Atlas experiment, and all these microglia cells, they are labeled by marker genes. And OK, on the right side is a evaluation metric, but basically the higher numbers means better performance. So RAG diffusion actually does great on the benchmark, but what really excited us is its runtime. And as you can see on all these B-line benchmarks, which I listed the size of the input data at the bottom. Most of them, they have roughly like 1,000 genes and 700 cells. Genie 3 took roughly like a little bit less than 3 hours. And DeepSam, the ensemble version, takes roughly like 5 minutes. But the RAG diffusion, it takes usually under 10 seconds on a GPU. And on the right side, you can see its performance on the Hummel microglia with 15,000 genes and 8,000 cells. The DeepSam ensemble version, it takes more than 40 hours GPU time. And we use a yellow bar to denote the compute time of RAG diffusion, but it's too short that you can't really see it. It's 3 minutes. So the source of speed gain is not only come from the elimination of the matrix inversion, but also it's because we actually use the fixed size bootstrap sampling. Basically, our single cell data, it comes with all kinds of sizes, like different studies, they have different number of cells, right? It's a little bit unfair to send all this data and let the model see all the cells and still train them at with the same number of steps. So so here we just simply we sample a fixed number of data, a fixed number of cells, and send them and let this thing go through the training network in each iteration. And then by observing the amount of changes in the adjacency matrix, we use that as a way to help us identify when the model actually converge. And what we find is that with the current setting, with the current default setting of regular diffusion, regardless of the size of the data, no matter it's small benchmarking data or the real data, the bigger data we use, you always follow a very similar converge curve. So there's no need to see all the data in each iteration. Here, as I said, regular diffusion actually does great on its on the actual prediction performance. There are two tables under different two different metrics. You don't have to read all these numbers, but in both of these two tables, regular diffusion is at the bottom row, and we use colors to indicate the top performer or second performer. It's always, in most cases, it's always the best or the second best. Okay, now let's see some interesting biological results to inspect the actual networks that's getting inferred. Remember, in this study, we tested it on two microglia data, one from experimental obtained microglia and one from others with marker genes. And here what you're seeing is a two-hop neighborhood around ApoE, which is a very actively researched AD genes. And we use these colored ovals to mark out confirmed regulatory relationship relative to ApoE. Okay, so and you can see there. So there are, I mean, a lot of these links. I'm not saying like all of them, but a lot of them, we actually can find biological evidence. And another interesting finding is like if you compare the results, compare these inferred networks from ApoE from two studies, we can actually see the one using marker genes is in fact very coherent from these bottom left regions of this inferred network from the experimental obtained regions. So our theory is like, because microglia itself also has a lot of subtypes. And if you just simply use marker genes, it will only help you identify those major microglia subtype. So you basically throw away all these minorities in microglia types. Before I move on, I would like to point your attention to this green oval, the cathepsin group. Cathepsin is a kind of protease whose responsibility is to break down unnecessary proteins. In this case, in the microglia case, because microglia is one of those immune cells, right? So it's perfect to make sense to see the group of cathepsins like interacting with basically this gray area is microglia core genes. It makes perfect sense. And at the same time, we also find cathepsins in some of those deep stem benchmarks, like for example, the mouse embryonic stem cell. And in mouse embryonic stem cell, we find that cathepsin T, for example, is interacting directly with the gene BTG1 and 2. BTG1 and 2, after we did some research, they are actually anti-proliferation factors. So as you can imagine, these factors they help the embryonic stem cell to eliminate unnecessary like overproduced product when they actually grow. So our conclusion is that regular diffusion, like when you apply it on the right data, it actually help us to capture different kind of very cell type specific regulatory strengths in different kind of cells. So here's our conclusion. Regular diffusion is very fast. It's very suitable for large scale data. And many of the links in the inferred networks, they are biological verifiable. And at the same time, it also comes with some kind of some limitations. So we gain a lot of, we have a lot of speed gain in rack diffusion, but it comes at the cost. I mean, there are no free lunch, right? It comes at the cost of the losing of the ability to do representation learning. And also, I mean, so far it's not capable to infer latent confounder node, just unobserved node. Also, at the same time, because there are a lot of feedback loops in biological system, we are not applying any DAG control at this moment. So if you check the network, you will see there are like a little bit too many like feedback loops. So we're actually exploring like possible options to do this kind of DAG control, but not entire DAG, maybe like few hop DAG control in a very in a better way. Yeah. And oh God, I forgot this. So, and we actually spent quite some time on developing a software interface for this. So this package is available on pip. It has a very simple interface. You can simply load the B-line benchmark, start a trainer and do the training, and export the inferred networks, and even generate some visualizations you just saw using one line of code. And finally, this group picture. As I said, I'm a final year PhD. I'm looking for postdoc positions, hopefully in Boston area.
Questions?
Hey, good talk. In terms of predict performance, have you thought about comparing with benchmarks on the basis of false positives and false negatives?
Yeah, FDR, and this is totally a valid metric. So far within the paper of that, we have not yet done that, but recently we're in some recent work we are starting to use this metric.
Okay, cool.
Well, I actually have some actual comment on that because one other thing I realized when we run all these kind of experiments, like our ground truth data, there are so many false positive and so many false negative. It's just hard to validate. Yeah.
Okay, thank you so much. All right, this ends the conference, I guess, officially. Thank you so much for the submitters, for the presenters, and the audience for such great questions. Hope to see you again in 2025. We'll be sending a survey, getting your feedback, and checking which locations you prefer. So make sure you respond to our survey. Thanks again for being here.